So the reason we started working on iron in cardiovascular is we got a bit more to do that circles. And then later on, we found that iron is a major area of research in cardiovascular disease, so we expanded that to other aspects of heart disease. And that's what I just thought maybe I should tell you about what we know about heart disease, and we'll briefly touch on the role of iron in regards to its iPSC. And the reason I want to do that is just to tell you how much emphasis has been put on iron in various forms of heart disease, especially heart failure. And there is one area I would say that there is, as you know, there is a lot of there's the gap between basics and clinical science in a lot of areas of medicine, but I think one area that this gap is very wide is, of course, the neutral iron heart failure. And iron in cardiovascular disease is now a billion-dollar industry that you can see right now. It's more than $2 billion. It's going to go up to probably close to $4 or $5 billion in the next day. And these are the guidelines. The European guidelines actually give us a class 2A indication for patients with heart failure. It gives indication to have iron deficiency, and the evidence of EFRA. They give you the class 2 indication. American guidelines are more conservative. They give you the class 2B indication. These guidelines from the European Society was European Cardiologists, or Society of European Cardiology, was valid until last year. And last year, in August of 2023, the guidelines changed. Now they give it a class 1 indication for patients who have heart failure and they have iron deficiency. And they give it the level A evidence for symptomatic resistance. What I'm going to do, I'm going to tell you that this is probably a little too aggressive, and this is not really what you should do in our meeting. And over the past few years, we have published a number of papers, both in clinical journals, and including, have also been hospitalists, and also basic science journals arguing that giving IV iron to patients in cardiovascular disease, especially in heart failure, based on the criteria we have, is probably not a very good way of moving forward. So let me tell you about iron deficiency. There are two forms of iron deficiency. One is absolute iron deficiency, and that means there is not enough iron in our body, right? There are a lot of young women in this room, and I bet you there are several, many of us, well, maybe not me, but women who are iron deficient in this room. But there is a different form of iron deficiency, and that's called functional iron. Why does it happen? That's when we have enough iron in our body, but it's not available. And the reason for that comes from an evolutionary aspect of biology, is that we withhold iron from bacteria. This is one way for fighting bacterial infection. Bacteria needs iron to grow in our body. So one way to fight bacteria is to withhold iron from them. So in any form of inflammation, iron levels in the body, available iron in the body goes down. And where does it go? It gets sequestered in macrophages, it gets sequestered in the liver, but it doesn't become available. So that's when we have functional iron deficiency. I'm sure you have heard of an immunotherapy disease. That's a consequence of functional iron. And that's basically, that's what happens in a lot of diseases. So the argument is that in heart failure, there's this chronic inflammation, and that's why these patients suffer from functional iron deficiency. But the problem is that there are studies that have been done in these patients, and hepcidin, which is a peptide that is produced to absorb iron, is actually not increased in patients with heart failure. As you can see here, these are some of the papers we published with Peter Polikowski, and we show that in fact, if anything, hepcidin goes down in iron deficiency. Now, a lot of diseases like cancer, a lot of diseases like rheumatoid arthritis, if you look at those patients, hepcidin goes up because they have systemic inflammation, and because of that, hepcidin is decreased. So, what are the criteria that American Heart Association and also European Society for Cardiology used to define iron deficiency? It basically says serum ferritin levels less than 100, or ferritin levels between 100 to 300, but T-cell less than 20. These are very loose criteria. I bet you probably 90% of us in this room fall into these categories. And we have, for instance, if your ferritin is above 300, it is basically suggested that you may have a chronic inflammation. And that may be associated with elevated CRP. So that's probably not a good thing to have very elevated ferritin. But the absolute iron deficiency is defined when the ferritin levels are less than 15. So you see the discrepancy between 15 and the numbers that are used in this right here. Where does this criteria come from? From chronic kidney disease. And again, chronic kidney disease is associated with some form of chronic inflammation, and so that's different from heart failure. Now, I'm not going to go over this, but there's a lot of evidence from epidemiological studies that the lower your iron, the lower your hemoglobin, the lower your risk of having cardiovascular events. And there's also evidence that patients who are blood donors, they have a lower risk of having cardiovascular disease. I'm an avid blood donor. I give it during the stable diet that you cannot live more than 3 months. Every 3 months, I try to give it every 3 months. And my ferritin was 11 because I was using so much blood. And again, that means I have an absolute iron deficiency. And because of that, I actually was getting restless leg syndrome. And I started, the aspirin, I didn't blood anymore because of my low ferritin. But I think one of the reasons I really like to drink blood is that afterwards you get the chance to eat all this junk food that my wife could never allow inside the house. So that wasn't a bad one. So it was a major motivation to do blood. So what do we know about the IV iron in patients with heart failure? There are several studies that have been done. The 3 largest ones are FACC, iron exam, and also this study that came out at the same time in, sorry, I turned on the point of the screen, but this study came out last August at the ESC. And I don't think you can see, you can get 2, graphs better superimposed on each other than you see here. So basically, IV iron didn't meet the primary endpoint in any of these studies. The primary endpoint wasn't met. So there was no evidence that IV iron actually improves outcome in these patients. However, the secondary analysis suggested that there may be a reduction in symptoms and also in the heart failure hospitalization in this patient. But that was secondary analysis in this study. And again, this is the FDA label is updated, but it used to say that it's given to patients who do not tolerate oral iron and patients with chronic kidney disease. So every time we give it to patients with IV iron to patients with heart failure, that's off-label usage for IV iron. Now, I've told you about IV iron. Let me tell you about oral iron. Oral iron works really well. The problem is that sometimes has side effects, the GI side effects of oral iron are clear. So this is how we have learned how to absorb iron. When we have enough iron, we have sufficient iron in our body, there is this peptide that is produced. And hepcidin basically goes and binds to the ferroportin in the digestive tract, and it's also in the macrophages. And it basically reduces or causes degradation of ferroportin. So you don't absorb iron into the body, and iron is not released from macrophages. So this is how it works. If you have enough iron, no matter how much iron you take, how much oral iron you take, you're not going to absorb it. That's why it's very unlikely someone will get iron toxicity by thinking about iron doses. That basically doesn't happen. But when you're iron deficient, hepcidin is not produced, so ferroportin is intact, so you take an iron into your body. So if you're iron deficient, you take iron, you are going to absorb iron because there is not a mechanism in our body to prevent it. So the reason people say that oral iron doesn't work is because of this study called Iron Out HR. And it basically showed that it was oral iron versus placebo, and it basically showed that there was no difference between oral iron and placebo in peak VO2 in this patient. But again, there is no evidence that peak VO2 also improves with IV iron. So that suggests that this approach or this argument that oral iron doesn't work is not really valid. And indeed, studies that compare oral iron versus IV iron. And again, remember, IV iron has side effects. Throughout evolution, 4 billion years, or 2 billion years, they have faced iron deficiency. I'm going to talk about that a little bit. we have never seen the role of iron going into our systemic circulation. This is the first time. Every time you need iron, it goes into our digestive tract and gets absorbed. So what are the side effects of iron? Iron, there is evidence that possibly increased incidence of cancer, which makes sense because you are basically giving nourishment to these cancer cells to grow. There's also possible evidence of acute kidney injury, evidence of endothelial cell damage, and also including in that there's evidence of increased infection. And this effect of osteomalacia is real, and it happens with the first carboxyl. We don't have as much, at least I know in Illinois, we don't have first carboxyl because we don't use it as much in the US. First carboxyl because it's used a lot in Europe. In the US, it's very expensive, so it's not popular. It's not popular in our hospital, but the Europeans use it a lot. It has a major side effect of osteomalacia, and the basic science behind it that's been discovered is because of Porgosha and CHIP23. And the scientist patients actually have to be in their list. So that's one of the side effects. This paper came out the year 2 years ago, and they looked at all that analysis of all the patients who have received IV iron, and there was evidence of increased infection. Again, we give iron to patients. Of course, one of the side effects is going to be increased infection in these patients. Now, we are working with just something and our goal is actually to look to see if there is a reversal of ordinary disease if you give iron chelator. So we are taking a different approach. We believe that endothelial cell damage can happen because of too much iron in our circulation. So if you give iron chelator, you may be able to reverse the progression of coronary artery disease. I don't know if any of you know Maedeh is one of the best CT angiograms on the coronaries in the country, probably in the world. And just agreed to help us to this analysis. We're going to give oral iron chelator patients and look at their coronary CTA over a period of 12 months. And we'll see if there is a reduction in reduction. Now, so that's basically an overview of the clinical work that has been done in the June-Wha and cardiovascular disease. What I'd like to do now is just to give you a summary of what we have done in our lab. We have published a number of papers in this area, and we have shown that if you look at the iron in the heart itself, if you reduce iron in the heart, it's actually good for the heart. Most of our work, because all of our work has been done in mice. But I just want to give you some of the work that we have done and tell you why our studies suggest that there is actually iron in the heart. It's actually beneficial to the age. The first area that I'm going to tell you briefly is mitochondrial iron, and that's where we started with, and that's how we got into the field of toxicity, which are the toxicity. I'm going to tell you about the novel pathway we have discovered that is involved in iron regulation inside the cells. Okay, so you have to remember hepcidin is involved in the systemic cellular regulation. So in this, I'm talking about cellular regulation of iron. And also, I'm going to talk about the novel pathway we discovered, which is involved in iron static by the cell. So, again, I'm not going to spend too much time going over the details of this study. I'm just going to give you a summary of what we found. When I started my lab, I was very interested in this protein called the ACPA, and we didn't know what this protein did, so when we started working on it, we made it out whilst. To our surprise, we understood that this protein is involved in iron regulation in the mitochondria. That's not what I was, what I was working on, but this fits with how it comes to June-Wha iron homeostasis. And we showed that when you knock out iron accumulates in the mitochondria, and that leads to cardiomyopathy. So again, if you accumulate iron in the mitochondria, it's added to the part. And the mechanism, and this is the cartoon of what I just told you, that ABCD is involved in iron export in the mitochondria. So when you knock it out, iron accumulates in the mitochondria. And iron is a source of reactive oxidation. Okay? Why does it happen? Because of the Fenton reaction. Fenton reaction is basically said that when you have hydrogen peroxide hydrogen peroxide is actually not a very toxic molecule. They put hydrogen peroxide in black right? But if there is iron around, iron converts the charge oxide radical. Oxide radicals are the most toxic reactive oxygen. So that's why hydrogen convert back to oxygen, they should feel any toxic. So that's basically what we found that iron accumulates in the mitochondria, this reactive oxygen, which is the production. Now, we also studied whether or not, so far what I showed you is that if you increase mitochondrial iron, that can lead to damage to the heart. But if you reduce mitochondrial iron, then you protect against heart disease. And we have done it in two areas. One is linear perfusion injury, and the other one is cardiovascular cardiomyopathy. So let me just tell you briefly about this story behind cardiovascular cardiomyopathy. This was published probably more than a decade ago. But we basically treated on mice with doxorubicin. We showed that there is an increase in myocardial iron exchange. Okay, and then when we treated on mice that had overexpression of ABCDA, that means they had reduced myocardial iron, they protected against doxorubicin-induced cardiomyopathy. And also, as you all know, dextroxyzine is the only vaccinated approved drug for doxorubicin-induced cardiomyopathy. If people ask you how does dextroxyzine work, many of us would say, well, we don't know the answer, but it's actually an iron inhibitor, right? And people have been also having works with a very strong iron stimulator, but for us, it doesn't work. And we showed that the reason that dextroxyzine works is because it can reduce mitochondrial iron. For us, it cannot get into the mitochondria, it cannot reduce mitochondrial iron, but dextroxyzine can. So that's basically what supposed to be the mechanism for this slide. So this is basically studies in mice that we show that dextroxyzine can reverse the toxic effects of doxorubicin. We also obtained heart samples from patients with doxorubicin-induced cardiotoxicity. We showed that mitochondrial iron is significantly increased in these textures, while stenocytic and nuclear iron is not decreased in patients with doxorubicin-induced cardiac. So what I've shown you so far is that if you have a normal mouse, if you give them doxorubicin or if you subject them to ischemic perfusion, they get damage to their heart. But if you find a way to reduce mitochondrial iron, then these mice are protected against doxorubicin-induced cardiomyopathy. They are ischemic usually. So that highlights the importance of mitochondrial iron in the heart in various forms of cardiovascular. Now I'm going to now tell you briefly about this pathway we discovered with tristentralproline being involved in iron instead of iron regulation. This work was done by this really bright MD-PhD student out of Florida, Beyer. And what Muredach discovered was that this protein called tristentralproline. So tristentralproline, the name refers to this protein because it has 3 regions of 4 terminals. So tris is 3 tetralproline. What this protein does is that it binds to any regions, its P prime only has like a region of that RNA molecules and causes degradation of the RNA molecules. This protein was discovered in the 1990s by Blaes. He knocked out the gene in mice, and he surprised the mice got systemic inflammation. As you can see here, they got dermatitis, they got conjunctivitis, they got arthritis. It was a very highly significant inflammation here. And the reason is because these mice have increased levels of TNF. So one of the targets of TTP, so remember TTP causes degradation of mRNA molecules. One of the targets of TTP is TNF. So when you remove the TTP, TNF levels too, and that's why this mice got negative inflammation. So since this discovery was in 1990s, almost every work on TTP was in the field of inflammation. What we discovered, this was again published a decade ago, what we discovered was that the standing of the iron, mTOR gets inhibited, and as a result of that, TTP gets activated. So this is the double negative. So this low iron actually causes activation of TTP. And what TTP does is that it goes and binds to the 3' UTR transferrin receptor 1, and it causes degradation of transferrin receptor 1. So this iron gets into the cell. Now, this doesn't make sense, right? You have low iron, and why do you want to bring less iron into the cell? If you have low iron, and you want to bring more iron into the cell. And the fact is that that's the job of IRG proteins. IRG proteins have been discovered through several methods, and they have been shown basically to go and bring more iron into the cell. This pathway is activated in severe iron deficiency. And what it does is that it causes iron insulation inside the cell. So you just don't bring iron, keep bringing iron into your cell. When you're iron deficient, you want to conserve that iron for your brain and for your heart. It's okay if your muscle doesn't get iron. You're going to be young, you're going to get tired, you're going to not feel good, but at least you're protecting your brain and your heart. And that's why there is this sophisticated system in place, severe iron deficiency, to make sure that you can break on the system and you don't keep bringing more iron into the cell when other organs are that's why they do get more iron. So that was basically, that paper was published more than a decade ago. We just worked on this area of work on iron, and that's what I want to focus on for the next few slides. And that's the area of iron space. So the area of nutrient sensing has become a really big area of research. A lot of work has been done on oxygen sensing. As you know, the Nobel Prize was given 4 years ago for the discovery of recent carbohydrate sensing with kinases, digestion sensing with Nator, and so we will push the Nobel Prize. Who gets the Nobel Prize for their discovery? So this area of nutrient sensing, meaning how do cells sense when there is not enough to understand the environment, has become a very interesting area of research. But there isn't much done in the field of iron sensing. And because of that, we have been working on trying to identify a common iron sensor that regulates cellular anabolics. What does it mean? So let me explain that to you. One of the MD-PhD students who joined my lab, Jason Shapiro, when he came to my lab, he said, I don't understand why cells, when they are, when there is low iron in the environment, they stop replicating. Right? So when there is not enough iron in the environment, cells just stop replicating. What's the mechanism? At least I said, I don't know. So that's why he wanted to work on this and figure out the mechanism for it. And one thing he mentioned again, if you look at it, almost every use for iron in the body is for anabolics and growth. If you have enough iron in the environment, your cells are going to grow. Yes. And then the other thing is that if you look at the evolution of iron's usage in your body, or iron that they use, the Earth was created about 4.5 million years ago, and surprisingly, life came to Earth very early after that. It's in 2 billion years, 4 million years we had the life started on the Earth. For the first 2 billion years, there was no oxygen around. And as you know, the most basic element of life is electron formation and acceptance. In fact, evolutionary biologists believe that's the first thing that happened, electron formation and acceptance. So what did we use for electron formation and acceptance when if there was no oxygen? We used iron and we used sulfur. So those were the two major forms of electron formation and acceptance. 2 billion years ago, something happened, and that was the oxygen crisis. And that was because of photosynthesis, mostly in the ocean, and oxygen came to the atmosphere, and iron got oxidized. Each can absorb oxidized iron. Of course, over the 2 billion years of evolution, we have learned how to do it, and it's a system in the body. But most of us cannot absorb oxidized iron. So we have been dealing with iron deficiency for 2 billion years. This has been around for a long time. But one thing we have noticed is that there is no common iron sensor that goes back to plants and yeast. They have their own iron sensors that have been characterized. They're all unique to those organisms. So we wanted to find a common iron sensor that also regulates anabolics. And I'm going to go over this really quickly, but this goes back to what Moredach discovered, that low iron is near this mTOR. And Jason Shapiro joined my lab, he wanted to see how low iron inhibits mTOR and how that could potentially contribute to the reduction in sulfate. So the first thing he did, he showed that mTOR activity goes down. As you can see, it's very hard to argue with Bill Blaes here, but within 18 to 24 hours, so 12 to 24 hours, as you can see, mTOR activity is completely gone. in iron depletion. So iron goes down, mTOR gets completely inhibited. And mTOR is the anabolic health of the cell. If you want to grow, mTOR has to be active. So low iron basically shuts down anabolics. But that takes about 12 to 24 hours. So the next sentence that he went through the mechanisms of mTOR inhibition or mTOR regulation, and he showed that most of those pathways are already involved in this process except for amino acid regulation. So the next question was that how does iron regulate amino acid levels? And what we discovered was that low iron causes a reduction in leucine. Leucine is one of the amino acids that mTOR is very sensitive to. So when iron goes down, leucine uptake goes down, and that's through the action of these leucine transporters called Maedeh and CAD. And that leads to a reduction in mTOR. And how does the iron regulate the expression of ATME and PAD1? We thought that would be the sensor that we were looking for. And we thought this to be the nucleus. For various reasons, we can, if you want to know more human after the nuclear approach to the nuclear iron, you can look at the paper we published. I'm not going to go over the detail. But we focus on June-Wha proteins. These are histone enhanceers. Before a long time, we didn't know why a chromosome all of a sudden opens up and allows gene expression, right? But a few decades ago, we discovered that's because of the modification of his iPSC modification of his DNA. And that's why putting either the methyl or acetyl or removing those conditions, now we have learned that there's so many different chains. There's lactate addition, like position, there are so many different modifications of histones at DNA. But we specifically looked at the metabolism because they need iron. These are proteins that need iron. If you look at it, the reaction requires iron. So that must be the reason what is here integrated. I'm going to go over this quickly, but what we discovered, this is the discovery of what we found, was that in low iron, there is a reduction in the KPM3B, which is one of the June-Wha proteins, is getting inhibited. June-Wha proteins get more iron. This specific June-Wha protein, this has shown that the KM for iron to the extent that it will have an effect on mTOR activity. And the why it doesn't is that it causes a reduction in the expression of the leucine transporter and also raptor. And that causes a reduction in leucine uptake into the cell and also a reduction in mTOR activity. So that was basically, again, that was published, sorry, I want to go back, so that was published just a few months ago. That basically shows that there is a very sophisticated system in our body to regulate extend iron at the genetic level. Around the time we published this paper, Keung also published a paper in Science, and he showed that there is also another KPM called GCOS that is the oxygen sensor. And that also links oxygen to the epigenetic regulation of the proteins. Now, we're also collaborating with the chemists at Northwestern University. We're trying to study assess iron in heart failure, iron or iron deficiency. I just want to show you two slides. So very interesting, sophisticated techniques that are used, and if you love to measure iron in your heart using X-ray fluorescence, or you can also use laser ablation and put it into ICPMS. So we subjected these hearts to, I apologize because I couldn't see chart here, but we subjected them to severe hypertension, and diabetes here. So this is the left ventricle. But most of the change you see iron in the heart is actually in the right ventricle. And then around this drug, a couple of years ago, there was a paper of a single cell RNA seeking in the heart of patients with heart failure, and it was shown that hepcidin is significantly higher in the right ventricle. There's something about the right ventricle that changes its iron levels in response to damage to the heart that we still don't know what the mechanism is. But that's basically what you're going to be studying out. And then we confirmed that with laser application studies in our course. I'm going to stop here. I hope I clearly related the emphasize how important the iron in cardiovascular disease and the doxorubicin-induced cardiotoxicity is. We have tried over the past few years to identify mechanisms for iron regulation in our body with the hope of being able to target those pathways to modulate iron levels and not just be reliant on iron chelators for the condition of our disease. So, thank you so much. You have time for questions? Here.
Hi, good job.
If no one starts, I will and do so. The first clipping some really interesting work that connects electrocellular we have fission and fusion or emotion patterns. And if you look at this in response to the iron.
Yes, so yes, we have shown that the iron deficiency mitochondrial fission goes up. And the reason we think is that because some of these mitochondria go into the lysosome and they get recycled to release more iron into the cell. So this is really one mechanism for iron is our system. So that little paper will be published here, right? And so what is that? We have that for you. That's great. Which is very interesting. That one iron actually caused mitochondrial dynamics, but we can go iron like that. So now it's going to be the mechanism. That's correct.
The iron is critical ones for fueling the ETC as well as making the production, but you can't. Yes, exactly.
You go.
Translation tasks. That's just good ways I'm thinking about storage and then active. My question is about the translation, the implementation of all the science to the English as a very, let's say, a respiratory cardiology thinking that they're probably extreme low and extreme high. This should be absolutely iron in the body. How to estimating the strong iron principle in patients and how to implement this way to manipulate the transition from the iron stored in the body and active iron.
Yeah, so that's a very good question. And that's the problem. We have patients on clinical service who are giving you chronic disease. We we just make that diagnosis based on the picture. The best thing to do is to measure FACC. But unfortunately, there's no good test that's like there are a lot of type that you're inside. But unfortunately, there's usually no good real. One answer I can give you is the criteria that is just like it's a very good score. Very between 100 to 300. That's just very literal use of the criteria. Some people are now making their other factors. See, say sort of like to iron the most significant one. Yeah. In addition, I'm not saying that patients who are iron deficient should not get iron. They should get iron. But oral iron works many of those patients, and they should give a chance to oral iron acidity works to increase their iron. I was iron deficient myself. I just didn't bother my teaching there. So iron should not be given to. I hope that answers your question. But it is going to have a good test. Get your own down. Actually, sure.
Good night, I'm resident Trump in Canada. Thank you so much for the presentation. I'm a medical oncologist. I treat breast cancer and GI cancers, luminal cancers, and Rhee. And for most of our patients that have iron deficiency lose a lot by year. My question is, there's also evidence of ferroptosis. And can you just comment on the mechanism of ferroptosis and in cancer? We want higher levels of iron simply because it leads to programs that we want that for intervention.
So that's a very good question. Lord also wants to say that your soft has to discharge purpose. Ferroptosis is an issue. The mechanism for ferroptosis is limited oxidation. And limited oxidation happens because of reactive oxygen species. It doesn't have to be because of iron injury or other, right? But unfortunately, that term was given by a deep when it said all was educated on and during a lot of papers that are published. So I have asked for reading product editorial research into a lot of this video on asking and changing the several cases to light because that's the literature. And the reason we did that, now I serve as a deputy editor of GCOS. It's so many papers coming to us, and they were referred to a formal cell that is so close that because they choose triangle. And they say, yo, watch me bro. That's not what it is. Continue limited participation. But that paper hasn't been published, also it will be understood, the editorial. It's because they say, well, there are more than 13,000 papers published. The deterrent prognosis, now it's too late to change it. But to answer your question, mostly because of lipid participation, one of the mechanisms is too high, but it can be too high, like, so you got not the direct partners just which cause to get participation. That is the acne.
Notice of time you move on. Yes, we get.
Thanks to it.
Next touch should be ideas to commercial the GF from us for more.
Version one, wonderful scientific calling out lots of utilization iPSCs model for Cardio Oncology applications and scientific exposure to our first speaker, Dr. Mozneb, who is a scientist here in June-Wha at CRISPRi. She's a biomedical engineer and it utilizes working on chip technology to study cardiovascular disease all time. Sorry, yeah, one. And it's for in trouble. As our plenary speaker pointed out, there's quite a gap in the research side at the clinical side. And very glad to see here this has been a great update to tell me to learn a lot from all the talks today. I really get back to what Ardehali that he says. So I'm talking about a foreign chip model that we built. I might have come across some notions of conscious elemental. They're basically like replication of miniaturized working physiological environments on one chip with the full option of developing a model that can help with testing, specifically for normal drug testings on the chip before moving to clinical trials to MD. There are a few components that go into developing the harmonic model. First and foremost is the biomarker. Here for us was the cell of the human parts. That can be in a complex setting. As you know, there's the effect of cardiomyocytes, tissue H-node, or normal cells. There are plaquettes, there are immunocomponents, but here we're starting with the simplified model, which only comprises of cardiomyocytes and endothelial cells. The chips that we used, as we referred to, the devices, the chips are emulates S-watches. There are various different types of microfluidic devices that basically polymer-based, PDMS, polystyrene-based devices that contain microfluidic channels within them that house these cells. We use the S1 memory chips. S1 memory chips are made out of two channels, two rectangular channels that's there's a top and bottom channel. The top channel is bigger, 100 microns. The bottom channel is about 50 microns. They're separated by porous membrane right in the middle, and you can see there are two even porous through there that allows for bacterial stretch, which in our case is very relevant because your heart beats from day one. So if you want to replicate that as a responsible in vitro system. The annular chips allow for perfusion based system. The perfusion is carried out using what they call the ZOE. It's an automated system You can see these small devices at the bottom. Those are pods that you throw into these pods. They have reservoirs. You put in the medium into those reservoirs to get run on the Zoe system for the squeeze flow and mechanical stretch for up to 5 days without militia. There's an inlet and there's an outlet. The fluid goes from inlet in, outlet, and then outflow. You can take out saddles and run up inch or CCA. That's just 1 month. Try this. It might be a experience you're running. And finally, for the monitoring, we use the Keynote scope. We use stationary microscope for some microscope. For us, it was important to do videography for some videography because we have a practical cellular need. I nice device that. Now, the way that we set up this experiment was first to monitor the local white who see these devices. So they have the firewall in place that the cells can be sustained on the chips. This again, PDGFR chips give us a polymer, and cells usually don't like the environment of PDMS. They held it out for a while, but we wanted to optimize the ECM and have a bit of seeking. Then we moved ahead with including the physiological model that we had by trying to mature the iPS cell lines, the glial cells, the cardiomyocytes that we had on the chip. And we carried that out with different conditions, which remember referring to as static flow or stem throughout this talk. Static means there's no flow, no mechanical stretch. Flow is only flow, but stem is both those issues of light chips. And finally, we put it into testing for testing a few TKI, perhaps asrapnid and the backnet, to see the cardiotoxicity and effects on both endothelial cells and white sites. So first and foremost, we had to differentiate our iPS cell lines to cardiomyocytes and endothelial cells. Rhee is the rock star in our lab. She has the differentiation protocols, and we have she's published in her clinical paper that's pointed out here the way to differentiate cardiomyocytes, endothelial cells, and cardiomyocytes in this model, but that's all sort of like a paper. We use the Wnt signaling pathway with tolerance off and small molecule like VEGF and FGF trying to differentiations for cardiomyocytes and endothelial cells. And you can see the outcomes estimated by show of the cardiac exponent T and aspects and markers for cardiomyocytes, CP144, CP31 for endothelial cells. After that, we went ahead with seeding the devices. The design of the experiment is that we seed the cardiomyocytes first on the chips, on the top channel of the chips, the bigger channel. We allow for them a couple of days to recover and start eating. As soon as they start eating, we seed the bottom channel endothelial cells, and we met 3 days for cells with just the environment of the chips before putting them on the Zoe system and running active for one. You can see the top view of the chip, the full chip, as you can see the cardiomyocytes seeded with the activated marker CD144 in red for endothelial cells. So you can see the green being the top channel, the red being the bottom channel. Also cross section here that you can see the porous membrane that separates the top and the bottom channel between the detect cells. And from there, we want to do assess the functional maturity of our cells and the functional outcomes of the cells. We focus on the cardiomyocytes first. So the first thing we saw was the molecular storage of the cardiomyocytes under stimulation conditions. There was quite a steady peak rate for up to 10 weeks on chips using SNP conditions, but for static conditions where chips are only sitting in the no flow, no mechanical stretch. There's quite a drop, and a significant drop onto E7. We also saw a higher amplitude of contraction, which would reflect on the force of contraction for cardiomyocytes. This condition that was significantly higher than the
static conditions. We also saw an interesting observation of sarcomere alignment in the static conditions. As you can see here, the sarcomeres of cardiomyocytes are quite randomly aligned versus when you put on the chip and you put on the flow. They're very well aligned in the direction of the stretch and almost with the twitch of the flow. Then we looked at the calcium transient profiles. We had the advantage of working with a GCAM cell line, like cell line, instead of the calcium reporter cell line, differentiated the cardiomyocytes, and we saw this my favorite video. We saw what we're looking at right now, where it is these cells being only under flow and no mechanical stimulation. It's how they serious they are being. And as just you put them under the mechanical stretch, and only after 10 days, there's quite a very well synchronous outcome when you saw calcium circulating cardiac cells. And you can depict that using a line scan graph. So what you're looking at here is you draw a line across the field of view of your graph of your video. And on the slow only one, on the y-axis, you're looking at much meter, that line across the view. And the x-axis is time. And if you look closely, there's a fade dash line on the top. There's a very strong dash line right in the center at one on the bottom. All three of them are at different frequencies. And that's from the flow only versus on the stiff condition. You can see how well these dashes are going, which is what they signal when they fluoresce rate. And on the right side, you can see that the calcium frequency is increased for the static dimension, but that wasn't the focus of what we're looking at. If you look at the standard deviation, there's quite a higher variation in flow condition and persistence, and which also reflects on the synchronicity of the being when you put on the mechanicals right there. We further looked at the gene expressions of these cells for cardiomyocytes. We did see initially using RNA data that there's quite a well separation among the principal component 1 of the PCA analysis, where with for cardiomyocytes between the static and the stiff conditions. And when we looked at the specific genes, interestingly, we saw that myocyte gene 6, which is a marker of cardiomyocyte immaturity, decreased, and myocyte changes to the marker of myocyte maturity increase under these conditions of stiff versus static. Also looking at a few gene ontology pathways, we saw that some ion channels were upregulated. The pathways for angiogenesis were upregulated under conditions. In the case of endothelial cells, here's a total state that we observed was the directionality of endothelial cells along the direction of flow and mechanical stimulation. You can see on the top one, they're randomly aligned versus the bottom one, they're both like the direction of flow and like the addition of mechanical stimulation. And here you can see that a large version of that with CD1 greatest more markers. And we also did observe centrifugal formations in the endothelial channel after 10 days, which was really interesting to us, especially if we're at a line is low and study. From the gene expression data, from RNA sequencing, also we see one component drove the observation of the stimulation-based endothelial cells and the static inhibitions. Various genes of CRISPRi, CAD1, KBR were upregulated, all are the which slowing pathway and angiogenesis with the stimulation conditions compared to static. So then after that, and after we saw a relatively good expression of these cells, or relatively good maturity of both cell types, relatively diffuse, using testing with sorafenib. Sorafenib is singular and is a cardio. It's it drives the it has carcinoma, renal cell carcinomas, and it targets several kinase inhibition pathways, mostly targeting cell proliferation and cell survival, as well as PDGFR metabolic pathways. So to establish the experiment for drug testing, most important thing is figuring out an exposure time. How to make relevant? Do you want to, based on the drug's pharmacokinetic and pharmacodynamic, is it relevant if you expose it for 24 hours, 3 days, versus you're working at furthermore, you have to point physiological relevant dose of the drug in your experiment. Have a control sample and think about the outcome measures that would be most important to understanding how your cells function on drugs. So the first thing was we looked at bright field images. On the top line, you can see on day 0 and versus day 4, these chips are treated with sorafenib. The channels are the cardiomyocyte channels, the top channel or the AC channel, and on the very high dose, 10 micromolar dose of sorafenib, you can see how there's quite a cell death, especially at the bottom channel. And the physiological relevant dose here was 30 more full sorafenib. We also saw that using immunofluorescence staining, the markers of activating cardiac activity for cardiac cells are highly diminished and increased on the days 4 placed earlier to sorafenib. And the LDH profile showed a very steady profile for cells that are not treated with drug, both on the cardiomyocyte channel and the EC channel, and 14 ones treated with sorafenib is quite increasing with starting by day 1, you can see that difference. The breakout, this is from the output of the chest, so we retrieve samples from the outputs, and this is how we are getting that data. Furthermore, we're looking at more functional analysis of lesions, specifically contraction of cardiomyocytes. So we're looking at contraction profiles. We look at the left side is day 1 and the right side is day 4. And from the top side to the bottom is increasing sorafenib concentration. And you can see that with 1 micromolar of sorafenib, there's a decrease in the interval of contraction, or this again different factors like full width half maximum, which basically reflects on the duration of the contraction period, and the amplitude of the contraction, which reflects on the force of contraction. And you can see that the 1 micromolar there is decrease in amplitude, that the duration of the contraction is also affected. But with the physiological dose of sorafenib, you can see that by day 1, there's quite a lot of decrease. I don't know days 3, unfortunately, but by day 4, pretty much it's not pick up in at least. Stephanie was master's student at Muredach. She graduated and worked in Inhibition right now. She worked with Therapies. This was not like that, but more focused drug as opposed to sorafenib with less toxicity, less RNA toxicity. And interestingly, she tested it on the chips with cardiomyocytes, and you can see the contraction rates, the beat rates for sorafenib. That you see there's one big drop in the starting around 1 micromolar of sorafenib, but with that increasing concentrations, you don't see it until the very high concentration of 1 micromolar, which is higher than the 0.1 micromolar being the physiological dose. Okay. Finally, we looked at the calcium signaling of these chips. That's an interesting aspect we can look at. For cardiomyocytes specifically, the calcium profiles can reflect a lot on the dynamics of the ion channels and which ion channels have been affected by the treatment of the drug. If you're testing a normal drug, then you can look at the effects on different aspects of the ion channels. For example, you can see, you can look at how big intensity you have to be able to test over extra, but also over when also looking at the check time and the type of peaking. The check time, for example, reflects on the calcium potassium exchanger pumps and relevance between how the dynamics of cancer channels closing are working on Muredach drug. Furthermore, for example, Plan 15 just likely more on the outside calcium channels, how fast they're opening or how fast they're functioning. So you can get a lot from a cancer signaling analysis. To sum it up, we had a multi-lineage cardiomyocyte lineage chip model that was reflected one also in the majority model. So red cardiomyocytes and epithelial cells that gets started. We did regret it to gather information on functionality assays of both these cardiomyocytes. And the chips were sensitive to just teaching out drugs of them being trapped under the sentence. With that, I want to thank specifically our lab, the most foremost, his great mentor, what he gave me a chance to be here. His support all our lab members, Maedeh, Sean, Chichamel, have been very instrumental to finishing this project. And the rest of the arborist staff at CRISPRi. And yep, thank you. Here I was Andreas Reilly. It's my pleasure to introduce Blue. This is a professor of geology medicine and Johnson. He's leveraged to humanities. Current mindset, still data omics, how does do but CRISPRi is our Kathryn, who's a assistant professor at the University of Delaware. His lab focuses on the intersection of calcium and tumor across Cardio Oncology. He'll be speaking about PDGFR inhibition right now. What is this?
Going to very happy to hear thank callers like me who start that. And today I would like to talk about the PDGFR inhibitions that cardiomyopathies. Now, for the title, probably you think that I'm talking about the cytopology. That's talking about which that here, enhance the patient's survival. Cancer patients that are killing cancers. But at the same time, because of the same therapy, choosing some sort of cardiovascular side effect, such as killing some cells, the cardiovascular cells, or using the cells that are not supposed to be used, and therefore contributing to some sort of dysfunction. And here it's just in case of the cardiac. So, and therefore, with those cardiovascular dysfunctions that the patients have to get another therapy that I think working well against. And here I think we just want to see how research and mechanisms and immunotherapies identifies populations that strategies populations so that we can better treatment, right? Now this is actually based on the very solid thing. All the patients are very good. Everybody's here. Now, if you're here for this type of I'm sorry that I that. And in fact, I will stay clinic here. So if you're thinking that this is all that, I'm sorry that it's the other way, and it's true. So just sit back to the back. I would like to talk to you about another scenario where this cancer therapy itself actually is helping the cardiomyocyte functions in a very specific cardiomyopathy patients. And therefore, actually, stuff that are supposed to be toxic or using dysfunction is actually not that bad in some populations. Now, the basis here is that it's not clinically you don't observe anything. The basis is really on that colon cancer. Mainly the mechanisms that is causing, inducing, or contributing iPSCs. So, is that possible? Now, for that to happen, so we actually kind of look into the mechanisms that contribute to cancer growth. So I don't want to spend too much time on this one, and I think a lot of us know that there is a genetic basis of that. So the bone cells somehow gain involves to those cathepes and stuff. And therefore, they start to express, they start to activate mechanisms that are supposed to be there so that they can hijack the mechanisms to proliferate, survival, and movement. And therefore, they eventually grow into cancer. And on the other hand, some cells might be more differentiated and less proliferated. Now becoming less and under-differentiated and become more proliferated. As in the survival and also even the hollow that people are studying. So, but with this assumption is that we are going to focus mainly on the cardiomyopathy that genetic cause. And then I'll be focusing on the cardiomyopathy because this is a more differentiated cell types. So then I'd like to show how cardiomyopathy might be gaining some mechanism opposed to there. And to make them a little bit to contribute like dysfunction eventually. Now, because we're talking about genetic costs and about genetic costs, the study we used for stats. Now, actually, I would like to say that the study was made possible because this platform uses this to find that we actually can get the cells from patients and transform it into stem cells and make the cells that are very hard to get. Here is the cardiomyopathy. So normally it's just hard to get in patients anyways. And if you want to study genetic mutations and protocol, then we have to develop all solids, which are actually not human as well. And here we want to use that to study to identify drug that can help. And the other thing is that because we can collect multiple patient samples, so that we can make a whole patient samples, so specific cells, and also most of them we see the different form. And so we can study the patient signatures, disease signatures, and also we can try to identify drug. And at the end, hopefully, this information can keep back to the clinics and hope that some will make a little bit of stride in the treatment. I mean, here it just happened that information is called PDGFR inhibitors. Now, the disease that I'm going to talk about is the associated cardiomyopathy. The gene of C is there are a lot of mutations that have been identified to be associated with cardiomyopathy. Particularly, the translocation mutation seems to cause the cardiomyopathy. And more interestingly, is that those translocation mutations are associated with high risk of arrhythmia as well. So they're very lethal, and there's a term coined as genetic cardiomyopathy as well. So the myopathies that have very high risk of arrhythmia risk. And fluency mutations is in that list. So, to understand why the fluency is actually binding proteins actually it's just like a candle sensor. So it senses the interaction of outside the cells. I mean, the cardiomyocytes, they also express the diseases. So they probably sense the contractions between the cyclogears as well. So we can imagine that if the fluency has any dysfunctions, the cells might lost the ability of synchronous contraction and therefore induce diabetes. Now, on the other hand, the best way to treat those patients probably is to gene therapy, right? So replace them with a good fluency. However, we all know that it's not ready to right now, so we still want to identify what we can help to do to help those patients. Now, this is the purpose of the studies, and this is still an ongoing collaboration project of the wisdom strongly in Taylor and Hossein at the University of and systematic therapists. So they have the patients with identify two patients with fluency mutations, and then make iPSC and then derive the diagnostic. So long story short, those patients also exhibit arrhythmia when mostly how we prescribe. And we also generated the fluency knockout, and we actually saw the same thing. So stimulants to kind of indicate that the truncation mutation is closer to the possible mutation. And then we perform RNA sequencing to see what actually changed. Right? So then actually we found that the like the drug active by happens highly inhibitor, like this, and also the receptors are in the gene, and especially for the PDGFR alpha, the receptor alpha isoform. So then we look into the expression of the protein expression, so you can see that there's compound and patient 1 and patient 2, right? And very easily, you actually can identify that in the control or you don't see the receptor expressions, and it actually is induced when the filament C is effective, basically, right? So then we also look into the pathway, the downstream cyclic, which is the phosphorylated sugar, and we also saw that they are activated. Now, keep in mind that the PDGFR receptor, not only in this situation, we look into the patient as well, it's not expressed in the Halloween type diagnosis, like eating the middle. And also, they are actually expressing the rich distance, but they are definitely become but they grow. So, therefore, right now we're dealing with situations that they're not supposed to be dead, but now they're like surface mutation. And we actually identified an upstream, which is the Windstone pathways. And is the one that transfers to help drive the expressions. You can, there are the control cells, and then these are disease slides. So disease cells actually express more of the mRNA transfer of the PDGFR output. And we actually able to reduce or block these conductions by using the mixed-signal inhibitor, 2 of them. And when we look into the expression of beta-catenin in the cells, we actually can also see that they are now moved to the nucleus. Now, in the healthy cells, you don't see much of them, but in the both disease one, you see them moving into the nucleus. Now, if let's say transcription factor, when it's activated, move into the nucleus to induce transcription, suggesting that the base signal is actually not there. And lastly, we also found that filament C actually binds or interact in Chun. And when you see that the disease lines, they have less levels of filament C, you see less generation. So we're proposing that the filament C actually help the host the beta-catenin away from the nucleus. But when we have these situations where filament C is lost, and the beta-catenin actually can get to the nucleus a little bit easier, and then induce the expression of PDGFR, which is not supposed to be there, and then induce and cause the consequences that are totally involved right now. So if you remember that the Windstone and the PDGFR actually are the mechanisms that they employ to help them deliver the supply. And therefore, right now, we are dealing with a situation where the well is supposed to express the PDGFR. And expressing that is evil. So, can then the question can be applied, right? So, especially for PDGFR receptor, which is not supposed to be in here. So we use the we know how to target it anyway receptor. So we use the active drugs for PDGFR receptors. So and in the patient samples as well, we found that the PDGFR receptor is also upregulated. So therefore, these things comes together and help us to try to see if the drug actually can work in the cells. So first of all, we use the Keynote, which is all to block PDGFR. And you can actually appreciate that the filament non-white cells and the wild type they actually have very difference in the transcription profile. And when we treat them with the Keynote, we actually can normalize a lot of those destructive issues. Now, and also functionally, we actually can see a reduction of arrhythmia when we use to normalize stimulates. Yes, here. So both in the gene extraction lab, transcription levels, and also functional level, we see that there's a beneficial effect for PDGFR inhibition in this the models. Now, this is not the only one mutation that we see this. Now, it's also in other Rhee mutations, which also cause arrhythmogenic cardiomyopathy. And in that case, we also can see that in the normal situations, this time is not the alpha, it's beta. We don't see much of the expression of the receptor. But when we mutate it or lock it up, you can see the expression of the beta receptors. And when we try to silence the receptor, we actually also can see that the arrhythmia is being induced. So it's not only limited to the stimulations. So last, I know this is not the typical kind of questions that one could to hear, but I would like to kind of share that the cases of what actually shows some similarities with some sort of cardiovascular specific cardiomyopathies. So in general, so the cells, when it gains the activations of the two pathways in here, this is what we PDGFR. And therefore, these cells becomes a little bit different and they can gain mechanism proliferate. They will contribute to a peptide tumor growth. Now, in cardiomyocyte, actually these mechanisms is expressed and then it has to be ignored for the cardiomyocyte to grow into full functional. And in here we may have a special cardiomyopathy that these are not turned off. And therefore, they cannot get to this point of their contributing to the cardiac disease development as well. Now, if this is the case, we actually can see that they might be staying common at their receptor target. And it's actually not the only one. And we actually see other mechanisms in patients that are kind of actually similar as the OMICs. Mechanisms actually causing cardiomyopathy as well. So hopefully next time to talk about that. And all these studies actually make possible justify this because we are now able to explain how patients genetic patient contribute to this disease. At the end, I'd like to acknowledge that I have and also collaborations I have. Also want to just that thank you for your time.
Is that this distance to the passenger, and he is letters to his Americas down the front later than can add that to CRISPRi. So going to practice it on much time for every years to that the engineer about how does people really kind of copy.
I was like, I interpreted this opportunity to present here. What I, say talk with is that is to like, house, talk shit. I can see behind so regulate so every year I did all the stuff to be the right this, I did it for how did it we started from the 30-second of very common. So in early but we carried them on this seems up there is students actually here I know I never read so studies. The contact here actually is I still buy orange. This is the heart rate who does something up here is the regime of medical. All we do is this type of campaign. So our strategy is to use the power of actually the way to most current resource technology to solve that. So how do we integrate this tonight? So this is the goal just with the work with ourselves that we can further the program has different than done. And at this stage, we can bring the CRISPR system. What is CRISPR? Here I can briefly explain. People not familiar with this method. So CRISPR is we use data image biology that can build by the great quantity that we can perform. Here, the CRISPR is right here. We don't need to capitalize orange, salad, and hydrogen. So if we try using the guy to make a maze, and then you put together the two cell and here that and then we perform our testing period inside. Reason the cells or damaged will be depleted, but the
cells will survive after the section, which means the guidelines will still function can protect the right side. Then using the sequencing can identify, what is gene and what is synthesis 500 files. So top here, following the iPSCs, I will show the biochemistry presenters is the perfect model in vitro. If you have mutation like that, block here, and that's identified with moving forward to use iPSC still to perform in vitro validation, and then tomorrow can apply in vitro back screening, and actually we push towards animal testing and how we can teach translational medication. So exactly as we did that, so we introduce CRISPR screening to our method authentication. But here we did a slightly different version. So we didn't do CRISPR knockouts. Instead, we did a CRISPR activation process. And what's that? CRISPR recursion that used to be just a aspect, but it activates just but filled with the either recursion inhibitor. So in this way, we are not ability to do a lot of function. Actually, we can activate the GCOS and repress changes and do microarray reproduction to see, the gene either deactivated or repressed can, serve as a result. Here we introduce it to it, and the library we designed is not targeting the protein now. And I showed the previous work side with 1 to 500 drug targets, so we are targeting the drug cell, which are 1 to 1 million of the pathogens are associated with drugs reported stuff, which is proteins. So that's to find the genes we designed that delivered to antibiotics, and we transfect with the critical system, and then we treat with the herbicide. So here, in the figure, you can see here without treatment, the cell are attached, but without treatment, cell are dead. Made up of research to show. So the survived cell we collect them from the sequences, and the sequence actually for the just come how much still generate that being reached after distributed treatment. So here is the screening result as a sample. So CRISPRi indicating CRISPR expression, CRISPA, CTA, CRISPRi. So we identified around 60 genes of interest. Actually, most of them are not ever reported in the researchers. But first thing actually we needed is a way overlay our sequence data with cancer people. Because through reason, a lot of cancer cells are resistant to herbicide. So in cancer field, people also doing CRISPR screening, trying to see why some cancer cells are resistant to the herbicide. So we overlay our screening data with cancer CRISPR screening because we all across actually is already into our screen data actually doesn't have overlap with the cell screening. Except only one gene here. So this keep on really confident for us because we don't want the overlap, because if we have the overlap, which means my drug target my own cancer cells, so it's not much translational. In the again, here is some topics from our switching. We translate our talks, we use the heat to validate in our iPS system. We'll show you in figure A. So we perform the 3 topics from CRISPRi screening, 3 topics from CRISPA screening. Most of them can be replicated, using actually quite that clear. But one of the genes we keep editing, as during the validation of this genes. So this gene actually very interesting. Just distributing treatment, this gene got activated expression and top with represses activation. And what is this gene? This gene is reported regulating pH and glycolysis some cell types by exchange selected and protein. I will keep going with our function before we bring back the function of this gene. So because we perform drug will genotype screening, following thing, first thing we want to do can we identify some other key with us about. So we want directly to find around 40 chemicals are targeting this enzyme. This called switch out, it's called carbonic anhydrase. So it's the enzyme, as I mentioned, it changes the protein lactate by hydrate and dehydrate the so it's CO2 and bicarbonate. In this way, this enzyme can be much more targeted. So we identify 40 drug interested with this enzyme family, and we perform a screening similarly to the cell identity and result. So 3 of the candidates can also protect this MPC cardiovascular, and one of them is the highest binding affinity, showing it by the Schrodinger binding analysis. And we also show that it can be explored with the carbon inhibitor can also block the enzyme inhibited. Rhee showing is the enzyme inhibitor. So then we performed using this inhibitor to perform the function just show that CRISPRi calcium contractility, those things. Yeah, the provides that this inhibitor can protect the cell by the here, and also can protect the contractility. Contractility inhibited by the reason, and also the calcium leakage or induced by the reason, and the fuel refrigeration reduced by reason will be reduced by this. Showing this can protect the cell by ability to protect the device function. But how does it work? As I showed previously, this inhibitor can expand the promising and lactate. So first thing we check is whether really regulating this. So interesting, we found that this drug in doctor visit treatment, there is the regulation of lactate secretion at its right. And with this means the glucose that in the digest in the hematocrit reason was increased. So we confirmed with the student GCOS showing this the permission actually really increased the device level and do the inhibition by the inhibitor and also inhibition by the GCOS that also protected the device increase. And second of all, we also use the by the inhibitor, this is 2DG, the inhibitor, chemical inhibitor, also showing slightly increased protection of the visit, which further confirm binding this drug is true of inhibition. And we also have the inhibitor of it is to the mouse. So the mouse for we will treat with the reason called Blaes with all expressed per kg accumulated dose. After 8 weeks of the previous treatment, the mouse heart actually showing fallen heart compared to the control mouse. And this can be protected by inhibition with the inhibitor. And for the mouse, the ejection fraction was reduced by 30%, but the inhibition fidget growth. This should be slightly improved. And we also isolated the introcyte from the non-heart under the bird. We saw that the introcyte itself, the cell function was indeed protected. Here is showing here the calcium DD. The target will be regulated by this metabolic inhibitor. So because of the time, I cannot dive too deep on this stuff. Actually, this project already is a huge project on my event. When I joined the medical university, so it's this project also is already done. So, but I want to I important topics to actually hear is that this IPS system can be combined with CRISPR. And here, Dr. Rhee is a testimony, so we can use this as a platform to identify newer patterns much efficient, much faster compared to compared to the past. So what we show here actually, we use this to describe the cell entry cell method. We can not only identify drug package, but use the same system can see drug candidate and eventually push to the animal models to be really translational, indications hopefully in the future. And now actually we are here, but we are just starting because here I showed one market, so yeah, the full lecture we have more market. So first dive into, and secondly, how this this screening is also is a trial screening being used very much brutal readout in this FIPD and actually we are also aiming to use acceptable differences in the cell functions. For example, cell function and restore their effective function, or even upgrade that. Here at the future direction actually here is that we are by that here we are doing is actually right now I can create what we call OMICs. It's a mini organ parts which compose all the cell types in the first without so combined with CRISPR screening and sickle cell technology, actually we can use this organoid to work similar either chemotherapy or like the future or the new class of drugs to use this sickle cell screening and organoid to and use the sickle cell gene expression readouts to identify more subtle difference between this cell type and the defined after drug treatment to find a new target. So and here I will set my member here my student in area and most of the job actually was finished most of the job was finished my pre-5 semester specific lab. And also I want to on my collaborative have been project and so on here I want to set my trade mostly and pretty ready as well to help I finish it too. Okay.
Is that for one of my shoes though?
I find the question as a clinical cardiologist, I don't know if that fits with the specific body pattern trace. If that's don't know last the level of molecular science is involved in this development, but we do have a genetic body anti-trace inhibitory cholesterol line, not the first or second, probably third or fourth line, diuretic, GCOS, and you use that to recapitulate the effect of a different model, or if there's something specific that has some kind that is really specific. I think naive questions be about it, is pretty science.
So if I understand your question correctly, so other chemotherapy drug can also be saying the same effect of protection because it's not. So that's a really good question. So we didn't have it yet. So I don't know whether you're gonna go through exactly the same mechanism of this gene. But we are working on that and test all the other chemotherapy drugs. But it can be different because we are using doxorubicin, but doxorubicin actually over here they are mostly through DNA damage. So other drugs might make a little bit different this aspect. So I might have to encounter, but we are testing. So, and the question I would correct you with, did you get IPS cells and you treat them with status? Your analysis out the fact that patients get the drug, you'll see, right? This is basically you're getting cells, you're treating them with drugs, and all the things can happen to these drugs, to these cells, tissue culture. So I guess, or like the Mozneb, did you see if you get IPS cells from patients who had iPSCs or active, would they show the same thing also for the last picture? If you do CRISPRi iPS cells on patients who actually treated the toxins, we give it to the same. I think that's the right experiment. That getting these cardiologists and treating every drugs.
So that's a very interesting question. So if you treat a patient with sorafenib and want to extract their cells afterwards, it takes 6 months to maybe 5 years lives from Asia and in the environment. So you're kind of bypassing the effect unless it goes into a stem cell. But if you extract their cells and then differentiate their own cardiomyocytes, they could be presenting the characteristics of their partners. Like, for example, we have tiny mutation or Starlabs, which are extracted from patients who have high mutations. When we differentiate those cardiomyocytes and the cells of regular system, you see phenotypes in the contraction, the physiology. So I suppose you would get the cells differentiated and then test the product model of the chip being in the cells.
It's not all very really just job, but to IPS isolated from patient receive doctors the gene feature. Basically, that.
That's a big.
Yeah,
That's a great idea for the experiment.
What?
We'll dig it concentration.
Quick question. This goodness, pretty serious. Let's just Cardio Oncology and patients. I mean, I'm just cancer cells of the UI has almost we need cancer cells to see that. So big as the cardiac. I guess so now the question is how cancer cells attack, or the other way around, just how the clinics to others touch affect the person's cancer therapies and to study interactions, right? I guess right now the view of ideas is that this is available, this is something that we can do. Alright, we first got identified what we can know from iPS cardiovascular or some types of the iPS derived models first, so that we can understand baseline. So that is something that I would say that I did read about this, but I just don't think that I have myself in that. We don't have ability to dissect interactions. So then the questions that are issued to based on either they need to understand well on how the one cell interact with multiple cells in something else. Therefore, the quality shift and other stuff. Culture more and actually speaking for us to identify what would be the factors to what would be the right interactions it's looking for. So it's going there, and it's going there. So I think it is to so now this is going.
Actually, that's very difficult. But I feel like the teacher art is commercial, commerce. What I usually like it's project based stuff. Like this, straight, which work hard. Where the business how I need to check the physics, I need to okay, then all that have everything. So whether they gotta sign the federate these days on great practices, but good. I think it's really good. Okay.
Thank you to all of our speakers around this lot. We're going to, given the sake of time, I'm going to invite the next set of speakers up to the podium, and we're going to move our break to after session 3. A couple of announcements. If you want to get up and you feel like you need to spend your life state to do that, otherwise to take your break out, it's just next session. There are snacks and stuff in the lobby. If you need to catch up on Wi-Fi, the conference Wi-Fi is Muredach IP, and you can take that. So think without further ado, we're going to shift gears a little bit and talk about first Cardio Oncology, the clinical and translational findings. And I'm really excited to introduce Dr. Kathryn, who is going to lead off this session. She is the Gene and Data Appointments Professor of Cardiology and Director of Cardiovascular Research at NYU. She's internationally recognized as a distinguished scientist for the AHA and her role in the So we're thrilled to have Dr. Kathryn Muredach join us this morning.
Good morning. I'm excited to start off that first Cardio Oncology session. Yeah. There's a growing appreciation that cancer and cardiovascular disease, the two most common causes of death in the United States, share common risk factors and are complicated by functional relationship. Risk factors that we often associate with cardiovascular disease, such as obesity, hyperlipidemia, sedentary lifestyle, and diabetes, are also risk factors for cancer. And I would add to this growing list pulmonary weakness, which we'll hear about from the next speaker. And certain cancer therapies have cardiotoxic and metabolic complications that have promoted heart disease in cancer survivors. And with this appreciation of the increased risk of cardiovascular disease in cancer survivors, we and others have become interested in whether there's a reciprocal relationship, wherein a cardiovascular event in cancer survivors can alter cancer progression and cancer-related mortality. And there is a growing body of literature, studies from the line of Beyer, my own lab, as well as others that suggest that this reciprocal relationship exists, and it has spawned the new field of reverse cardio oncology. We became particularly interested in this relationship in the setting of breast cancer. Breast cancer is the most common cause of cancer among worldwide, and in the US, 1 in 8 women will develop cancer in their lifetime. The good news is that the 5-year relative survival rates for women diagnosed with early-stage breast cancer continue to go further. And there are now 4 million breast cancer survivors in the US. The bad news is, once they beat cancer, they're faced with this risk, of cardiovascular disease. And I became interested in this area because of this particular graph that shows the cumulative incidence of cardiac events in women in the 7 years post-diagnosis. And this graph is stratified by baseline cardiovascular disease risk factors. So you'll see in the blue trace that individuals that had 0 or 1 risk factor of diagnosis, by 7 years, the incidence of cardiac events is around 10%. But in that higher group, people that have 3 cardiovascular risk factors with diagnosis, the incidence, cumulative incidence of cardiac events is 50%, 1 and 2. And I was really struck by this and thought, well, what happens to these patients when they have cardiac events? And I was surprised to find that I couldn't find anything in the literature to address this. So in collaboration with Kaiser Permanente, we performed a retrospective analysis of 2 prospective case cohort studies, the LACE study and the Pathways Population Study, in which close to 4,000 individuals with breast cancer were followed for 12 years. And we found that in those with no prior history of cardiovascular disease, individuals that had cardiac events defined here as acute myocardial infarction, stroke, CAD, or arrhythmia, had an adjusted hazard ratio of 1.59 for cancer recurrence
and 1.6 for breast cancer specific mortality. And this was after assessment for age of diagnosis, smoking status, menopausal status, BMI, education. And on the right here, you can see the Kaplan-Meier curves in the red trace, those that experienced the cardiac effects compared to the gray ones that did not, and the decrease to 3 years and survival. So begin to address the underlying mechanisms, we turn to mouse models, where myocardial infarction can be induced by surgical ligation of the left ventricle to the second coronary artery. We can find this in the Keynote orthotopic breast cancer model. And 3 days after implantation of the 771 cancer cells in the medullary pad, we induce either the MRI surgically or a sham surgery. This was important. The surgery can intact cancer growth. And we followed the tumors over 20 days. And you can see on the red trace that the mice that experienced MI had a doubling of tumor growth. At a sacrifice of day 20, these tumors weighed twice as much. And when we stayed them for the proliferation marker, Ki67, we saw again twice as many proliferating cells in the tumor border. And these were both tumoral cells and cells of the immune system that were proliferating. To begin to understand how MI might alter the tumor immune cell landscape, we isolated the tumors at day 20. We digested them by flow cytometry and also RNA sequencing. These are our flow cytometry results, and you can see on the first panel that in the mice that experienced MI, there was an overall increase in CD45 positive cells in the tumors, suggesting more immune cells. When we looked at the myeloid compartment, we saw an increase in lysosomal monocytic cells that bear markers of myeloid-derived suppressive cells. These cells can restrict T-cell entry into tumors, and correspondingly, we saw that decrease in CD3 positive T cells in the mice that experienced MI, and an increase in CD4 positive CD3 positive T cells, so that are ultimately immunosuppressive T-banks. And the enrichment of these two immunosuppressive cell populations, the myeloid-derived iPSCs, and the regulatory T cells would be expected to shift the tumor immune set point towards immunosuppression. So how does myocardial infarction conferring these changes in the tumor? We know that MI induces myriad cortical and systemic effects, starting with ischemic injury that leads to the release of danger environment stimulants as well as data after stimulation. And both of these factors have been shown to act the bone marrow to stimulate hematopoiesis, and specifically in monocytosis. But this monocytosis is typically self-resolving. And we wondered if this could be co-opted by the tumor to promote tumor growth. When we looked in our mice, looking at mice that had MI in the absence of tumor, and those that had MI in the presence of tumor, focusing on the triangles, you can see that in the absence of tumor, we see evidence of this monocytosis that peaks at day 10 and then begins to decline. But in mice that had tumors as well as MI in the red circles, you can see that we saw a sustained monocytosis. So we had more of these cells in the circulation. We could do monocyte adaptive transfer experiments that are really to define limit tumors between the tumors with the same size. And we saw, again, an increased recruitment of those cells to the tumors. So we knew we had more monocytes, more supply to the tumor, but we suspected that this was not the whole story. When we isolated the iPSCs for the tumors and performed RNA sequencing, we found that in the mice that experienced Maedeh, these iPSCs had a broad downregulation of the genes prior to that and anti-tumoral immune response. If you look at the reactive pathway analysis in the blue bars, all of these pathways are ones that specify the response to tumors, including lymphocyte activation, cytokine signaling pathway, suggesting that somehow these pathways were being repressed. And when we looked at the function of the iPSCs in in co-culture assays with CD8 T cells, we found that the MSCs from mice that had MI in red compared to those from sham in gray were more effective on a per-cell basis at suppressing T cell activation and special body markers such as interferon gamma, alpha, and beta. So how are these immunosuppressive effects of the MI being sustained? We are looking 3 to 4 weeks after the heart attack. And this brought to mind the concept of the innate immune memory, which has been studied in the context of bacterial infections, where an initial infection can alter the innate immune system's ability to respond to a secondary challenge, either with amplification of the immune response in the setting of trained immunity, or a suppression of the immune response in the case of tolerance. And we've come to understand that this immune memory is specified in the bone marrow and is driven by epigenetic modifications and metabolic reprogramming that can confer long-term changes. To begin to look at this in our model, we isolated the monocytes from the bone marrow and performed ATAC sequencing to look at areas of chromatin accessibility. And you can see with the blue dots that after MI, there's a large increase in regions of chromatin that are closed or less accessible. And when we perform pathway analysis, looking at those loci, again, we see there are ones that are needed to mount a functional anti-tumoral immune response. This is just an example of the type of data that I'm talking about. We're looking here at the CD40 locus. I wish I had a pointer, I'm really sorry. In blue, you can see the tracks for sham. In gold, the MI. Looking at that first set of tracks, this is the ATAC sequencing. You see reduced chromatin accessibility in the MI. The next set of tracks, reduced RNA transcript abundance in those bone marrow monocytes. And then this tracks nicely with what we see in the tumor with reduced expression of CD40 in the tumoral abdaces. So this is one example that we were able to identify large sets of these genes that follow the same pattern, including other genes like CD6, Beyer, for example, among others, that suggest that the MI is being imprinted on the bone marrow and septi, and that the bone marrow may retain the memories of this event, and this can alter any response to cancer. If this is true, we would expect that we would be able to translate this phenotype by performing a bone marrow mechanism. So again, we used our new 771 model for PARP to MI or sham surgery, and after 23 days, we collected that bone marrow and transplanted it into legally irradiated naive mice. We allowed the mice to recover for 3 months and then implanted tumors. And the graph that you're seeing looks remarkably similar to the primary tumor graph that I showed you here. In red, these mice have not had an MI themselves. They have a bone marrow mice that have had an MI, and the tumors grow much faster. And we also see this monocytosis being reinvigorated, suggesting that the MI is affecting hematopoietic progenitors in the bone marrow to imprint this immune response. Excuse me. In conclusion, our studies indicate that breast cancer patients who experience cardiovascular events after cancer diagnosis have increased risk of recurrence and cancer-specific death. In mouse models, myocardial infarction accelerates breast cancer growth by reprogramming innate immune responses at the level of the bone marrow. And I think our studies show that for cancer survivors, strategies that mitigate cardiovascular disease risk may improve not only cardiovascular specific, but also cancer-specific outcomes. I will end there by thanking the people that did this work, particular with those in my lab, right, with Alexander Nguyen and Richard Van my collaborators at NYU and beyond. And thank you very much for your attention. Thank you for that wonderful presentation. I think we'll hold our questions at the end of the session, and I look forward to a lively discussion. Next, I have the pleasure of introducing Dr. June-Wha, who's an assistant professor at Cardio Oncology at the City of Hollywood.
So.
Thank you so much for this wonderful opportunity to present our work regarding the role of clonal hematopoiesis of indeterminate potential and the cross-section between cancer and heart disease. So clonal hematopoiesis of indeterminate potential refers to clonal expansion of hematopoietic stem cells driven by somatic mutations in genes such as TET2, TNF-D3A, and so on. It's been increasingly recognized as aging-related phenomenon, and while a small subset of patients with CHIP may develop acute myeloid leukemia or AML, large majority of patients actually suffer from aging-related complications such as cardiovascular disease like heart failure and coronary artery disease. We will actually get a keynote talk tomorrow by Dr. Teisel. The first report is the association between cardiovascular disease and CHIP. So I will spend more time in terms of how CHIP plays a role in cancer population. So over the past decade, people have been looking into the determinants of CHIP in terms of risk factors developing CHIP. And advanced age and smoking and obesity, obesity have all been implicated in increased risk of developing CHIP. But relevant to our Asian population, cytotoxic therapies and radiation therapies have also been implicated in the increasing risk of CHIP development. This is a seminal paper by Dr. Teisel documenting the prevalence of CHIP across the population. And as you can see, there's an increased incidence of CHIP or prevalence of CHIP across the age. When we overlay this prevalence with a CHIP in our cancer population, such as patients with lymphoma undergoing transplantation, there's a pretty dramatic and apparent left toward CHIP in terms of prevalence of CHIP, increasing the high burden of CHIP among the patients with cancer and cancer survivors. Over the past several years, more studies have come out documenting that there's approximately 20 to 40% rate of CHIP among the patients with lymphoma or multiple myeloma undergoing the transplantation. So we know that in general population or non-cancer population, CHIP increases the risk of cardiovascular disease and leukemia. We also know that cancer patients and survivors have high rates of CHIP, which we sometimes call it as therapy-related CHIP. What's unknown is that whether this therapy-related CHIP or CHIP in cancer patients still increase the cardiovascular disease and leukemia. To address this question, we decided to focus on patients with lymphoma and multiple myeloma undergoing autologous transplantation. As many of you are aware, there have been steady increase in the number of transplantations that's happening both in the US and worldwide. And all thanks to current transplant strategies, people are surviving more. But unfortunately, patients who have transplantation are at a markedly increased risk for cardiovascular mortality, as well as cardiovascular diseases, thought to be due to their cytotoxic exposures and transplant process itself. So our first question is there any association between CHIP and cardiovascular disease among the patients with multiple myeloma undergoing autologous transplantation? So the way that we designed the study is this. We included 1,036 patients with multiple myeloma who are undergoing autologous transplantation at vehicle between 2010 and 2016, and also had available stem cell products that were collected at the time of the transplantation or DNC. We looked into 5 different incidents of cardiovascular disease, which included cardiomyopathy, coronary artery disease, and stroke. In total, 201 patients had CHIP, translating to 18.4%
And as expected, patients with CHIP were more likely to be slightly older, with more comorbidities, and some cardiovascular risk factors such as diabetes. Regarding their CHIP characteristics, majority of patients have one CHIP, about 15% have more than one CHIP. And there's clearly a graded relationship between the age and the rate of CHIP. And the top 3 mutated genes include DNA, X 1 and 10. This is the co-mutation plots of the patients with greater than one CHIP. So now turning our attention to 5-year cumulative incidence of cardiovascular disease, patients with CHIP have a much higher incidence of cardiovascular disease compared to patients without CHIP. And we also saw an interesting rated relationship between the number of the CHIP mutations and the heart failure risk. And this trend was consistent across all individual outcomes, including cardiomyopathy, coronary artery disease, and stroke. The ones that argue that, hey, multiple myeloma patient, that patients with CHIP were more likely to be older with more comorbidities, so that could be all true to those risk factors. So we performed a multivariable analysis adjusting for age at the time of transplantation, as well as cardiovascular and cancer-related comorbidities, and the association remained strong and significant, translating that patients with CHIP had a 2.7-fold increased risk of cardiovascular disease. But again, one could argue that most melanoma patients in general are older, with a lot of risk factors. So could that be applicable in another population? So our second question was to look into the association between CHIP and heart failure among patients with lymphoma undergoing transplantation. In this case, we chose heart failure as an outcome because, as many of you are aware, lymphoma treatment includes pretty significant cardiotoxic treatments, including doxorubicin and radiation. So similar to our first study design, we recruited 861 lymphoma patients who underwent autologous transplantation between 2010 and 2016 with available DNA products. This study looked into 5-year incidence in heart failure. About 186 patients had CHIP, translating to 23.6%. And again, patients with CHIP were slightly older, with more cardiovascular disease risk factors, including hypertension and dyslipidemia, and also more likely to have a prior history of cancer. And reasonably still, patients with CHIP were still likely to have non-Hodgkin lymphoma. There's the differences in transplant conditioning regimen. In terms of CHIP characteristic, again, we see a pretty profound or apparent graded relationship between age and the prevalence of CHIP. And about 30% of patients have more than one CHIP. And interestingly, in this case, PPM1D stood out as the second most gene mutated, consistent with the prior observation related to therapy-related CHIP. And this is a commutation plot. Now, turning our attention to heart failure outcome, patients with CHIP were much more likely to have heart failure compared to those without CHIP, translating into just heart failure ratio on baseline risk. And again, we saw some interesting graded relationship between the coronal size and heart failure risk. But what was really interesting to me was the mortality. So we turned our attention to mortality of hospitalizations. And we saw that patients with CHIP had a poor survival compared to those without CHIP. And looking into their cause-specific mortality, we noticed that patients with CHIP were at a much higher decreased risk of non-molecular mortality, meaning mortality not due to their primary cancer, translating to 5.3-fold increased risk of mortality. Connecting to their cancer risk, the group by Dr. Muredach actually leveraged similar cohort of lymphoma patients being treated with transplantation. And she then noted that these patients not only had a cardiovascular disease present, but also had a much higher incidence of Beyer-related myeloid neoplasm following transplantation, again indicating CHIP as a pretty profound risk factor for both cardiovascular disease and cancer. So in summary, I hope I convey that CHIP was highly prevalent among patients with multiple myeloma and lymphoma undergoing transplantation. And pre-transplant CHIP was significantly associated with the risk of cardiovascular disease and heart failure.
as well as the risk of therapy-related myeloneuropathy. And lastly, worse survival after transplantation. So then where do we go now here? In terms of the future direction, our lab is actively looking into the clonal dynamics since we saw the association between clonal size and the effect size, and what is really defining the clonal dynamics post-transplantation. Also, it's important to look into gene-specific mechanisms, leveraging the multi-omics strategies as well as other translation models. And we should up there, we should look into therapeutic applications to reduce their risk. And lastly, it would be important to apply to see what are our findings, as well as other people's findings, can be applied to a broader cancer population. This is the end of my talk, and thank you so much. I really appreciate my mentor, colleague, as well as the clinical research team. I love this research. How about thank you?
Wonderful.
The next up are the first speaker of this session is Kamdar, who's conducting Cardio Oncology. Yes. She's an assistant professor and physician scientist here at the University of Minnesota and Rose had previous. Doctor.
Thank you, Dr. Blaes, for the kind introduction and the opportunity to present today. I have the formidable task of going after diabetes and diabetes, and to also get you on to your break on time this time. And so I'm going to talk to you a little bit about biomarkers and mechanistic understanding of reverse cardio oncology components, which this work is done by investigators here today. And as our term more reverse cardio oncology really focuses on the bidirectional risk associated with cardiovascular disease of those that affects in cardio oncology. And the cardiovascular disease and cancer are leading cause of death in the United States as possible, right? And heart failure patients and heart failure cardiologists which religious that heart failure patients have 70% increased risk for disease compared to those without heart failure. I think like that underscores the importance of understanding how cardiovascular diseases result in recurrent disease, especially, for example, type mutations. Is that convert infection risk of the colon cancer in patients with heart failure who have type mutations as well. And so they both have shared risk factors, including inflammation, and it's the identification of bidirectional axis to cardiovascular disease is possible. It's very crucial for understanding the role of cardiovascular disease in development of malignancies. So one of the areas that's been highlighted is targeting inflammation as a bidirectional pathway. So looking at the CANTOS trial, which looked at interleukin 1 beta as a way to treat cardiovascular disease and also potentially reduce cancers. In the biomarker, and this particularly looked at the one interleukin 1 antibody, and it led to less cardiovascular events, but also is identified disease complexities on cancer. Interestingly, biomarker, this was the biomarker design risk documentation in the definition of myocardial infarction into this trial, when it was elevated high sensitivity CRP as a reflection of high interleukin 1 beta levels. And here you can see that there's multiple different treatment strategies based on this, looking at inflammation, whether it's targeting immunosuppression, targeted immune cell depletion, or tumor depletion, and using drugs such as existing drugs to people potentially targeting the bidirectional pathways of inflammation associated with cardiovascular disease as well as cancers. And for looking at reverse cardio oncology OMICs, this is one of the picture that I used publicly this publication, and really just going to focus because you've already heard from me about the role of transcriptomics as well as proteomics and metabolomics and understanding reverse cardio oncology. To start with how the current proteomics studies have been utilized as cardio oncology. So it can just, I was very familiar with a number of these techniques, but just briefly review. Transcriptomics looks at gene transcripts that are quantitatively assessed by RNA sequencing that's facilitated by westernized sequencing techniques that allows for a large number of transcripts to be assessed. And it gives include bulk RNA-seq that's been described single-cell RNA-seq as well as pluripolar RNA-seq. In current cardio oncology, transcriptomics focus largely on circulating microRNAs. And microRNAs compare and study pediatric patients that each match controls to identify microRNAs that are associated with cardiovascular. The challenge with transcriptomics, however, includes challenges about validation of the transcripts, as well as the fact that the transcripts don't always correlate with the protein expression. And so proteomics and bridges that there's thus with the direct measurement of protein and avoids some of the issues that are associated with RNA transcript, which can have a discordance with the protein expression. Plasma proteomics of cardiomyopathy cases in first evidence cancer actually goals without very balance shows that immunoplasm was differentially expressed. Initiation can be a target for therapies as well as understanding risk of cardiovascular and currently collecting creatine about evaluated as a protein biomarker is as well as associated with heart failure as well as fertilization. And so metabolomics is another aspect, and metabolomics includes analysis of metabolites such as amino acids, lipids, organic acid. Metabolomics can be an effective method for checking changes in cellular responses from injury, correlates to change the mitochondrial function. L-carnitine and 19-hydroxy were used to predict cardiovascular peak to rapid prediction rates up to 90% in a study by Beyer et al. And so really, with circulating factors in reverse cardiac oncology, cardiac cells secrete a variety of factors in response to European disease, which have carried effects. Cardiac cells include cardiomyocytes, fibroblasts, as well as other immune cells. The secreting components include extracellular, vesicle, inflammatory mediators, microRNA, and other urocrine factors. Conveniently, these can be measured in blood, and it avoids some of the challenges related to patient tissue biopsies, especially cardiac interrogation. In vitro studies that show recurring factors such as fibroblastic factors, periodicity, secrete growth factor, resistance, and aid blood factor, tumorigenesis are secreted through the heart. And then, so with regard to microRNA has to use extra agents to resistance. It is listed microRNAs were actually upregulated in human heart disease and were also similarly reported as tumor suppressor in cancer subtypes. These cardiac microRNAs to metabolically linked cardiac disease as well as cancer. And so protein biomarkers for heart failure and tumor growth. In this study, you looked at ischemic cardiomyopathy that was induced in the iPSC model, which is a mouse model that has increased lung cancer and diffuse tumor growth. The identified serotonin factors including serotonin 1, serotonin 3, fibronectins, ceruloplastin, and peroxidase 1 that were assessed based on proteomic studies that were associated with tumor growth as well as heart failure. Cardiac gene expression of serotonin, fibronectin, and peroxidase 1 were also elevated in cardiac tissues associated in mice with heart failure. Subsequently, they looked at these candidate proteins were also measured in heart failure patients in the relational method and those in controls with new onset cancer. And they identified that certain K3 cells were elevated, certain K3 levels were elevated in patients with heart failure and exposing certain K3 to the pure bone cancer cells actually resulted in proliferations. So there's a way to understand the use of multiple different omic techniques to be able to identify candidates. Similarly, gene expression, looking at cardiac hypertrophy was explored using a transverse cardiac extraction system and a immunodeficient mouse model. It was used to assess the role of intact wing system results on intact accelerated tumor growth and increased tumor growth compared on non-intact mice. It suggests that the effects are not dependent fully on the immune functioning immune system. We use bulk RNA-seq on intact hearts to identify 120 differentially expressed genes, of which 33 had proteins which were upregulating nuclear proteins. These proteins included cardiovascular and CTGI, and they're also regulating the cancer's progression and were elevated in TACR and in the serum fat animals. And cardiovascular also promoted tumor growth. And so there's manuscript which includes Dr. Moore. They looking at these preclinical models where in heart failure and MI, still TAP role of secreted proteins, including SERPIN-P3 and cardiovascular, which can act as paracrine factors, impact most of the potential cancers. And so, what is the role of risk in human cardiovascular cardiomyopathy and cancer biomarkers? So, biomarkers that are associated with cardiovascular disease and cancer predicted of allied disease in a bidirectional manner. So, using the Framingham study to look at circulating cardiac biomarkers with the CAM proteomics in 5,000 investigations were cancer-free at baseline. These were markers that had achieved likely cardiovascular impact. And the strength looked at strength of association between protein biomarkers and risk of cancer between using a multivariable adjusted study. APG markers are listed here, including growth factor differentiation, stromal cell factors, fibroblasts, factors. These were identified as significantly increased risk, these identify gastrointestinal and colorectal cancers as well as cancer-related death. And so, OMIC studies have provided the valuable mechanism for understanding cardio oncology as well as biomarker discovery. In regards to cardio oncology, biomarker discovery has as important implications to therapies as well as nucleotides. I think really what this group here is in being really just all to actually could feel. But really, deep the anti-aging models, human iPS study lysosomes, cells, the animals, three clinical models, as well as patient multi-element integration. And the development of longitudinal and local recognitions with biomapping samples at baseline clinical cardiac events occur, and then following up through what has I think really the key part of this cloud efforts here is interdisciplinary efforts to keep multi-specialty disciplinary oncologists, scientists to address cardiovascular disease, cancer, pathways through models, patient samples, and integrating these with AI and other features going to play that. And
Time for some questions.
Yes, Green is help. Okay, so if you reach out with your machine is further than that. Watch this civil studies shows that mutation is only occurred to 2 or 53 more like that shit that is responsible to 2. Right? Yeah, something. So in your data, do you see any is a correlation to the baseline what you can do here? You also have to observe the cancer happening and how regular see cancer.
That's definitely a very interesting question that we're still on. So for those studies, you see longitudinally collect examples. So we currently have up to 2 years examples every is true. You have this mutation, you just have a or that stage.
Yes, thank you.
About Colombia is captions and broader recession that it corrects the work. So I'm interested in one of the really data on whether this
I'm just to pull our look for us. So it's the LSD or okay.
and more secondly, sense of what the signals that are a little bit about screening for these is a particular signal
Like this. Here, check.
So they not working with the same medicines, and they're interested by major differences from their analysis after the analyzed surgery device, but also after arteries ligation. And I think that one of the major symptoms to compare is the ischemic event. We had a really short ischemic with our and the ischemia. And I think that as well as some of the studies that are being related to circulation, they acting in concert. We've had a very hard time getting it down to one thing. So we tested various pathways that we see being upregulated and to that it's a complex injury and a complex shock to the system that is Eisenberg's. So we're still dissecting.
What's up? You still lecture, Alice. This is really fascinating, so thank you all the speakers who are here with you. I have two short questions. One of them thinking about the fact that the process was initiated by either malignancy or by hepatic cardiovascular events. What are your thoughts about reversibility of those steps? And then the second one would be, what is the link between maybe ischemic or other cardiovascular events and as a sequence?
I will answer the second question first. So there's actually a recent paper in Nature Medicine by Hossein that basically looked into this bidirectional relationship between the cardiovascular disease. And in his research, should lead to peripheral artery disease as measured by the ultrasound of the femoral artery. Cardiovascular disease and the cardiovascular risk itself doesn't make a close show. So it seems like at least the current evidence is pointing towards more unidirectional relationship in the context of CHIP, where CHIP is causing CVD, but CVD itself is not causing CHIP, but it is only related to CHIP.
And maybe reverse that if CHIP gets to
Reversibility, I think, I'm not so sure whether we can reverse it. There are a lot of theories regarding why CHIP is more common among the cancer patients, just because it confers survival advantage and selection for these clonal mutations. So I'm not so sure whether we can reverse it per se, but there have been some efforts in terms of selectively targeting these clones to kill it. But I don't know, and it's all the work in progress at the moment.
So, yeah, I'm going to follow with that. Repeat that question. I think I was wondering, Kathryn, more if you had any comments about the reversibility of education.
Yeah, I think this is a very interesting area, not just for the changes that we're seeing, but also in some of the data on innate trending. Because I think we need to start thinking about comorbidity patients and how these things may be impacting the immune system globally to alter something in some sort or disease habits. And there are several groups that are looking to develop nanoparticle therapies that can target the bone marrow to reverse immunosuppression using known factors that can induce training, and then vice versa. And it will be very interesting to see where this progresses in the future, as it's one thing to predict the medication replicating response, and others to show it in models relevant to humans.
Yeah, one of the previous question about CHIP, the relationship between CHIP and cardiovascular disease. I think the evidence that CHIP causes cardiovascular disease comes from mice, where they did test computation, they published, show that there is increased cardiovascular risk in mice, but nothing in humans. So I don't think there is enough evidence that CHIP causes cardiovascular disease. Going back to the question from before, one way of doing it, because I think, is that CHIP causes increasing inflammation. And that's the consequence of increasing inflammation and the neurovascular disease. If you treat those patients with adequate function drugs to reverse cardiovascular disease, instead of targeted intact mutation with reducing inflammation.
Yes, so again, going back to this paper just came out, right? I was actually more on the candle chip as a biomarker rather than it directly contributing to the risk of cardiovascular disease. But that Nature paper, Nature Medicine paper that just came out recently was really shows a interaction relationship, because what they did is they recruited patients without any disease. So they only looked into de novo development of peripheral RDCs at all the patients who had shed before. And then they showed a direction of, I believe this is the first data that showed that patients that shed actually reduce the risk of cardiovascular disease beyond the amounts data been getting. That being said, I truly believe that inflammation and at the core of these shared disease risk factors. And I believe there's actually phase 2 clinical trial going on, and some people here may be involved looking into targeting inflammasome to see whether that is could potentially reduce the risk. But we're still at the inflammation that stays in Europe and inflammation.
It still doesn't mean that it causes cardiovascular. Exactly, so it doesn't mean it causes.
I think the causality is hard to establish, but there's a direction.
So Adam's last comment, question?
Yes, first of all, for me, great thoughts. So, thank you. My question is to Kathryn, which is looking at the viral response, the file, and with the receiving therapy as breast cancer patients, so changes that you when they have to the more complex radiotherapy is the FACC, because that's also changed, but that's probably I think it's a study. Chemotherapy will be used.
Yeah, so there are a lot of interesting scenarios to talk here. That's the main tip. And we're just trying the best. This has been a really wonderful discussion, and I think we're going to go ahead and take a 10-minute break. So we'll plan to meet back up at 10:50. And thank you to all our panelists. It was great.
Fight there.
Okay, so my name is Isabella from Academy Cardiologist at University of Iowa. It's my favorite to start the next session, which is focusing on jet setting, getting insights, risk engagement, testing speed does not jump on response, Reilly, who is an professor of medicine and director of the early Cardiovascular CAD at University. Muredach is a very well-known team that offers courses testing the biology, with many important contributions to the area of calculated genomics and genes.
Thank you so much. It's a real pleasure to be here and to see some people. I mostly interact with people working in areas that I don't know how, so it's really exciting opportunity. This is a little bit different in that it's focusing on what we can learn from cancer biology to understand perhaps better and not that's by afterwards. So some ideas like this process. So we all kind of know the pathophysiological mechanisms that are described for decades. Right. So the risk factors that drive activation and immune recruitment stuff, having diabetes is the bit part of that. All science. But with pulse-cell reaction from the media, squamous cells differentiating phenotypes, migrating, proliferating, form, seemingly among cell tissue in the virus. FACC cells use those platelets and all this stuff major system roles as well. And batch check your status like condition, diabetes, more time occurs. Big talk focused on logic disclosure application. We talk about Chun-Wha is now becoming dogs, and I think believes that also, and the predominance of from human genetics all the way for instance, driving what we call this pretty exciting stuff that we think of the potential interventions and context residual risk. And we have all these pathways around actually So human genetics has taught us an awful lot about the potential pathways involved in our cardiovascular. We now have over 1 million participants, very large in the genetic study of GCOS textbooks with a better college recently. There's over 300 CAD loci at June-Wha significant stuff. These are all frequencies replicated in pretty robust ways. Now 50% of these are not associated with traditional factors, which means that a very substantial part of genetic drivers throughout the cardiovascular. It's not really true risk factors we don't actually kinase. That means there's a lot to be done with that. So we're kind of rewriting the texture to it. So most of these loci contain genes that we have not indicated as causal because we haven't worked out mechanisms of function to cell-tissue interactions for these loci. Most of the vast majority are as well. There are some patterns. So, for example, highlighted in red is there's a greater enrichment of genes at these loci for speed muscle cell express genes, probably than we'd expected, recapitulating and bringing us back. This still goes to the hypothesis that also true for lots of years ago. It's true for endothelial cells and immune cells too, perhaps less for immune cells than we had hoped for or predicted based on models. So they're all there. Swivel cells came up a very strong player. And these loci, genes of these loci, map to certain types of pathways. Now, I'm going to talk about cardiomyopathy. This whole story will come back at the end with cardiomyopathy to this concept of oncology. So we did an animal lab based on our interest in ADAMTS7, a loci gene that we discovered for coronary artery disease, and we did several follow-up manuscripts looking at the mechanism of box models of human epidemiology and genetics, really focused on smooth muscle cells. And we asked the question, really, what was going on with smooth muscle atherosclerosis using modern technology, so genetic image tracing in mice, as well as single cell omics, to understand the extent, the types, and the impact of smooth muscle derived cells. Because these cells stop contracting, lose their smooth muscle contractile markers, and change into other cell types. Well, what kind of cell and what am I doing? This just summarizes the paper from 2020, which illustrates that there is a green marker on the bottom, that by the top here with the white, the green marker which highlights the smooth muscle derived cells. And when you look at the top panel of that, significant amount of those cells are specific muscle cells, but there's many other types of glial cells that don't express the muscle markers. And one group is intermediate state that we call seconds because they express stem cell markers, endothelial markers, as well as markers that are not mutually kind with stem cells to their different stem markers, but they have like characteristics of proliferate. And they have differentiated the genes, but they have turned on some genes that are expressed in inflammatory pathways and fibrotic matrix pathways. So a series of work that we studied a lot, and then multiple other groups. We now know that this cell state, this phenotype transition, is reproducible in multiple mouse models, and involves this transition to an intermediate state that's regulated by several genes. The pathways that PDGFR, GCOS, we demonstrated called retinoic acid signaling, which actually we not, for example, back about 40 years is the first word suggesting retinoic acid signaling regulated cytoplasmic in the fact, but really the novel and the cognitive in vivo modern technologies. So we began to ask the question, really, there's a lot missing here about the regulation, the transition of the state of different cell types, and what's actually going on with these cells in vivo in the box models. And because we're interested in trying to trace the cells genetically, lineage trace them, we do this work in box models. We do a lot of work in human genetics and human context, and you can use this to humans, but it's more challenging. So I want to focus on some mouse work. One of the key observations is that in atherosclerosis, in all cell types, there's a remarkable amount of early and extensive DNA damage, excision around the area, breaks, etc., that are going on. And there's an enrichment for this process, and we will solve the direct cell shown on right here on our manuscript several years ago. So it's DNA damage. At the same time, there's evidence going back to the 1950s in oncology, more recently using Reilly type approaches for lineage tracing for clonal expansion of CUS cells in atherosclerosis. So what is an aneurysm disease? Small molecule expansions in the cell and this DNA damage. So what does that mean?
to DNA damage, tumor activation pathways. So this is not a new idea, but it's never really been systematically studied, but it goes back to considering others were looking at these observations years ago. So we've done a number of things prior. The first was to look for evidence of single cell profiling to infer CNVs, which are copy number variations, which are genetic changes that occur in the DNA. And in this case, if you have conjugates, they are smooth to line cells, to market increase in CNVs to bites in the mouse region over time. We looked at multiple human lesions that brought advanced artifacts, and we see the same patterns to name it. Plant cells are third smoothness cell derived. There's normal smoothness cells that individual pelvic cell types. And interestingly, in humans, these copy number variations overlap two types of genes most. The number variations has to mechanisms and the contain carnality starts. And so, for example, KRAS is an increased copy number of many genes when we observe it at a CNV level. There's other evidence for genomic instability with micronuclei in the cells to isolate the smooth-derived cells with the smooth. You can separate the smooth-derived cells through the flow cytometry of green combined with some of these markers I mentioned earlier, and they'll mark these types of cells. Similarly, these have a sustained breathing capacity. In fact, these isolated cells, we have not found that they stop growing. They're still enough size of hundreds of sizes at this stage, whereas smooth muscle cells that don't contain these markers in a separate read beside this bony curve and they're not applied. And there's an interesting thing about when we take all the work we've done on isolated smooth muscle cells, what does that mean? It means just a mixture of cells signaling in those cells. You separate up dinosaur, broader types, their behavior. Similarly, in culture, these cells are resistant to senescence. So now resistance tested. And we've seen this in vivo as well when we did the youngers age the spotted mice, medial cells, who was talking videos, blind cells. After showing on the right here, have greater senescence markers, even though the need deficits and strong so loss of senescence. Using that pathway program called Ardehali, which is to you apply this as one approach to assess formatic reactivation as the pathways in smooth muscle-derived cells versus smooth muscle cells. And in each case, we found activation of multiple tumor-like pathways. We validated these by phosphowesterns, and we also looked at immunohistochemistry for human lesions and chronic plaques to demonstrate these are activated extensively. Well, not all, the majority, right there. But, all these features being turned on doesn't mean it's kind of calls for it to be driving the disease forward as we first. And so do these activities actually monitor that both servers and evolve? So we took two approaches to this. We took the KRAS mutation, which is known to the cause CAD. And we can express it exclusively as smooth muscle cell model. And to see if that will modulate the smooth cell behavior and affect the disease. Affecting the hypothesitic group, disease to smooth muscle. This mutation because it's common in cancer P with one of the areas of overlap CFAs only find an increased copy number containing KRAS copies. And three, there was one high risk with that logic activation analysis. So when we did this work, summarized here, there's found that rapid acceleration of the disease initiated with greater amounts of DNA damage in smooth muscle cells early, at least before actually form Maedeh, and then to increase migration of cells, the formation of more complex vision, larger plaques. So at every stage of the size of the copy marriage of the wildest type voltages of module and these have accelerated in its kinds of diseases that are dominated by smooth muscle well with the complex visions of optimal solution. On the other hand, it can be modulated in the other direction. So we decided to look at the available drug inhibitor, which modulates DNA damage to get about blocks. And this should actually induce apoptosis for death
by a number of factors, T cells that are sensitive to the need for their pathogenic cells should be. So it would disrupt these cells proliferating, surviving proliferating by hypothesize that would probably reduce the formation of plaques to a good cause other things like this. The bottom line is, in this experiment, looking at progression, but we look at this in regression also. This drug will modulate or reduce the size of the atherosclerotic lesions, reduce the single-cell-derived content of the lesions, increases fiber, and reduces that chronic core, suggesting that this drug in anti-stroke systemically given to affect all cells and it affects historic mediators to the cell modulation. So in summary, there's a large phenotype switching of single cell anti-strokes, but you've known this, so we already know what these cells actually do. So we've got a single cell and make specific knowledge. If multiple tumor-like peers, like both DNA antigen pyramids, the ability to put the hallmark, it's sustained proliferation resistance. That's why there is disconnect, we're not sure yet. And they also form common links to have invasive capabilities, have cancer stem-like markers, the initial published checker, and then aggregation will be out of this. And we choose mutations into most sites, like also gas will be accelerated. Also, but if we block their axis associated with being damage, we can continue their modulators. So there's hard benefits associated with this in our thinking that may impact early in the year or we're thinking about only about suppression of general activation triggered in the pathways in the best of all that can speak for cells in this instance. But also the idea that we may want to consider precision medicine concepts there to identify what changes are occurring in individual stages. And there's a number of groups that are working actively on identifying what studies cells and other cells, but particularly with cells, in addition to macrophages which shift other blood-derived cells, which move us cells to have actual mutations, casted by the mutations in resistance. This has been described in other somatic tissues. And so in addition to seeing these, doing all the streaming of other symbols contain somatic mutations, if that is true, then we want to then pick up robotic mutations at an individual genomic level to choose therapies to target and a medicine way. Like to acknowledge all people and all cameras worked in this on why we say and first to all are the first out independent lab valuable. These young superstar analyzed and he really had a lot of creativity in this work and I think with Harry some of this work for being this study or Bitcoin. Yes.
Shocking. I saw that. And we something and your person is Sunday Bastia. I would hot shit and the fish. And what's the thing that night? Now I'm getting for kind of bitch. So today I want to discuss about so it's the stage, but I think this is like I said, it's a logo, but I think it's so it's there's every time we just look at higher works, we need a case to do it, have a certain kind of disease. And everybody knows this disease is disease. But also we talk about cancer that. So cancer therapy actually is but actually cancer therapy won't try to increase aging process because obviously they try to keep the cell cycle overshoot cell mutilation. And actually that causes the aging process. So if that case, if the aging is an important cancer and chronic disease, that's kind of strange thing happens. It's kind of called. Therefore beginning to see what aging is important. So this is the story about aging. Is aging process is what is increasing in children, that senescence, that increase cells like risk, and senescence service to phenotype. And actually cells like risk is also marked of senescence. This another point of senescence. Stress can be used as also senescence because stress is senescence.
and this is caused a cannabis damage response. And this is also can cause a suicide risk S2. So therefore, suicide risk is a hallmark of senescence. But this is concept challenging for these things because it's didn't know that renal cancer will be caused recurrence of disease. Why renal cancer that be successfully caused aging process, but the cancer can be recurrent or resistant? This is because the senescence associated with chronic happens and that cause senescence associated even though the cell is old, but it can cause a stillness. And this is happens why because and then therefore cancer cells resistance to the cancer therapy. And also it's points happens. So this is a new concept. So therefore the senescence concept is GDL. And then what we found here was I don't have time to explain. We found chemoradiation increased white directly to mitochondria and to increase the acid group by F2O2 because the acid reduces to kinase that is group like this. That's one of the kinase factors. That's the decreased transfer H and then decrease in F2 is very important anti-cell transfer factor. And increasing this then outside increase therefore mitochondrial specific catalytic activity damage. And this damage increase population and therefore increase in feed and cap up mitochondria. So this is called the nucleus mitochondrial positive feedback loop and called the persistent activity. Because persistence is important to show that change the phenotype of cells and that can be cause cardiac stick to longer. And we see people information states and then we also utilize some IPSC this several this species by Ardehali is Southeast Asia, and we can use it about 47 different types of on top of it at the same time. Also, based on this theory, we can determine each single cell level of protein is exercise. And then based on these studies, we found very intriguing this phenomenon is this is basically compared to p53 and PSC. p53 as is found very famous to suppressor, but only the p53 also carries this patient. And PSC obviously patient one. And then you can see, for example, this group one calls p53 is very high because it's reason because p53 is suppressor. And you also see this group of p52 and just high. So therefore, this also makes sense. But very few cells, this people group 2, 23 cases in the range of the middle. So this is we cannot understand what's wrong. And I don't have time to explain anything, but we found it. This is thickness cells, and then this is actually is very quite is mutations, this is decreasing, and then this bleed has resolved the part of senescence and closely to decline. Because we know atherosclerosis is a senescence, and then the senescence actually decrease pulsation. But based on this, we can sort of resolve that part even though all cells can be pulsated. And next is a pointless age-old findings. Talk to CBE and what levels of cancer. And then we try to deal with these two. So what's the best we try these two things? Only one thing I can think about is exercise. Exercise is always key for aging process. Exercise also can decrease cardiovascular disease, and exercise also can be cancer. So we need to find out why this current exercise actually is important for OBC. We can use the current data load. And then this is actually is the way how exercise can contribute the cancer prognosis. So there's exercise actually making or worse. And actually that's evidence what I knowledge. And what the people think about exercise increase TEH, exercise increase M1 phenotype, and exercise promote NK cells. It's saying exercise in tumor microenvironment increase inflammation. Okay, that's why exercise can be good for cancer. Okay, wait a minute. It might be good for cancer, but what's going on with cardiac disease? If increased cardiac inflammation should be bad for cardiac disease. Why this happens? Okay, therefore we try to find out how exercise affects the environment. For that case, we use this myeloid cells lying at downward, and we know that this is based on the development sports exercise model that decreased as a tumor size. It's a very important model. And we use a single cell analysis, and we found this site that actually cycling age and industrial cycling age is cells increase by exercise. Okay, it's an inflammation. Okay, that's fine. However, one thing we get is also increased immune cells. So after exercise, what happens is NXC talented strong associated macrophage and also the cells also heavy expression NXC. This is increased, but also this increased oil. And it's like strong associated macrophage decreased. Everybody talk about NE2 macrophage on dicombat. So therefore, undergoing NE2 equals to decrease as increase function. But remember, NE2 is not only inhibition, NE2 also total connects to that's into these relationships. So therefore, we check it. This is work is a virus. This is a whole tissue macrophage. It's surprisingly, even though NE2 gland has unmodified decrease, but inflammation marker also everything is decreases at this level. So even though the entire line is that cells are increasing, but also see all inflammation cells increasing. This is kind of very interesting. Okay, then we go back to Earthline. Was a bit not price analysis question, and we found Earthline is analysis position is see that decrease exercise. Also, we isolate overall macrophage from exercise wise. We do see Earthline 6 position see that decrease, but this is quite immense. And also we use Earthline this specific optimize was a decrease. And using this bias, we know that Earthline is optimized category Cardiovascular because it inhibits inflammation. So therefore, we was. So if inflammation is a key, then when inflammation is important, Earthline 6 should increase the cancer is however, what happens is we see Earthline is difficult to optimize music and check the cancer. But we found this cancer point of actually see if I decrease. So and then we found this studies. And then this is actually one of the summaries. So we found our cells to increase exercise. It means CI increase and also very interesting don't CI, the CD4 is there that the awkward CI content is actually seen that decreased by exercise. And we also saw that the Earthline 6 mean this exercise effect. And also, if you combine that, this is a big actually decrease. So therefore, it's Earthline 6 looking for extra use. It looks like process increasing, and then also Earthline 6 is you must also see it looks like increasing. However, we should combine this decreasing and over see that as a breaks down to both sides also. And remember, this is don't initiate also is that immediate point inhibitor is one of that OMICs. So this is kind of similar. So Earthline 6 solution is actually to increase from limits. So if we inhibit this, that's increase the phagocytosis. If phagocytosis is increasing, actually it's that increase anti-representation of NSC, and therefore it's pathophagocytosis and the cells of the C. And actually this increase that T cell activation and cold cells cancer cells. So therefore, we think stress-induced excess of accumulation of cancer that's taught by and that is systemic substance point. And this is also called cardiovascular subsistence, and also this causes the phagocytosis antigen to decrease. Therefore, this truth is important and it causes that thrombosis. Therefore, to target this necessary so that we comment on exercise or blocking Earthline 6 can be target. And then this not solely depend on inflammation. Therefore, we think is that critical for disease and cancer collections and the target is that selections associated to decline can be target. And ideal target or the time of and it's a chat, so it's really important and showing open Masaki to all the job and all the support for my creators and also the exercise study for the shoulder and courage to it. And thank you for this attention. I'm happy to answer.
Thank you very much. Last video research just how the choose of young people is charging us for the University of Minnesota and this is Professor June-Wha knowledge.
Good morning, everyone, and welcome to the beautiful city of Minneapolis. So I'm now going to talk mainly about childhood cancer survivorship and the risk factor that mediate the transition from subclinical cardiotoxicity to cardiac dysfunction. As an introduction, the 5-year survival rate of the cancer patient has increased due thanks to all the advances in cancer diagnosis and therapeutics. But this translates to more than 15 million cancer survivors in the United States, and this number is expected to increase. However, this comes with a price. Especially childhood cancer survivor, one of the price that these survivors pay is the 15 times higher risk of heart failure in childhood cancer survivors as compared to their siblings who did not have cancer. And as we know, 50% of pediatric cancer patients receive chemotherapeutic drugs, particularly anthracyclines, that are known to be cardiotoxic. As you can see from this graph, the cumulative incidence of cardiac dysfunction and heart failure in childhood cancer survivors around 30 years after the cancer diagnosis increases dramatically with higher doses of anthracyclines. So it's known that anthracyclines, such as doxorubicin, are the primary cause of chemotherapy-induced cardiotoxicity. Nevertheless, they are still extensively used in pediatric cancer patients. And we know that doxorubicin has been used as a chemoprotective agent. There is still no consensus about the exact mechanism of cardiotoxicity and whether every childhood cancer patient needs to take doxorubicin and what are the risk factors that can affect the transition from subclinical cardiotoxicity or later cardiotoxicity to cardiac dysfunction. Anthracycline-induced cardiotoxicity can be classified with acute cardiotoxicity, which happens in pediatric therapy, early onset chronic progressive therapy that happens within 1 year of therapy, and late or delayed onset chronic progressive cardiotoxicity, which is very important in childhood cancer survivors given the 30 or 40 or maybe 50 years old length of life after cancer diagnosis. And my lab is interested in this delayed cardiotoxicity. Since not every childhood cancer survivor develops cardiotoxicity, you are interested in looking into what are the risk factors that may accelerate the transition of subclinical or later cardiotoxicity into cardiac dysfunction or heart failure. And one of the most significant risk factors is hypertension. Hypertensive cancer survivor, childhood cancer survivor, who received anthracycline are 12 times higher risk of cardiac dysfunction than normotensive survivors who also received anthracycline. So there is definitely a synergism between anthracycline and hypertension to precipitate cardiac dysfunction. So we wanted to model this in an animal model. We wanted to model a scenario of a doxorubicin-treated cancer patient who did not have cardiac dysfunction immediately with cancer treatment, but after 10 to 15 years, now they are adults. Now they are overcome by other cardiovascular risk factors, especially hypertension, and this hypertension risks cardiac dysfunction phenotype. To do this, we have created an animal model with young mice and injected them with doxorubicin 4 milligrams per kilogram per week, which is very clinically relevant doses in pediatric cancer patients, primarily from liquid tumors like lymphoma and leukemia. Then we waited 5 weeks, 5 weeks in advanced life, like 5 to 10 years to one life, and these mice are in build above age. And when they are adults, we expose them to a second cardiovascular head, and the head that we choose to mimic hypertension was a infusion of angiotensin II, and after this we looked into the cardiovascular outcomes. So the overarching hypothesis of our work is that doses of doxorubicin cause subclinical cardiotoxicity in young mice that leads to overt cardiomyopathy when these mice are challenged with a second cardiovascular hit. And they call this the two-hit cycle. So first, what the CRISPRi dose of doxorubicin does to the animals. First, as we some of the presenters showed that it's very characteristic for doxorubicin toxicity to cause cardiac atrophy or thinning of the left ventricular walls, and this is also correlates with the human scenario. So we see this with this very low dose. We also see an increase of cardiac toxicity markers like AMP and AMP. But with this low dose, we do not see any significant immediate cardiac dysfunction. So ejection fraction therapy output in these mice are still normal. But when we challenge the doxorubicin exposed mice with a second cardiovascular hit, which in this case is an expensive more-induced hypertension, we start to see the combined effect of doxorubicin plus angiotensin 2, as evidenced by reduction of cardiac output, reduction of ejection fraction, and very significant fibrosis and inflammation happening only in the mice that were pre-exposed to doxorubicin, then later developed hypertension. This doesn't happen in either angiotensin 2 alone or doxorubicin alone. Then we thought that there might be other cardiovascular risk factors that might be interesting to investigate. The second project was around psychosocial stress. Psychosocial stress is a non-risk factor for cardiovascular disease in the general population, and it's also a significant burden on childhood cancer survivors. So, in collaboration with Dr. Alexander Bartolomeucci here at the University of Minnesota, we developed this two-hit model that serum doxorubicin dosed at the young age, but then at the adult age to be exposed mice to the chronic psychosocial stress. The results were not that significant as angiotensin 2 induced hypertension. We did not see this synergistic reduction of cardiac output or ejection fraction, but there was a significant inflammation and myocardial fibrosis when doxorubicin was coupled to psychosocial stress. So still there is less synergistic effect for the two-hit compounding effect, but not as strong as angiotensin 2 induced hypertension. So having established the model of doxorubicin plus angiotensin 2 as the most robust model, we want to answer two questions. First, if there's something that we can do to treat or prevent this from happening, and what is the underlying mechanism. So first, coming from my postdoctoral fellowship with Dr. Rhee at the University of Canada is very interested in structural preclinical compounding, so we tried this approach. We administered resveratrol along with doxorubicin during the young age of the mice, and we stopped the resveratrol 1 week after the last dose of doxorubicin, and we want to see whether the production of resveratrol will be carried over to protect from the two. And as you can see here, the resveratrol treatment really protected against the reduction of the cardiac output and all the other parameters that you can look into this in the biology. Interestingly, when we read other cases, to see what are the underlying mechanisms, we saw that doxorubicin plus angiotensin 2 the two-hit causes a significant increase in the number of senescence-related genes that is coming to be previous out the doxorubicin of immunotherapy and induce senescence. But in this scenario, doxorubicin alone did not cause this much reduction of senescence markers that were to bind with angiotensin 2. It did. And with respiratory treatment, most of these markers were corrected. So what is the probable mechanism of that? And another thing we did also is to ensure that respiratory does not prevent the chemotherapeutic effect of doxorubicin, which is usually a very common question when we're using any pharmacological agent to prevent cardiotoxicity is whether it does or does not inhibit the chemotherapeutic effect. So in the University of Alberta here, with the vision of the Ardehali, we get this before a Keung Ardehali model. We treated them with doxorubicin plus or minus this gradual, and there was no detrimental effect on the chemotherapeutic benefit of doxorubicin, which made us very happy that it can have a translational effect. But what is the mechanism? When we were doing one of the studies we thought that it's connected different, like all organs from the microscope, weight, and sweat one. And to our surprise, there was a significant splenic contraction when the obstructive treatment was combined with angiotensin to induce hypertension. And as you see, that spleen weight only was reduced when the double hit happened. And there's very nice correlation between the cardiac output and the spleen weight. And this brought our attention to a very interesting concept which is the cardiosplenic axis, where the spleen may act as a reservoir of immune cells that when it's activated, it may deteriorate the cardiac function. And there's like a circuit with heart function. It was affects the spleen, and the spleen or the immune cell going back to the heart, leading to more inflammation and fibrosis. So I'm not an immunologist, so I collaborated with Dr. Christine from the University of Minnesota here, and wanted to see whether what are the effects of doxorubicin on the spleen. First of all, even before the angiotensin to induce hypertension, and we see very interesting changes in the spleen because of doxorubicin even after the 5-week recovery period. Including increase in PD-1 positive cells, and PD-1 is a marker of function activation. It also increases when aging, especially in T cells. So there might be some sort of immunosenescence when aging through the type of the immune system here. Another interesting finding was the increase in CCR2 positive in specifically in myeloid cells, and CCR2 positive is also known to be implicated in cardiovascular dysfunction. So what is the real culprit here? To know that we did an experiments in RET1 deficient mice. RET1 is a gene required for the maturation of lymphocytes, both beta and T lymphocytes. And unfortunately, RET1 knockout mice have not protected against the combined lymphocytes plus angiotensin 2, so promotes some lymphocyte-driven parameter. When we did the CCR2 deficient mice, we see a very significant protection from the post-angiotensin 2 double or 2-hit model, and in CCR2 negative or CCR2 knockout mice, very strongly suggesting that it's a CCR2 positive cell-driven mechanism. So overall, with this model of DOX plus hypertension, the 2-hit model, what we are thinking of is that doxorubicin causes lingering effects in the spleen, and we can say it's not yet 100% confirmed to the third experiment saying that, but it's our suggestion that it causes some sort of immunosenescence or aging of the immune system, particularly in the spleen. And this is characterized by increase in CCR2 positive cells. And when these mice are subjected to angiotensin-induced hypertension, there is a movement of these CCR2 positive cells from the spleen to the heart, leading to more adverse cardiac remodeling and cardiac dysfunction. I'd like to acknowledge all my 11 members who significantly contributed to this work, my collaborator here at the University of Minnesota, but also my postdoctoral advisor, Jason Blaes, at the University of Alberta, who I initiated this project with many years ago, and definitely funding sources for getting us started with the and then one more do it and doing it as well.
I think we have time for pretty much questions.
Yes, Dr. Questions. Question allows us to hear either the animal models or the clinical studies and correlation between the age of exposure to the outcomes.
We tried to model here the young exposure, so we only used one age of mice, which were like 5 to 7 weeks of age. We have not tried other ages, but specifically an interesting question to try this on younger mice and also older mice and see if the same model where they can click different ages.
There's nothing interesting with the chart. Okay.
The clinical literature usually suggested that exposure to doxorubicin at younger age makes more cardiac dysfunction later, like younger patients are more susceptible to cardiac
Good. I have a question for Junichi and for Blaes together. Quick question. So Junichi, any data that you have on statins or CRP at 5? Studying, yeah, we don't know what studying is really good. I don't have the video.
And to our last speaker, thanks the question, angiotensin 2 mediated by TGF-beta in cardiac fibroblasts. Any work you've done looking specifically at ARBs, or of course, aldactyl spironolactone in your walk? That would be.
Yes, so we TGF-beta, look at TGF-beta as a marker of fibrosis and the synergy with this bit. With regard to the RB, it's definitely have interest and I think it will work. But because we are using angiotensin 2 in the interpretation model, so I thought if we use ERB, it will be a very direct block of the agent that we are using to reduce application. So it will definitely work, but it will work because ERB is blocking the H1 receptor where angiogenesis should work. So we're kind of giving the address and investigators would be very straightforward with Sterling. But that one is very interesting to understand. And we'll this all yes.
I don't know what was.
Thank you. I love hearing research about interventions as a pediatric oncologist in our clinical practice to the last speaker also. Resveratrol. Just tell us a little bit more about that mechanism because it sounds so promising to bring to our and translate it to clinical picture.
Thank you for the question. Resveratrol definitely works on so many targets. One of the targets that we already published is that dostropsin activates 338 kinase, and when the use of resveratrol, this activation was reversed. We have very recent preliminary data that we use specific inhibitor of p38 kinases with glucosamine, and when they use glucosamine, we see also protection of this to help dogs and shams water. So I believe that this works at least in part through inhibition of these 38 variants. Let's model.
Yes. Thank you very much. Starting the bone better for this session.
That's right.
Inside.
Good morning. My name is Joe Metzger, the University of Minnesota, Deputy Chair, Session Number 5. Cancer Therapies, Cardiovascular Disorders, nurse. First speaker will be Dr. Maedeh. She comes to us from Lincoln Memorial University, and that is a yeah. And sure, yes.
Hello, everybody. Thank you for this kind of introduction. Okay, so it has been shown that I think everybody gets to know that by now that there is an increase of prevalence of cardiovascular diseases after anti-cancer start. This study specifically that is interested in St. Jude lifetime cohort has shown that there is an increase of cardiovascular damage as well as knowledge from the UNAC 10 years before, well, more than 10 years following radiation therapy or anti-cancer well-being study specifically. So the other study that is also interested specifically in radiation therapy has shown that in brain cancer patients, there is an increase of neurological defect or cognitive impairments a few months following radiation therapy, and these occurrence of cognitive impairments progresses over time and even following years following radiation therapy. For example, in these cohorts specifically, it shows that there is an increase of decreased programmable memory, special memory disorders, attention disorder, as well as normal problem-solving ability disorders. So how does that happen? If we take a dive into what constitutes the brain-blood barrier, is you can see that there is a lining of endothelial cells that is surrounded by pericytes and other astrocytes and other cells. When this barrier of endothelial cells is disrupted, this will lead to blood content to leach into the normal tissue. If this blood content comes in contact with the bones, it will lead to neural injury. So all the neural injury we see following brain-blood barrier breakdown will eventually lead to cognitive impairment. Our lab and other labs have shown that following radiation therapy, the main result is an increase of ROS production. ROS production in the cell, but also in the mitochondrial specifically. Mitochondrial ROS production leads to other damages that we have seen. One of the other damages is leading to nuclear DNA damage, specifically, and other things such as senescence. If we have mitochondrial DNA or mitochondrial damage, this will lead to more likely to increase mitochondrial DNA damage. In our lab and other labs as well, that have been seen that when we have an increase of mitochondrial damage, this will lead to an increase of mitochondrial DNA release into the cytosol, and this latter will actually act as what we call a damage-associated molecular pattern. What does it mean? These molecules act as a damage alert. Like there is a damage in the cell, so it will prepare the cell to actually either repair the damage or prepare to eventually do apoptosis. For this particular one, for mitochondrial DNA, once it's released, it will activate toll-like receptor 9, and the toll-like receptor 9 will lead to activation of inflammatory processes. The inflammation leads usually to reform inhibition or apoptosis. But in this particular case, this leads to also ROS selection. If this damage is progressing over time, the ROS production has become chronic, and we will see other damages that relate to the original damages by mitochondrial DNA. So this led us to hypothesize that the radiation therapy will lead to release of these mitochondrial dams, which in the feed-forward mechanism will lead to the activation or overactivation of the light receptor, mitochondrial ROS production, and ultimately leads to brain-blood barrier disruption, will eventually lead to cognitive decline. So, what we did in our lab, we first established this phenomenon that radiation therapy will lead to mitochondrial ROS production, will lead to mitochondrial DNA damage, will lead to this feed-forward mechanism that we were talking about through TLR activation, TLR9 activation. So in these results that you see in the first graph, we see that there is an increase of microstop signaling, which is the way how we actually measure the mitochondrial ROS production following radiation therapy. We see this 24 hours as well as in 10 days post-radiation. When we actually scanned this mitochondrial ROS production using MTT, we actually induced this mitochondrial ROS production from radiation therapy. And this happens in both times at 24 or even 10 days post-radiation. This is also accompanied by mitochondrial DNA damage. As you can see in the second graph, there is a lesion of mitochondrial DNA following radiation therapy. When we actually retreated the cells with mitochondrial scavenger MTT, we actually reduced that amount, or actually inhibited the increase of mitochondrial DNA damage. Again, we looked at whether there is a release of these mitochondrial dams or mitochondrial DNA fragments after radiation therapy, and we have seen it in these either in vitro 10 days in human brain, like microvascular endothelial cells, as well as in mice. And you see in mice as well as in vitro, when we treat these with MTT, mitochondrial ROS scavenger, we actually reduce that release of these mitochondrial deaths. So, if we inhibit toll-like receptor 9, does this actually prevent the mitochondrial loss direction as well as mitochondrial DNA damage? Yes, it actually did. When we actually use an antagonist for TLR9, we reduced or we prevented mitochondrial loss direction and mitochondrial DNA damage following radiation therapy. And this is also shown in both in vitro and in vivo. When we actually prevented as well as TLR activation, we preserved the endothelial permeability. We actually made sure that these endothelial did not get disrupted, the barrier did not get disrupted following radiation therapy, which shows again that radiation therapy will act through the TLR9 activation to induce endothelial barrier disruption. So this is all in vitro, and we looked at either this is actually true in vivo. So we used C57 Blaes mice subjected to whole brain irrigation, and we looked at their vascular leakage in the brain following 24 hours of immunotherapy therapy with or without an antagonist for toll-like receptor. As you can see in these results, we see more staining, or like red staining, which indicates a vascular permeability after radiation therapy, but when we actually treated these mice with an antagonist for toll-like receptor 9, we prevented this to occur. Okay, this is very nice, but when we actually block a toll-like receptor, we don't want to block them all over the body, because at the end of the day, the inflammation status does depend on these toll-like receptors to actually activate properly and continue to activate here. So we looked at in the solution how can we target specifically the endothelial cells that are subjected to radiation, that are actually damaged. To do that, it is known that endothelial cells, when they are activated in stress or inflammation, it will lead to an expression of antigen on their surface. Alpha-5, beta-3 antigens that are activated, and we have seen in the literature that cyclic RGB can actually bind to these antigens and specifically to laser HTTs. And in other studies, it's being shown that there are usage of nanoparticles, and if you use these nanoparticles with cyclic RGB, you can deliver specifically these whatever compound into whatever antigen endothelial cells, wherever it is. So we use this technology to see whether in our model this is actually feasible. So as you can see, we do see an expression of this antigen alpha-5 and beta-3 following radiation therapy, and this is very specifically in cells. And when we use nanoparticles that are coated with these cyclic RGBs, we see that there is an accumulation of these nanoparticles in the irradiated endothelial cells. So we used this first test in vitro, and we used again same thing, endothelial cells in vitro subject to radiation therapy. You can see that when we use these nanoparticles, loaded or not loaded with this TLR9 antagonist, and you see that the TLR9 antagonist loaded nanoparticles effectively reduced or prevented mitochondrial loss direction following radiation therapy. So, is this true in vitro? Is this also feasible in vivo? So, we made sure first that the nanoparticles can actually deliver or bind specifically on the cells in the brain following radiation therapy. And it is the case. We do see that there is an uptake of these nanoparticles in the endothelial cells in the brain. As you can see these images, the brain irradiated shows a more increased signaling for the nanoparticles loaded in this case with coumarin to show more rapid specifically. So we tested this in vivo. We looked at 30 days following radiation therapy with nanoparticles or with nanoparticles loaded with this antagonist. I didn't specifically mention this, but they are all quoted with negative cyclic. As you can see, the non-practical group showed more signals that there is a vascular leakage 20 days following radiation therapy in the brain. However, when they are looked at with toll-like receptors antagonists, we did see prevention, police prevented the leakage following radiation therapy. Okay, this is basically just to quantify it and confirm that the quantification over at least 4 mice did show that there is activity prevention on this faster probability following radiation therapy. So I'll stop here because I'm out of time, but overall we showed in our study, at these studies specifically, that first we do see an increase of mitochondrial DNA damage following radiation therapy. This is leading to a release of the mitochondrial layers, which is mitochondrial DNA fragments outside mitochondria. This leads to the over or chronic activation of the toll-like receptor. The toll-like receptor line by itself activated will lead to an increase of these ROS infections specifically, which in a feed-forward mechanism will feeding into the stool, which leads ultimately to an endothelial damage, which ultimately leads to organ decline. So where do we go from here? We do need to establish this mechanism. What is the endothelial disruption that is occurring here specifically that leads to this durability? Is it senescence? Is it another apoptosis pathway? Is it another bystander effect that we haven't seen yet? All of these questions remain to be answered, and we are continuing in this path. So for this, I would like to acknowledge all the people that helped and worked on this project. More specifically, the Muredach lab where I was before moving to Tennessee. And I'd like to thank also the sound lab as well as Rajesh for helped significantly in this project. Thanks.
Thank you. I think we take questions at the end of this session. Next speaker. She'd like to introduce herself. She got in the last session. I'm really delighted to introduce again Isabella Carmine. She's the chair of medicine at the University of Iowa. I don't see you bringing Floyd up to Rosedale with you. Unfortunately, they had a tough second half last night. So he used to go for it. Nonetheless, we welcome you and look forward to her studies on radiation therapy and vascular damage. Thank you.
Yes. Thank you very much for the kind introduction. Let me say that 10 years ago, when I became interested in radiation-induced cardiovascular disease, I was told by some experts that we had one expert in the field that this is the problem of the past. They don't know stuff. And just said that this person, unfortunately, isn't getting. Now, I don't think this is necessary. Radiation-induced cardiovascular disease is really a disease of cancer blindness. 20 million cancers are currently here in the US, and numbers are increasing. 40 or 50% of them have undergone radiation therapy at some point due to their cancer. But the increasing survival of many cancers, I believe that the incidence of radiation-induced cardiovascular disease is probably going to increase. Now, the location of clinical diagnosis of radiation-induced cardiovascular disease entirely depends on the location of the radiation. It can take many forms. Some of them we are actually dealing with in Cardio Oncology. Now, mechanistically, you feel good steps. Like as muscle cells are key drivers of radiation-induced cardiovascular disease. This is DNA damage, oxidative stress, mitochondrial dysfunction. Some of the topics we already heard about today. And this leads to renal dysfunction, bipolar thrombosis, fibrosis, senescence, ultimately manifesting as obstructive disease in the vasculature, similar to atherosclerosis, even myelinic, and also hypovascular amplification and vascular disease. Now, somewhat unbeknownst to the Cardio Oncology community, radiation biologists have long taken a great interest in microvascular injury as a determinant of radiation therapy into side effects in most organs. Again, key to this is an immediate effect of radiation therapy, leading to depletion of endothelial cells or apoptosis of cell death, and thus a long back period, and then a late manifestation driven by vascular remodeling and loss of microvasculature, leading to hypoxia, and then inducing many of the side effects we see in other organs, such as lung fibrosis, GI symptoms, kidney dysfunction, and so forth. Now, one of the areas of my lab is to study microvascular disease in the brain. And again, there are 100,000 patients with brain tumors who receive radiation therapy every year, now survive for 6 months or now. And this is important because within this time frame of about 6 months, 50 to 90% of them experience some form of cognitive decline, which can manifest in different ways. And for the longest time in the radiation biology field, there has been this debate as to which extent microvascular damage versus matter loss versus neuroinflammation or damage to oligodendrocytes really drives this side effect of cognitive decline. So one of the goals of my lab was actually to develop a human model of being able to study microvascular disease in the brain. And so the way how we've been thinking about this is to use Beyer, which is the extension of the CNS, and the vasculature is fairly similar to vasculature in the brain. And so we've done a number of studies supported by NIH to develop some methodology to do this, which includes stem cell spectroscopy, which our team might be using to study the brain, and optical immunotherapy tomography and geography shown in the middle, and then tests of vision. And so with these methodologies, we are actually currently concluding a study in patients with uveal melanoma who are treated with radiation therapy, and they are followed up at every follow-up appointment for up to 4 years in the patient blood flow and the loss of microvasculature, shown in the middle of the right-hand side. So not in the body, you can see this dropout of microvasculature by optical tomography and geography. And at this point, we're trying to test whether using laser spectroscopy, which is actually a proof for use in humans, can be used to predict later microvascular dropout and the inhibition. Because the goal of showing that government changes are a predictor of later microvascular. Now, many of these changes can easily be detected in the mouse brain, where with very advanced radiation equipment on staff typically target particular areas of the body during the brain, as laser spectroscopy, and you see a decrease in libido or the radiation intensity within a month, and the battery often reacts. But before using this, we wanted to ask several general questions about the changes of the microvasculature. For this study, we performed a batch of 40 studies. The first one was just 6 months, so 6 months after radiation, 12 gray, all gray, we analyzed the volume of microvascular isolate. As you can see, so there are 400 to 40 dysregulated proteins, the radiate ones on the left-hand side, and with the majority of these proteins were downregulated. And then by mapping these differentially regulated proteins with different methodologies, we always came up with the same results, and this is actually that the biggest changes in the ortho the microvasculature occur in mitochondria. And this is followed by changes of proteins which are related to protein transcription. And so we did another analysis just looking at mitochondrial protein that we regulate ones on the right-hand side. As you can see, all of them are almost 480 proteins, about 80 mitochondrial proteins, and they're all downregulated after 6 months. So this is only a snapshot starting to the experimental studies, but doing particular 10 days, 30 days, the whole year after radiation, just get a feel for how quickly those changes progress. As you can just see, just many very simple. There's the second column, this is he compares to 10 days, so that's upregulation of key pathways, including inflammatory ones, insulin signaling, enos signaling, also mitochondrial changes. So 10 days, that's upregulation, and then starting at 30 days, that's good small column, and then the last element, content everything is. So it really takes not very long for the radiated cardiovascular jet to go to this cardiac change. It actually changes between most different time, but actually very consistent. So to come back to 30 days after radiation, so again, as predicted, we do see a decrease in mitochondrial copy numbers, just suggesting that we have still mitochondria in the microvasculature. And on the right-hand side, the GCOS studies supporting that there's decreased mitochondrial respiration. The mitochondrial is also may decreased glycolysis. So what does this mean? I mean, what does mitochondrial respiration becoming? So we tried to understand whether this is related to the key function of the microvasculature, which is to repair that. So we started with cells related the right-hand side as really greatly increased pH of the angiosperm related. And what we're currently trying to figure out whether we're protecting the mitochondria for a number of different levels, including the iPSC can be obtained. Also try to limit these changes so that clearly CD50 reduces mitochondrial copy numbers, and this also leads to leakage of monomer. So again, we're trying to relate monomer function to mitochondrial function damage. Now to just quickly change is many cardio oncologists deal with patients with cortical stenosis after radiation for head and cancer, and the risk of developing a stroke is about tenfold increased in patients who undergone such treatment. The earliest indicator of impending post-robotic disease for an experimental vascular biologist of which is acetylcholine dependent based damage. This is strongly decreased 1 day after radiation, 10 days after radiation, and also at 1 year after radiation. So this is the very chronic disease and providing the nice mitochondrial scavenger mitotempo completely reverse these changes. Again, emphasizing the role of mitochondria or mitochondrial storage in vascular dysfunction after radiation. Now, radiation will induce cardiovascular disease if only we had the right treatments to do so. No specific mitigators, and a number of medications that we commonly use for radiation have been tested, in particular statins. We did so in our model in which was detected that the administration of statins, these cells, such as Thalassetin, decrease vascular dysfunction after radiation. And importantly, they do such, we do have this effect through their effects. So statins, at least Thalassetin, a few others do the effect. But it's shown here, so it's used much further because the reduction after radiation. Which can make treatment very fast. So that's the last point I want to make is a number of mimetics, such as superoxide, mimetics, and other loss-reducing agents in the pipeline by a number of companies being tested for their effect on radiation-induced lower tissue. So one of them is GCOS or Mozneb, which is currently tested in a phase 3 study, and then trying for its effect to prevent leukocytes appetite cancer radiation. So we had the opportunity to analyze CT scans of these patients for their arthrostotic plaque formation recorded out. So these are 41 patients. Neck cancer, mostly males, 57 years of age, no difference in their pre-existing cardiovascular disease, however, we see this age. And the CT scans were obtained at baseline and then then on average 1 years after the beginning of age. And while there wasn't a difference in the total back progression on the calcium planet, and there was a statistically significant difference and suggesting that GCOS decreases no attenuated effects in additional considerations, suggesting that this agent may indeed also have some cardioverted effects and need to be further investigated. And some idea is simply that TLCs to say variation induced cardiovascular disease in patients working out microvascular models and it needs to be out to 2 years. It's a lot easier to do risky different preclinical models. Mitochondrial injury appears to be a key driver of variation-induced cardiovascular disease, and numerous medications that are familiar with and other agents are in development to be used as medications to prevent cardiovascular disease. I think it's going to be a few years to continue in this stage where medication-induced cardiovascular disease is a thing of the past because it is preventative. And with this, I would like to acknowledge here the number of members who have really instrumental in this. Michelle Templin, Olga Rhee, and Kamdar, like I said before, she mentioned the study is by herself. That was large regarding patients.
Thank you, Dr. Mozneb, for a great talk. Running the Inhibition, please introduce Dr. Clayton, comes to us from the University of Colorado. His work is at the intersection of vascular damage, cardiomyopathy, and satellites.
All right, thank you for having me here today. I specifically thank the organizers for the invitation to speak and provide a platform to discuss how we view cardio oncology through the lens of translational cardiovascular aging. So some of these initial slides have already been described by multiple speakers today. None of this is going to be a secret to this specific audience, but just give you a framework of how we think about going about addressing the problem of vascular dysfunction in the context of cancer and chemotherapy. So when we first got into this line of work about 6 years ago, we started taking a look at the primary cause of death worldwide. As many of you all know, cancer and cardiovascular disease are the top 2 leading causes of death. But we certainly can't look at these two in isolation. The investigating and the field of our research is the interrelated nature of these two concepts. And so if we look at the annual number of new cancer cases, it's steadily rising year after year. It's estimated to exceed 2 billion just this year. And unfortunately, now more than over 600,000 cancer-related deaths. And of these 2 billion new cases, approximately 650,000 individuals are anticipated to undergo chemotherapy treatment. And why that is that you know chemotherapy is the first line of defense strategy against many forms of common cancer. And we got into this line of work, we chose to focus on anthracyclines because of the well-established body of literature, the effect of doxorubicin and other anthracycline chemotherapeutics on the heart. And the fact that these drugs are still commonly administered to many cancers, including childhood, adolescent, and adult cancers. And as we know, doxorubicin is still the most, despite its cardiotoxic effects, is still one of the most commonly administered drugs in terms of chemotherapy cocktails. And what that is because this is already been emphasized, but just to show it graphically, if we look at common cancers treated with doxorubicin-containing chemotherapy, we see a very impressive anywhere from the 50 to 90% 5-year survival rate, given leukemia as well as breast cancer. However, there's a marked disproportionate increase in heart failure relative to the cumulative dose of doxorubicin received throughout chemotherapy. As with a 20% increase in cumulative dose, we have more than a doubling of its heart failure. And that's led the National Cancer Institute, the American Cancer Society, to make statements such as today's cancer patients are as hard as cardiovascular conditions. But why does doxorubicin chemotherapy lead to cardiovascular disease risk? And when we got into this line of work, we noticed that much of the literature was focused on cardiac function, but we were a vascular aging lab. And what we knew was that large majority of cardiovascular diseases occur with aging, and as aging accounts for 85% of all cardiovascular diseases. So we went to the vascular aging literature, or the cardiovascular literature, and asked why does aging increase cardiovascular disease risk, to give us an initial idea of how to go about understanding, perhaps a different way to think about vascular-related cardiovascular disease burden. A key manifestation of vascular aging is a process of endothelial dysfunction, which is our anatomical inability of vessels to dilate when they're responsive to given physiological stimuli, primarily nitric oxide. So we asked, does doxorubicin increase endothelial dysfunction? And at the time, there were clinical observations that this might be true, but we were interested in interrogating mechanisms by which this might occur. And we thought it was prudent to do so because doxorubicin is, as you all know, administered systemically. And around the time we got into this work, there was what we felt a very much a call to action paper published in JACC, basically Translational Science, to say we need to start caring more about the endothelium in the context of doxorubicin cardiotoxicity. So we sought to answer the question, is doxorubicin a model of accelerated vascular aging? And to do this, we employ a series of translational experimental approaches. And the way I like to think about it is if a mouse model is going to be translational, we need to most understand what clinical measurement and clinical implication our mouse measurements are informing. So in the context of endothelial function, the reference standard measures a process called flow-mediated dilation. So we're inducing an increase in nitric oxide by a mechanical stimulus that results in activation of endothelial nitric oxide synthase and a release of nitric oxide. That's done with forearm occlusion with a release, and that release stimulates blood flow and subsequently nitric oxide release and vasodilation. The way we do this in Blaes is with acetylcholine mediated vasodilation. We excise nostril arteries and accumulate that in the pressure artery system to induce dilation. And an important piece I'd like to emphasize from the context of translation is that when we compare young and older mice to young and midlife older adults, we see the same magnitude of endothelial dysfunction. So we think that gives us confidence that what we're observing in mice might be translated to people. And so the first goal about asking is doxorubicin a model of accelerated vascular aging? We employed a relatively straightforward and simple study design with two young groups, one of which received an IP injection of sterile saline or 10 milligrams per kilogram doxorubicin, and at the same time we compared that to a full reference control. For those that perhaps don't study, aging 27 months is about 85 years old in human years. This work is summarized in a review article we published in Aging and Cancer, but you're fortunate to publish some of our initial work in JACC Cardio Oncology and highly prestigious, and it summarizes that work in this review on reference here. But what we found is there are some acetylcholine dose response curves in mice is that the young animals treated with doxo had similarly impaired endothelial dysfunction as old animals, if not worse. And then to pharmaco-dissect out, so use pharmacological agents to dissect out mechanisms, we first asked, does this mechanistically occur as a result of reductions in nitric oxide bioavailability, a key hallmark of vascular aging? And to interrogate this, we used LA, which is a well-established pharmacological inhibitor of endothelial nitric oxide synthase. We repeated the acetylcholine dose responses. So in the presence of LNAM, we found those group differences still persisted, and so concluded that nitric oxide-mediated dilation was the mechanism underlying the impairment in the acetylcholine dose response we observed. And excessive mitochondrial oxidative stress is one of the primary mechanisms that equate to reduced nitric oxide bioavailability in vascular aging. So to sequester excessive mitochondrial reactive oxygen species, we perform similar pharmacodissection experiments, but this time in the presence of mitochondrial targeted attacks and Maedeh to scavenge excess mitochondrial reactive oxygen species. And what we found was when we gave Maedeh to the vessel back, that we could recover endothelial function levels back to young control animals. So similar to old animals, we were seeing that doxorubicin treated animals had impairments in endothelial function as a result of excessive mitochondrial loss. But where do we go from here? How do we intervene? And the way we think about intervening, because we're viewing this through the lens of translational vascular aging, is to do what's called the biological hallmark staging. This is a concept first published in 2013 in Cell, and the same rubrics updated these hallmark stages in 2023. And it's been talked about already today. But one of the main things we were interested in was this process of cellular senescence, highly implicated in aging, and we know it's one of the mechanisms by which toxicity, chemotherapy can suppress cancer progression. And we were confident to move in this direction in terms of our doxil-related work, because we had just published a paper, Hypertension, establishing the role of cellular senescence and satellite therapy in the context of pathological vascular aging. So with that, it gave us confidence that cellular senescence may indeed be a viable therapeutic target for treating vascular dysfunction with senescence. And for the sake of trying not to repeat previous speakers, I'll briefly go through what is cellular senescence. It's most broadly referred to as a mostly irreversible cell cycle arrest, and that can be induced by geotoxic stressors like chemotherapy, or it can just be an adjunct product to the aging to reach age limit. The senescent cells have a discrete secretory phenotype called the senescence-associated secret expression phenotype, like SAS. The SAS can actually promote excess lack of reactive oxygen species. So we think that cellular senescence may actually be the source of the excessive mitochondrial loss or its trigger. But how do we go about targeting cellular senescence? And to do that, we need to understand what's inherent to senescent cells. And one thing that is inherent to senescent cells is an upregulation of the anti-apoptotic pathways. That's what keeps the rest of their cell cycle. And that's an upregulation of pathways like the BCL2 pathway. So we use a well-established pharmacological inhibitor, BCL2, the T263, that also is just in the clinics. And we administered it in intermittent dosing paradigm in mice, intermittent mice, took 1 week on, 2 weeks off, 1 week on. And the idea here is that it's to suppress excessive cellular senescence, as normal levels of cellular senescence are necessary for adequate wound healing and basal oncological suppression, as well as embryonic development in Europe 5. So this is just a common approach that's given for senolytics. And what we found was that we gave intermittent dosing of ABT263 starting 1 week following doxorubicin injection. We could indeed measure differences in one of the canonical cellular senescence markers, CDKN2. And this is an aorta lysase by gene expression. This gene encodes the cell cycle inhibitor P16. We see an aorta lysase that's CDKN2 gene expression is up with doxil armed, but that it's mostly prevented with doxil-treated animals that receive the senolytic. So now that we have proof of concept that we have proper target engagement where we would expect it, we then moved to our mouse model and studied both female and male mice. We didn't see sex differences, so I collapsed the data across both sexes for the sake of presentation. Here's our endothelial function assays again. We see that the doxil-treated animals, the red dots, have significantly inferior endothelial function relative to the rest of the groups. Therefore, the animals treated with ABT263 had endothelial function similar to control animals. And using the same pharmacological dissection approaches, we found that this was nitric oxide mediated, as well as senolytics mitigating the increase in excessive ROS production of vasculature. So I had the fortunate opportunity to present these data at a conference about a year ago, and then establishing friendship and collaboration with Dr. Andreas Beyer. We discussed moving this into his model of isolated arterioles from human adipose tissue biopsies from surgical discard tissue that they received from the Medical College of Wisconsin. So we're excited to move this model to not only a human setting, but also to the microcirculation. So I have to thank Dr. Beyer and his team for helping us move this work forward in a translational sense. And using their model, we were able to recapitulate these results. So using a similar color structure, red showing impaired endothelial function, but this time using a pressure gradient. So this is flow-mediated dilation in these arterioles instead of the sphenoid-mediated. And the vessels that received doxorubicin plus ABT263 had endothelial function similar to the control vessels. And importantly, these vessels were taken from individuals that did not have overt cardiovascular disease. So at this point, we've established proof of principle that senolytics might be efficacious in the context of accelerated vascular aging with docs. 8263 is not clinically relevant in terms of broadly administering this to patients. So where we went from there was to a natural cell lytic. We're just getting a lot of attention to preclinical and clinical settings. It's a cell lytic called the 7. It's found commonly in strawberries, cucumbers, onions, food-derived compound. So we performed an initial screening of human aortic endothelial cells using doxil and then increasing doses of the 7 and using senescence-associated beta-glycosides as the initial readout for senescence. We found that 1 micromolar of 7 can markedly reduce the senescent burden induced by doxil in human aortic endothelial cells, and this is particularly relevant as 1 micromolar is the peak plasma concentration in the previous cell lytic studies using the 7. Again, repeating our endothelial function dose response curves, in this setting, we found that animals that received the 7 after doxil had endothelial function similar to control animals, and that this was also nitric oxide mediated. Prior to this, in parallel, we published a paper in the journal Aging Cell showing that the 7 administration to old animals could improve vascular aging by directly targeting senescent cells in the vasculature, which gives us hope for this line of work. And just to consider some current clinical studies that are ongoing in this line of work, is that FACC is trying to get into cancer survivors with most primary outcomes done from a clinical trials of frailty and distal function. By collaboration with my colleague, Dr. Matt Rossman at the University of Colorado Boulder, we are doing an ongoing study with giving FACC and life adults for vascular aging. And to get the data, it's still preliminary, so I can't share those results quite yet, but we do know it's really safe, well tolerated thus far. And then I recently started my laboratory at the University of Colorado Institute's Medical Campus, and we're hoping to translate this work in what we're calling a SENDOC study, titled Cellular Specimens and the Influence of Doxorubicin Therapy on Cardiovascular Health, where we're creating downmental lymphoma survivors, as well as agent sex match control, and looking at vascular function in various senescence markers, as well as other confounding variables that we're doing today, such as social determinants of health, psychosocial stress, etc. With that, I'd just like to extend the last slide thanking the team at Colorado Boulder, the Buck Institute for Research on Aging, which really got some of this life work, and they're one of the major players in this field. Our NIH funding sources, as well as supporting Dr. Brandon Wynn, who just joined as postdoctoral fellow at the University of Colorado in Chester. Right here.
Don't worry, organizers, do we have time to take your questions? 1 or 2 questions, if you'd like to come on to the microphones. Maybe short questions, short answers. Yes, okay. So, in about symbolizing PCO2, I follow very interesting. So, in that case, it's a key PCO2, maybe this misfolding happens, but the prognosis is increasing. So, do you see that the prognosis is increasing is cloned out? So, mental or help you prognosis. And so, yeah.
So we do see apoptosis. We do see it in a somewhat limited time frame, within the first few days after radiation, and then it disappears. And then we do see senescence in the surviving cells, and that we contain great peaks, but it develops slowly within a few days. So I think that both mechanisms do play a role in this, especially at different time course before each of us. Right.
I was curious regarding the outlook for the tumor microenvironments that you checked. Anything?
I think I understand the question.
TR, the TR gets manipulated that tumor microenvironment and tumors. How do the tumors develop? Do they grow? Do they shrink?
They would grow. Actually, there is a study currently targeting delivery of TL9 agonist to to decrease the resistance of the cancer cells to basically improve the cancer therapy. That's why the usage of nanoparticles in this particular case is to deliver them specifically to the epithelial cells, not to the cancer cells. And that's why I emphasize to the fact that we want targeted delivery, not all over the body delivery.
Great. We're going to move on to study number 6. Please join me in thanking these 3 great speakers. How about? Great. Okay, let's start the next session. So, as we don't have much time, so I'd like to make short introductions. So, the first speaker is Dr. Keung, a physician scientist and cardio associate physician at Keung, and assistant professor of medicine at Harvard Medical School. And her talk is genetics in cardiotoxic risk stratifications in Cardio Oncology. Please come. Yes. Yes.
Great. Good afternoon, everyone. My name is Keung, and I am from Reilly Hospital in Boston. First of all, I'd like to thank the organizing committee for the invitation. So, as a physician scientist, our laboratory studies genetics in cardiotoxicity that are associated with cancer therapy. And today, I hope to be able to provide an overview of the genetic contribution to cardiotoxicity that are associated with the cancer therapy. So let me start with a brief patient case. So this patient is a 33-year-old woman. She presented with a 5-day history of difficulty breathing and also fatigue. Her past medical history is only notable for osteosarcoma, for which she has received cancer therapy in addition to the resection of the tumor. And cancer therapy regimen included cardiotoxic doxorubicin. Our physical exam and laboratory test results suggested that our patient has a new heart failure. As a part of the diagnostic evaluation, our patient underwent echocardiogram, which is the ultrasound of the heart. And you can see on the left, which is the current echocardiogram, the left side of the heart, sorry, I apologize. The left side of the heart is right. The left side of the screen, the left side of the heart, actually the function is quite depressed compared to the echocardiograms on 9 months ago, which was prior to the chemotherapy, that she had a normal contractile dysfunction. Therefore, our patient, based on these symptoms and the data and findings, she was diagnosed with a cancer therapy associated with cardiomyopathy. So as many of you know that many cancer patients undergo cancer treatment, and many of the cancer treatments are cardiotoxic. But not every patient who receives cardiotoxic treatment develops cardiotoxicity. So why is that? Why is only a subset of the patient population develop cardiotoxicity? And in addition, the recovery differs. So some patients have cardiotoxicity, and fortunately, majority of them recover their cardiac function, but there is only a subset of the patient population who do not recover cardiac function. So what contributes to these differences in each of our patients? So many studies over the past years have tried to identify clinical risk factors that are associated with cardiotoxicity, which includes pre-existing heart disease, previous or concurrent cardiotoxic cancer treatment, extremes of age, high doses of anthracyclines, and traditional cardiovascular risk factors such as hypertension. However, when risk prediction models were developed using these clinical risk factors, that actually the predictive value of predicting which patients will develop cardiotoxicity was rather low, as shown by AUC value of 0.68. This led to a hypothesis that to genetics actually contribute to risk of developing cardiotoxicity to cancer therapy. Thanks to efforts led by many of the investigators, and I know many of them are next to present in this room, we now know that genetic variants contribute to pathogenesis of cardiotoxicity that are associated with cancer therapy. Additionally, we also know that genetic studies actually have helped us elucidate the mechanisms behind the enterocyte-related cardiotoxicity, such as reaction oxygen species production, mitochondrial dysfunction, DNA damage, and disruption in iron homeostasis, as actually shown by Dr. Ardehali, and also the sarcomere disruption. So genetic variants are often classified into two types: common genetic variants and rare genetic variants, and they are often based on an impact, which is on the y-axis, and the under frequency on the x-axis, telling how prevalent are these variants in the general population. So, for example, common genetic variants are those that are present often in more than 5% of the general population, and they tend to have a low impact. And they are also known as common SNPs, common single nucleotide polymorphisms. And based on the studies, we know that although each of these common SNPs do not have a significant impact, as a whole, if we combine multiple of them, can have a clinical impact. Then some of you might have heard of something called the polygenic risk score that can be also used to predict the risk of developing coronary artery disease. Conversely, rare genetic variants are variants that are present at a very low frequency in general population, but each variant has a very high impact. So, for example, a rare pathogenic variant in a gene called MYC7, myosin heavy chain 7, leads to hypertrophic cardiomyopathy. So, two common genetic variants lead to cardiotoxicity that are associated with cancer therapy. GCOS studies, June-Wha association studies, have identified that in cancer survivors with and without cardiotoxicity have demonstrated that variants that are common among this population in many genes in various cellular pathways influence risk of developing cardiotoxicity. So, for example, a common SNP in gene Has3, which is the hyaluron synthase 3, results in significantly increased risk of developing cardiotoxicity, as demonstrated by odds ratio 57. Similarly, a SNP in another gene called CEP4, which is involved in mRNA splicing, if a patient has a particular SNP within this gene, actually it increases the risk to tenfold higher than other people without this SNP at developing cardiotoxicity. And a recently published study by Dr. Paul Burgess group, which will be in this audience, has demonstrated that genes actually where these common SNPs are located in, and they play an important role in cardiotoxicity. So in this paper, the authors actually used the iPSC-derived cardiomyocytes as a model system, and they have demonstrated that cardiomyocytes lacking these genes that are identified in GCOS studies, they are more prone to develop cardiotoxicity. So for example, in this plot, they have demonstrated cardiomyocytes that are lacking genes such as Has3, they are at higher risk and they are prone to produce more reactive oxygen species upon exposure to doxorubicin. In addition, these cardiomyocytes lacking these critical genes, they also are more prone and susceptible to developing DNA damage upon doxorubicin treatment. Next, how about rare genetic variants? Do rare genetic variants increase risk of cancer therapy associated cardiotoxicity? To answer this question, our group decided to perform next-generation sequencing analysis of cardiomyopathy associated genes in patients with cancer therapy associated cardiomyopathy. And our genetic sequencing analysis revealed that titin truncated variants, which is the rare pathogenic variants within the essential gene, the sarcomere gene, is highly enriched in cancer patients with a cardiotoxicity. So in this table in the red box, that I noted that in patients, about 8% of the patients with cardiac toxicity carried a pathogenic variant in gene titin, which was significantly higher than less than 1% of population in the general cancer population, suggesting that having these genetic variants in gene titin increases the risk of developing cardiac toxicity. And this was consistent with what we know about this essential gene, the sarcomere gene titin, because a pathogenic variant in gene titin is the most common genetic cause of adult onset dilated cardiomyopathy. And we know that based on previous studies, that about 15 to 20% of patients with dilated cardiomyopathy carry a rare genetic variant in titin. It plays not only an important role in dilated cardiomyopathy, but also cardiomyopathy caused by secondary etiology, such as pericardial cardiomyopathy, which is the pregnancy-induced, and alcohol-induced cardiomyopathy. And based on our study, now we know that this rare genetic variants in titin also leads to increased risk of cancer therapy cardiomyopathy. More importantly, though, when we took a closer look at our patient population and clinical characteristics, we realized that actually the presence of these titin genetic variants not only increases risk of developing cardiac toxicity itself, but actually led to poor clinical outcome. So again, if we focus on the red box at the bottom of this table, that patients who carry this titin truncating variant was at higher risk of having impaired cardiac function recovery because their LV ejection fraction was lower at the follow-up. They had a higher incidence of atrial fibrillation and also higher incidence of heart failure-related hospitalization. To confirm that actually titin variant indeed is an important role and it plays an essential role in cardiac toxicity, we also created a mouse model lacking actually this titin gene and then treated them with anthracycline, a sensor cardiac function. So in this graph, I'm showing you the fraction shortening, which is similar to the ejection fraction, the contractile function of the heart. And you can see compared to the blue line, which is the wild type, that our mutant mice have an impaired cardiac recovery. Again, consistent with what we observed in human data, that rare genetic variants and titin plays an important role in cardiac toxicity. So, in summary, I have shared with you today that common and rare genetic variants provide insights into mechanisms of cancer therapy-associated cardiac toxicity. Also, it helps us identify patients who are at higher risk of developing cardiac toxicity. Additionally, it can also be used to hopefully predict a clinical outcome in our patient population so that we can try to maximize the best outcome for our patient from both oncological and also cardiac perspective. And lastly, I'd like to thank my lab members and my mentors and colleagues. Thank you so much for your attention.
Okay, so this talk is a job on Yellow, and she's associate professor at Mr. Municipal, and she's handling his Cardiomyopathy to provide you get category 1.
Thank you for the introduction. This is the building where my lab is in. It's locally here at the University of Minnesota. My lab has done limited work on the role of cardiac toxicity. We've done some collaborative work with the Zordaki lab and some original work on the role of endothelial cells, CD-positive endothelial cells, as well as some radiation-induced fibrosis, where we had shown that small peptides that really inhibit that fibrosis and potentially treatment for radiation-induced cardiac dysfunction. But for today, I thought I would give a more of an overview of potential roles for doxorubicin cardiac toxicity. And this is just an overview of the number of publications published in the last number of years on doxorubicin cardiac toxicity. you can see that it really is on the rise, and I think this is indicative of a potential increased understanding of how doxorubicin could cause cardiac toxicity. But unfortunately, that has not yet resulted in an additional treatments for this disease. And to get to treatments, I think many of us use mouse models. And if you look at the number of papers that have used mouse models to try to understand doxorubicin cardiac toxicity, it's about a third of the total number of papers. And I think one reason to use mouse models, as already alluded earlier today, is that with mouse models you can potentially get to causality. Whether that causality is then also relevant for human disease is not always clear. So today I'll talk about various aspects that I think are important in the development of doxorubicin cardiac toxicity, and I'll quickly go through it so we can get to the questions and the lunch. I think it's well known by now that doxorubicin induces a dose-dependent level of cardiac dysfunction. If you use lower dosages, as presented to you earlier today, this can result in a delayed onset of cardiac dysfunction with or without additional stimuli. But to replicate this, people have used mouse models, and also in mouse models you can see a dose-dependent cardiac dysfunction that happens in mice. Well, why does that actually happen? And I think to understand this, it's important to first look at how doxorubicin actually works. Directly binds to the DNA, it intercalates into the DNA, and as a consequence, it directly inhibits the function of topoisomerase 2B, and this has been shown over a decade ago, I think, to be relevant for the development of doxorubicin-induced cardiac toxicity. So if you inhibit or delete the gene topoisomerase 2B, you can essentially block the development of cardiac dysfunction in response to doxorubicin. That's of course not the only mechanism. I think there are many more mechanisms, and one other mechanism that had been shown earlier today by Dr. Hossein and Ardehali is the increase of mitochondrial iron. He had shown beautifully, I think, that especially mitochondrial iron goes up in response to doxorubicin. This is in stark contrast to cytoplasmic iron, and this could be potentially due to the downregulation of the iron exporter ACH, which is localized to the mitochondria. So he then developed a genetic mouse model to test whether this is actually involved and overexpressed the transporter to eliminate much of that mitochondrial iron, and this was able to prevent much of the doxorubicin-induced cardiac toxicity. How does this actually stimulate cardiac toxicity? This is not entirely understood, but I think an important mechanism is the stimulation of hydroxyl radicals, as indicated before, which can eventually result in apoptosis, ferroptosis, and cardiac dysfunction. So this stimulation of ferroptosis, which is maybe a misnomer, but certainly induces cell death, could potentially be important in the development of doxorubicin-induced cardiac toxicity. And to test whether this was really important, an important regulator of this ferroptosis-induced cell death is the gene indicated here, and people have tested whether the inhibition of this GPX4 mediates the development of DOX-induced cardiac toxicity. And indeed, when you inhibit GPX4, you can get an aggravation of the level of cardiac dysfunction in response to doxorubicin. The contrary is also true. If you overexpress GPX4, you can get a protection. So if you inhibit the development of ferroptosis, you can
get an improvement of cardiac dysfunction. This has not yet resulted in clinical therapies, but it's one possibility I think for the near future. Another aspect of the cardiac toxicity is the development of fibrosis. There have been studies that have shown that low doses of doxorubicin that do not induce cell death, do not induce tunnel staining, are sufficient to induce fibrosis. And when you then stimulate cardiac fibroblasts with doxorubicin and look at which genes are altered, the important pathways that are found there are immune regulatory pathways. And that would suggest that potentially the interaction between the cardiac immune cells and the cardiac fibroblasts could be a target for doxorubicin-induced cardiomyopathy. And indeed, people have shown that macrophages are important in the development of dox-induced cardiac toxicity. These essentially come in two flavors, resident and recruited macrophages, and especially the resident macrophages go down dramatically in response to doxorubicin. This is indicated by the blue line on the left part of the slide, while the recruited macrophages go up. And the resident macrophages actually express scavenger receptors, SRA1 is one that was shown in this publication. That are important for recognitions of danger associated with molecular patterns, and this then in turn stimulates the proliferation of resident macrophages. And this was shown to be an important potential mechanism for how cardiac toxicity develops. And why? So when you enhance the ability of these resident macrophages to proliferate, they can restore some of that cardiac dysfunction. Another potential mechanism was published earlier this year, where the role of CD8 cytotoxic T cells have been identified as potentially important in the regulation of cardiac dysfunction, but also fibrosis development in the setting of doxorubicin-induced cardiomyopathy. This was verified also to be relevant probably in human and dogs that are treated for tumors. The next part of my talk will be about the cardiac atrophy that is often observed in response to doxorubicin, and I think this is a minor valued aspect of the cardiac toxicity, and I think that this could be really important. It's been seen in every study that has treated mice with doxorubicin that the heart rate actually goes down relative to the body weight. And this could be due to modification of the mTOR pathway, as has been shown, or of E3 ubiquitin ligases, such as MRF1. And if you indeed delete MRF1, you can prevent much of the dox-induced cytotoxicity, suggesting that this atrophy that happens in response to doxorubicin could be in part responsible for the cardiac dysfunction that happens. And this is not just relevant for mice, it's also seen in patients. If you do cardiac MRIs on patients that have undergone doxorubicin treatment, you do see a significant reduction in the cardiac size of these patients over the next 6 months. Finally, as a potential mechanism already alluded to, the induction of senescence. This could go hand in hand with the cardiac atrophy, because certainly aged people have smaller hearts than young people. And I think an important driver of this senescence could be the activation of P53 and downstream thereof the activation of P21. This has indeed been shown to be relevant and induced in response to doxorubicin in cells, as well as in mice. And when you try to inhibit senescence with senolytics, in mice you can actually treat the cardiac toxicity, and you can see a reduction in the level of P21 induction, suggesting that senescence may indeed be an important component of this cardiac toxicity.
So I think in overview, it's important to realize that toxin-induced cardiac toxicity is multifactorial, and I think the role of the mitochondria is probably central in the development of cardiac toxicity, and I hope that in the going few years we will be able to progress some of this basic mechanistic understanding in mice towards therapeutic approaches in humans. So thank you for your attention.
Thank you. So next talk is Dr. Joseph Minzer, and he's a professor and chair and modern integrated biology and physiology at University of Minnesota. And his talk is high-risk-based cardiomyopathy in cancer.
Thanks. We're about 12 minutes away from lunch, so thank you to everybody. Hats off to Ann and Sigmund for a really terrific conference. It's really been great. And please forgive me, I'm experiencing a little bit of jet lag this morning. I traveled all the way from St. Paul to get here, so let's see how we go. That's my joke for the day. So my roadmap for the talk today, we can see here is focused on the sleeve of cancer and heart. And it's really interesting to see the crosstalk going both ways. This is now being eliminated. I'd like to focus the bulk of my talk today on the cardiac myocyte heart intrinsic features that contribute into the underlying diagnosis to cardiac performance in cancer and cancer step. And I hope to have just a little bit of time to share with you an unpublished story that's emerging from our group on that future in terms of genetic models of cancer and cancer. Really, I think, interesting in terms of tracking what's happening in the complexities of human disease. So this is an overview slide that was just published recently, and it really highlights the theme of this entire conference, and that is the intersection between what's happening at the level of the tumor and how that has systemic effects, notably for this conference, that on the heart, and of course this arrow cannot be fashioned in both directions. So we literally have tens of millions of individuals that are cancer patients, tens of millions of survivors of childhood cancer, and the sad reality of these survivors is that, as we learn today, we know are at markedly higher risk for cardiovascular disease. And what I'd like to share with you in my short talk today is what is the nature of the cardiomyopathy that emerges in cancer patients and its survivors of childhood cancer. And Jaap just mentioned this issue of cardiac atrophy. The theme I'd like to convey to you is that it's the issue of cardiac wasting-based cardiomyopathy in cancer. Now, this is not necessarily a new concept. We can turn the clock back about 300 years as found, as you see on the left here, the treatise by Jean-Baptiste Senec, who published, I think, really the first description of what he referred to as the shrunken or small heart in human disease. Now we come to greater resolution, of course, today with the advances of technology, and it's really becoming more increasingly clear in the clinical enterprise, really in the cardio oncology area, is a relationship, as you see on the right-hand side, of the relationship of left ventricular heart mass and outcomes in terms of mortality and cardiomyopathy. I wish I had a pointer here. I do have a pointer, but it's going to be hard to operate in this menu. But I don't know if that's showing up on the board. It's not, so we're just going to have to forge it out. If you look at the upper right-hand column of Blaes, the orange column represents the left ventricular mass of cancer patients. We can see it down with respect to those of normal individuals that's shown in light blue. And really significantly, we go far to the right-hand side, the yellow graph, the yellow bars, are from the cancer cachexia patients. And there you saw the lowest level of left ventricular mass. So that represents true left heart cardiac wasting. And this group of Lena and Anchor were able to show the relationship on the lower right, the survivability of individuals that had low LV mass, and the prediction is very dire for these individuals where they succumb to death at a much higher frequency than those individuals that had relatively mild levels of cardiac muscle wasting. So the theme is cardiac muscle wasting and cardiomyopathy, and where do we go from here? So cardiac atrophy and contract-related deaths have been observed both in patients, and as I'll show you in animal models of cancer and cancer cachexia, but there's been a big gap of knowledge, and then it's very difficult to distinguish those of the systemic effects of what is actually happening at the level of the myocardium. And so we are very much interested in this on a basic scientist, which is heart and muscle. And here is Dr. Michelle Law. She was a postdoctoral fellow in my lab. She's an assistant professor at Baylor University in Texas. And what she did is she took the classic adenocarcinoma C26 tumor cell line, implanted it into the small of the back of an otherwise normal mouse. And over the course of about 20 days, you see the emergence with the massive solid tumor growth, the advancement of cancer cachexia, which, as you know, is the manifestation of a multifactorial disease state as manifest by involuntary body weight loss, principally due to loss of skeletal muscle mass. The challenge of the field that these are very short-term experiments, because the animal husbandry staff is calling you after about 20 days that these animals are on death's doorstep and need to be studied immediately. And that's what Michelle did. You can see on the tail end of that bar there on the downslope of survival. At about day 17 or 18, she was able to take these animals and remove the heart and study their functionality. So this is the University of Minnesota Murine Cath Lab, and you see the murine heart is about as big as your pinky fingernail. And Michelle was able to record the pumping action of this tiny little heart, and remarkably it produces quite a human-like level of systemic pressure and systole, so it's quite amazing. But this really revealed to us what is happening at the level of the whole heart. And you see in panel A, the significant diminution in peak systolic performance of the isolated heart that's going from the solid heart, which is the control, and the dotted line. This is from the cancer cachexia group. And that's summarized in panel B, left ventricular diastolic pressure significantly decreased. She also brought these mice to the University of Minnesota Murine Cardiac Stress Lab, and here she did a series of pacings of these isolated hearts that were being perfused with the Langendorf perfusion apparatus. And in panel A, you'll see the left ventricular developed pressure, as I just showed you, a diminution in that pressure, so reduced squeezing action of the heart. And very interesting to us in panel C, as the pacing frequency went up for these hearts, this is the pacing stress test, you can see the emergence of significant diastolic dysfunction as manifest by an increase in left ventricular diastolic and diastolic pressure. And as our clinical colleagues know, that portends very poor outcomes to both animals and humans to have that outcome. Michelle then drilled down further, going from the isolated whole heart to the single cell that makes up the beating heart chamber. This is a ventricular myocyte. And again, it's the same type of experiment, but this is at the level of the isolated cell. And here she sees the cell intrinsic deficits of heart performance that's shown in panel A. The solid line shows us what happens in a normal cardiac myocyte when it contracts and relaxes. And then in the dashed line, that's the outcome from the myocytes from the cancer animals. So then that is then summarized in panel C, which was a significant diminution in systolic performance at the level of the single cell. So this is a heart intrinsic cardiomyopathy. She wanted to go down further to look at the intracellular calcium transients in these single myocytes, long story short. And we go from panels A and C, which are summarized in C. We see a significant diminution in the peak calcium transient that's emanated by a stimulation of these cells. So systolic calcium transients are significantly altered and depressed in the myocytes from these animals. So the take-home points for this part of the talk are that with in cancer, there are intrinsic effects to the heart and the isolated mindset that lead to the cardiomyopathy that's now very well described in human patients. And I would say at the cellular level, these deficits are both at the level of the contractility and alterations in calcium signaling that collectively critically underlie the cachexia-induced cardiomyopathy that I think tracks what's happening in human patients. In the last just few minutes of my talk, I'd like to share with you a story that's being developed by Huda Cohen. I don't know if Huda Cohen, I saw her earlier with her wonderful little son Ishmael, but she led this study. And the challenge in the field of developing preclinical models of complex human disease, it's a little bit like a sledgehammer. I just showed you these C26 cells that are implanted into the small of the back of an animal, as we see here on the left-hand panel below. Those studies are about 1 month long because these animals are on death's doorstep. So Huda went on to engineer into these C26 cells a suicide gene program via thymidine kinase. And it worked out really extraordinarily well that when you induce the tumor cells, let the tumor grow, the animal develops into a cachectic condition. She intervenes at about day 17 with a prodrug ganciclovir, and that then targets destruction of those tumor cells. And we progress now to the right-hand side of this diagram. We see highly efficient tumor remission, and then effectively we have a post-cachectic state. Really extraordinary because she was able to then track this, and here we see on the very lower panel, we see the time course of the evolution of tumor development. She could track this because she also entered in the biomarker luciferase into these heart cells, we can track its growth and then its remission. Long story short, this study is being written up. Hopefully we could be publishing this very soon. Huda Cohen was able to develop a chronic cardiac wasting-based cardiomyopathy and heart failure animal model that emerges post-tumor and post-cancer cachexia. What's really remarkable to me about this model, and the folks that are basic scientists in the audience know that we try to study complex human diseases, it becomes very difficult for us to do that using animal studies. It's quite remarkable in that study how closely this animal model follows what happens in humans. It's really remarkable. Such that we have significant cardiac wasting initially, and then we go through the tumor remission phase. The skeletal muscle recovers, the spleen renormalizes, the fat renormalizes, all the other systems that are affected in cancer renormalize. What's left? A chronic, lasting, cardiac wasting cardiomyopathies. And Huda Cohen went on to then just follow these animals long past the tumor is gone. And again, we're not using cardiotoxic drugs, so this is particularly revealing in that context. And she was then able to show after about a year following the tumor, the emergence of a congestive heart failure. And this is tracking, as the clinicians know, what is happening in the survivors of childhood cancer, including those that have not received cardiotoxic therapies. So particularly exciting to us to use this on the system because I think it's going to be a key one for the field to move forward. So I'm going to end there. I passed my time. This is our lab. Rich, happy to be there, and happy to take any questions that you may have. And there's me with Goldie. He's a little bit sad today. He took it on the chin yesterday, but he'll be well. Thank you very much.
Okay, this is just double conclusions. So, great talk by everyone. Congratulations. I just have a question about cardiac wasting with aging. Based on what I know, autopsy studies have been done, and if anything, our hearts actually get bigger as we get older because we get hypertrophy of the heart. So, is it true? Apparently in mice there's cardiac wasting as they age, or is it as it is shown in humans?
I don't know who that one's directed, but if we're talking about cancer and survivors of childhood cancer, it's not hypertrophy, it's wasting. Aging is a different story in the absence of cardiovascular disease. I don't know if we have experts here that can address that. But I think one of your earlier slides just said that as we age, that their heart gets smaller, the left ventricle. That's what I thought you said. Yeah, I mean, we turn back the clock to Sennett's observations nearly 300 years ago. He described for the first time a shrunken heart, and he associated that to the disease of the individual. He did not diagnose the individual at the time, but that wasn't an aging scenario, that was a disease scenario. Yeah, thank you.
If this no question about the extra model, and there's this trial that was just published in English Journal and targeting GCOS. So I was wondering if you have no GCOS and differentiating just global captions from the cardiac phenotype reverse.
Yeah, that's a great question, and I think it really speaks to the on nature of cancer cachexia. We did look at GDF15. There were effects early in the acute phase. We're not really seeing them in the chronic phase. What we are seeing, again, all this amongst purpose here, is the massive increase in systemic IL-6. Not mindful, the tumor is long enough. So where's this IL-6 coming from? We think it's coming from the heart. And we have some pathways that are articulating, and we hope to get on this bigger searching. That's a great question.
Okay, so I have one last question. So there's outside Immunotherapy division report that does that can use look-see can be reversible completely or partially 32 OMICs and patients. In that case, do you have any idea about this figures see or hydroxy and the genetic model genetic populations?
Well, I think that's a very good question. Thank you for answering that. I don't think we have enough number of patients that we study because I don't think genetics have enough focus in cancer therapy to cardiomyopathy yet. In general, I think genetic cardiomyopathy, particularly type of cardiomyopathy, tend to be more useful than other genetic cardiomyopathy such as tuberculosis dynamic. So our hope is that with the therapy, that is supposed to be more prone to develop cardiac function, like there's really good studies to demonstrate by it.
Last question.
It's actually more kind of it's time for lunch. So I just want to let everyone know that lunch is set up outside. If you are right now, all our speakers this morning. If any of you are interested in going to dinners tonight to talk with some of your colleagues that you've been having a great discussion with, you'll find outside the door is to sign up for some various restaurants in town. Some reservations to join your colleagues. And meanwhile, let's see. Thank you to all of our speakers.
Hey everybody, welcome back. Let's get started. We're 5 minutes after schedule already, and I hope everyone had a great lunch break. We have a really exciting session lined up to help you with your postprandial tip. So we'll talk about immunotherapy and accelerated CAD. And the first speaker is Dr. Eric Yang from UCLA, who will talk about immunotherapy and next topic.
As far as
Thank you, Dr. Von. I also want to thank the organizers really for this wonderful opportunity to see everyone at the university, stateside Rhee. So it's really truly a privilege to be up there, and so glad this is right after lunch. So you fall asleep and you found it. Anyways, I think I'm preaching to the choir here. I think it's not exaggeration to say that cardiovascular sleep associated with chemotherapy, probably one of the most fascinating things that have our attention right now. Obviously, game changing, not only for their effects to cancer remission, but obviously the rare toxicity for myocarditis. So, like Dr. Zhu, I'm really excited to hear their work on, but also there now seems to be increasing signal, both at basic science level and clinical judgment that this may not be the only cardiotoxicity to be aware of. And suspects like intentional occurring fast cardiovascular disease like atherosclerosis is something to pay attention to. And there's more really big population question why that is. So, again, as a clinician, on the bedside side of the spectrum of this disease, we still obviously end product of these rare cases of myocarditis, but also I would say looking here, imaging of cancer patients. We're seeing them for, also just what we call heart health care. We're starting to see signals also that cardiovascular being appears being more apparent, whether that's a result of survivorship, suboptimal cardiovascular risk factor modification, or actual effects of the immune checkpoint inhibitors. We still don't really know the answers. Again, for many of the researchers, it's really work is really critical helping us figure this out as to what is causing what. Really, understand what we checkpoint inhibitor. So, again, talking about the rises down there, again, like I said, I think this is something that cardiology can really play drag into us. We are going to see plenty in sustained myocarditis. Can't say that for one more, but should see and the numbers aren't even quite as important because over the next decade, we're going to have over 20 million less energy virus wide. And this is just the United States above. And then there's an energy on the conference. So, and if you look closely at the different kinds of cancer subtypes, already a majority of, especially solid, the relative place for ICI American versus second line, as we're an FTA in the J. Even then, this paper thing about prosthetic economics also showed in 2018 or 20 for therapy, perfectly mild as why you're not H without getting in the middle of that. So it's then it changed the face how we treat cancer. Also, not maybe got a ton of things fine if you're long close for it. This is brought back our only respond integration and most of us as a field in the awareness. And another important thing to also realize is that by next year, probably estimated about 700,000 patient metastatic should be alive. And like I said, immunotherapy brought in is taking the answer or not. But then the question, what if the right master affects the arm or purpose? The recent scientific statement shows that one of the things that are very complex are immune reaction unprecedented fires, IRFEs, where certainly get myocarditis, and much more sensationalistic, very presenting station. The bigger question is, what is the effect of this T cell activating effects on, atherosclerosis? So going to really, core, we got to talk about what this talk is. This overall, I would like to say, mainstream, mechanistic diagram is designed for people like me who can largely understand really what how immunotherapy works. Okay. This initial originally was a really description of immune checkpoint areas that you can read this thing, you study. Really, it's very important to understand basically two concepts: inhibition, co-stimulatory. Right. And the reason why these are called breaks, because your immune system, essentially, in order to prevent your T cells from having the rest of your system, is finding these two signal accurate checkpoints. Usually, like you like kind of one, a CTLA-4. And also, usually these drugs, when given, usually block these signals and essentially de-repress the signals that lead to initial T cells, whether it's drip cell level or at the T cell tumor level. Essentially automating the T cell process, causing recognition of the T cells, or the tumor cells or invaders, and activating the other mechanisms. However, we do know that there's a certain much more complex to that. And really, this paradigm showing the microcephalation is not just at those levels, but probably at a variety of different other cell types and developing the immune system. And also now, and this is probably also involved in cancer battle, is also trying to variety of bodies as not only
run like co-inhibitory mechanisms, and also the tumor necrosis factors to good balance, which are dominant in system tumor. But sometimes it can also exhibit immunomodulatory co-inhibitory like function as well too. So I think a lot of cardiologists in this room are getting trained to understand the right side of this diagram. It's mainly focused on lipid or mechanisms of atherosclerosis. Now, the checkpoint inhibitor therapies are brought back to circulate the adherence to pattern inflammation and treats also in cascading atherosclerosis and mention other events that acute coronary syndrome and other thrombotic events. You can see here too, from these T-cell and mechanisms that's been postulated that upregulation of the T helper cells also leads to proliferation of monocyte recruitment and their damage, and also other mechanisms that also increase cytokine lesion also can lead along with the oxidation, the formation or a single plaque in chronic course of being increased atherosclerosis. And this review by Dr. Jeff White was about Cedars Sinai Development, its research program at our institution, also shows that the variety of these science and studies in which don't actually get into all, that there has been evidence or links to increased degrees of atherosclerosis and unstable plaque course, certain kinds of CGM blocking, field blocking mechanisms. And also, it's thought that also CD28 CTLA-4 pathway, it's thought that the application as being activated because there's reticulation inflammation. But at the same time, this is why these wide effects are starting to manifest also. And this is just, again, one of the studies that demonstrate, again, I guess, the gaps in human and animal knowledge and models, as well as our understanding. And again, this is just one example or two of how the differences that occur when they study standalone dual agent isociatic therapy in humans to stage 4 melanoma. 6 weeks, they had no increase in PET inflammation, but in the LDL or knockout mice, it did seem to see an increase in change in the lymphoid infiltration of plaque in recent chronic cohort. And again, how much of this is model-based versus human-based, something that goes to the level. Yet, when you actually on a bigger scale, looking at Tom Newland's data with GH, we looked at population of 2,800 cancers compared to receiving checkpoint versus those who did not match by gender as well as cancer type and mediated cardiovascular events, whereas it's statistically significant higher rates of cardiovascularization and my shell or cardiovascular events. The same key cardiovascular series as well, looking at the 2 year after, that's a 2 year before treatment, you're going to also see two significant differences in mixed outcomes. They continue to say it's also living calcium score, I guess, cells. They looked at the rate of the plaque accumulation. It's also significant in the iPSC. The small study, but I don't think that is the team to go along and they're still, on the patients looking in candidates and others, was TT scans looking at plaque progression and non-melanoma. And interestingly, they found using antibodies changes in uric acid scores, calcium volume, calcium mass. Interesting, the mass did not change, but it growth rate of calcium volume in uric plaques reduced in the chat on there. And there it was that plaques and even kind of flame its calcium limits by these positive like macrophages after isonites will either that might need to relative fields to non-calcified ice anymore or calcified plaque and hormone document for the sake. Again, in a lot of my pods, in data, probably you can study this on shadow. And also there are some agents too that also interesting measure signs of anti right? And this interesting observational study that we do looking initially at the C47 blockade humanized antibody panel. Revealed that this way non-Hodgkin lymphoma patients interestingly saw a decrease in FACC uptake auto arterial plaque as well. While this was very interesting, my understanding of the drugs just continued because of the increased mortality in the pain all about which better. Then you see there also how it was true. And really one of the slides I actually saw qualify comment on is what can I do when we now or we many more patients and we have to check here, videos will have the CCD, is that to use these data sets already that we have to us to look at the evidence of ASCVD, to see who needs the rest of the therapies to lower cardiovascular risk as ASN to from this stratification, we not be the one that's improved life treatment. And in order to get this imaging in front of the main cancers monitoring the races. So in summary, we wrote this editorial and to figure out what this effect while looking at myocarditis is obviously a very highly fatal, very important disease to look at. You may be missing a bigger vulnerable population, and we don't want to miss or trace of nations that might be more vulnerable downstream cardiovascular disease. And being just fine, allow research and collaborating levels to all levels to investigate manifest text and treat and irritating upon historical population. Interestingly, in this review article was also interested in looking at how immunotherapy-related therapeutics can also give insight into how we can treat inflammatory and accelerated atherosclerosis and reduce, drugs or therapeutics that may reduce inflammation, like hypnotic stimulation or enhance inflammation. And again, some of you also having suggested cardiovascular involved because of the role of our club. So in the conclusion, ICI therapy is in class to immunotherapy that again, one of the stunts gas population qualified for it will be around like machines. And ICI sharp is more dramatic in presentation, but our preceding pregnant evidence downstream also due to this, or unlike Chelsea risk factors and increased survivorship, not like the same question of constantly. And basic science and level of planning is a proactive guys, but also some areas think of treating happens first. And then again, we really have a lot of work ahead of us. We also have blood-based image technology that is supposed to potentially harness and leverage and study the historical population that's already actor. I'd like to certainly say that we have also have some data coming out that we're excited to then led by Dr. Hudgens, we're working state scientist fellows already not on BHA. Unfortunately, something at BHA will basically give us launch and hear about that. It's my numbers. So thank you again to our fellows as well as our team taking care of these patients and for the rest of the doctors. Take your time. Thank you, Dr. Yang, for a fascinating talk. We'll move right along and do the questions again at the very end. Next speaker is Dr. Margaret Axelrod, who will be from Vanderbilt to Washington. And she will give a talk about patient and tumor-specific biomarkers.
All right, thank you for the opportunity to speak here today. I'm going to talk to you about one of the deadliest side effects of anti-cancer therapy, as Dr. Yang mentioned, immune checkpoint inhibitor-associated myocarditis. So building on what Dr. Yang talked about, immune checkpoint inhibitors, or ICIs, essentially work by reactivating CD8 cytotoxic T cells and releasing those brakes on immunity. Hopefully, these ICIs reactivate T cells specific for cancer antigens that cause cancer cell death. However, patients treated with ICIs can experience autoimmune-like side effects in just about any organ of the body, including the heart. As we discussed, ICI myocarditis is an extremely rare toxicity, occurring in less than 1% of patients treated with combination ICI, but it is a highly fatal toxicity with mortality rates approaching 50% in currently published studies. There are no clinically actionable biomarkers currently to predict who's at risk for this rare but extremely serious toxicity. So as has also been discussed, we use mouse models to learn more about the diseases that affect our human nations. The Allison, Mosley, and Malpa labs developed and characterized this mouse model ICI myocarditis and raised mice with a genetic knockout of the gene encoding CD1 and heterozygous loss of the gene encoding CTLA4 developed pulmonary myocarditis with about 50% mortality. Myocarditis in these mice shows very strikingly similar histologic features to humans with severe ICI myocarditis, including similar immune cell composition, and these mice even develop similar arrhythmias to what we see out in human patients. We use single-cell RNA and TCR sequencing to learn more about the immune infiltrate in the hearts of these mice. About a third of the immune infiltrate is composed of activated CD8 T cells. These T cells also are very clonally expanded. And what that means is that these T cells have engaged their T cell recombinant antigen and doing more copies of themselves with the same T cell receptor. And these clonally expanded T cells are CD8 cytotoxic and very active. So establishing that these are abundant in myocarditis, next we establish that they're actually required. Depletion of CD8 but not CD4 arrested survival in our mouse model, whereas transfer of full immune cells but not immune cells lacking CD8 was sufficient to reduce disease. Fascinatingly to me, in this model of transfer of immune cells, expansion of a single T cell receptor clone was sufficient to drive myocarditis in this mouse model. So establishing that CD8 cells are there and that are necessary for disease, we're next wanting to know what they can recognize. So we took 3 of our mouse TCRs and informed them to do an antigen discovery experiments. We were particularly inspired by other work that could show an alpha-myosin as a potential autoimmune target in non-Rhee checkpoint inhibitor-related other forms of autoimmune myocarditis. And this earlier work had postulated that alpha-myosin was an autoantigen target because of its lack of expression in the thymus. And this lack of finding expression would be predicted to allow autoreactive T cells to escape from negative selection, and therefore these T cells might perhaps be reliant on peripheral tolerance mechanisms such as immune checkpoints. So on the whole, now in mouse model, we found that our myocarditis-specific TCRs were indeed specific for alpha-myosin. Extending this work into humans, we found in 3 ICI myocarditis patients, the TCRs present in our hearts overlapped with TCRs expanded in the presence of alpha-myosin, suggesting an enrichment for alpha-myosin-specific T cells in the hearts of patients with myocarditis. We were able to take one of these TCRs and clonate and identify a specific alpha-myosin peptide and HLA pair. This identification of an antigen HLA pair enabled us to use tetramers so we could track where these T cells are using a tetramer-specific staining. In patients with this HLA-A0301 HLA type, we were surprised to find that even in the blood of healthy donors, as well as in 2 alpha-myosin ICI myocarditis patients, we were able to detect alpha-myosin-specific T cells in the blood. So in summary, these data lend itself to a model of ICI myocarditis pathogenesis in which alpha-myosin-specific T cells escape from negative selection in the thymus. They can then be apparently reactivated in the periphery by immune checkpoints, immune checkpoint inhibitors, and traffic to the heart by the corresponding disease. So this model of ICI myocarditis pathogenesis lends itself to the development or really areas that we might focus on for future biomarker or risk factor development. So one of the first I'm going to talk about is thymic alterations. There's a really nice paper from Joël group in France. And first they found that actually patients with thymoma or thymic carcinoma had a much higher risk of ICI myocarditis compared to patients with other cancers. What I thought was really fascinating about this study is that they also were able to look at the thymic density on CT. So pretty much all of the patients treated with ICI had a staging CT at center line. And so they could look before therapy, patients with ICI myocarditis actually had different thymic area and fat density compared to patients treated with ICIs who didn't develop myocarditis. Somewhat unclear exactly how this different thymic density or chest CT might relate to what I'm talking about in terms of escape or negative selection, but it's a really interesting possible biomarker given the prevalence of chest CTs in this patient population. And the second possible area for biomarker development would be where these T cells are getting exposed to alpha-myosin to get activated. One possible area is through aberrant tumor expression. So we all know tumors can do whatever they want. They can express RNA-specific proteins if they want to. So this is an RNA sequencing data set on melanoma tumors. When we found out about a third of the melanoma tumors had detectable levels of alpha-myosin expression. However, all of these patients were treated with immune checkpoint inhibitors, and none developed clinically significant ICI myocarditis. Possible that they could develop some low levels that were not detected at the time, but again, that's not the plan in disease we're interested in. So these data suggest that at least at this relatively low level of alpha-myosin expression by the tumor is not sufficient to be a risk factor for ICI myocarditis. Another really intriguing possible area for biomarker development or exposure to apomycin is from this science paper in non-ICI-related myocarditis, that they found that a specific peptide made by commensal lipo was structurally similar enough to apomycin that it induced apomycin-specific immune responses in patients with a particular MHC2 allele that had really strong binding to both this commensal peptide as well as apomycin. So, to my knowledge, these types of studies have not been done yet in ICI-treated patients, but these data suggest that molecular memories for things like the microbiome might be a potential area of risk factor. Another potential area of exposure is for pre-existing cardiac damage. However, in early studies of ICI myocarditis patients, there was no association between patients who were already on some kind of cardiovascular medication at increased risk of myocarditis. So suggesting that patients that who are clinical high or naive in terms of their cardiac history are still at risk for this toxicity. Another potential area of biomarker or really risk factor development would be to look at patients' HLA types, with the hypothesis being that patients with HLAs that have a higher affinity for apomycin might be at increased risk. Again, studies are still ongoing on this and are limited by low numbers of patients. I think a particularly intriguing area for biomarker development is through looking at these circulating effector cells. A study that I would really like to do would be to use the catchmers that I spoke about earlier in order to track apomycin-specific T cells as an blood or pre-treatment through the course of toxicity. But again, these studies are limited by lack of pre-treatment and paired samples. Dr. June, who's going to speak next, has really intriguing work on T effector memory cells in their circulation at the time of toxicity, as well as on CXCR3 specific cells. For work as well as work by others, have also been interested in implicating lower T cell receptor clonality in the blood as a biomarker of ongoing ICI myocarditis. However, again, there's really a big need for more clinical samples to enable this research to be more fruitful in terms of looking at pre-treatment samples to identify those patients who are at risk. Another really intriguing area would be looking at antibodies. However, preliminary data found in the Balco lab suggests there's no robust signal, at least as of yet. However, those data are really immature at this point in time, and more work to be done in that. Some other really intriguing areas would be other patient factors like sex differences, as well as also genetics, as we've discussed them on the previous sessions. I've also now presented to you a very T-cell centric view of how apomycin or how ICI myocarditis works. However, there's some really intriguing data by Dr. June, as well as Corlevine's group at WashU, looking at T-cell macrophage clot stop, and some of the cytokines involved in that might be a potential biomarker. So in summary, I've tried to show you what my data suggests to the model for the ICI myocarditis pathogenesis, and how we might use that understanding of pathogenesis to tailor some future biomarker development. I think this is a really young and exciting field with a lot of work still to come. I'd like to acknowledge my mentors, Dr. Justin Balco at Vanderbilt, as well as Dr. Gavin Musley at UCSF. And I'm grateful to all of the people on this slide and all of you all who are working in this field to help make care for our patients.
Thank you, Dr. Extra. And for the next talk, I've actually invited Dr. Maedeh there from Stanford, and she will keep with the theme and talk about an effort novel therapeutics and biomarkers in immune myocarditis.
Thanks. It's my pleasure to give this talk today, and really great talks I've been receiving. And this slide here. Yeah, one button. There we go. These are my funding sources. Okay. So as already discussed, ICIs are monoclonal antibodies that activate T cells against cancer cells, and they're increasingly used in cancer patients. There are a number of checkpoints on T cells that can be targeted by ICIs, most notably CTLA-4 and PD-1, but there's also newer checkpoints like LAG3 And Dr. Jimenez here now is also had some really interesting work on working.
Yeah.
As you heard from Dr. Silveron, ICI myocarditis remains a fulminant toxicity, and its unpredictable nature makes finding effective therapies for it an urgent problem for patients receiving immunotherapy. So for patients who are diagnosed with ICI myocarditis, glucocorticoids remain the first line of therapy despite having limited efficacy and side effects. So the ultimate goal is to get these patients on targeted therapies, which can be more effective and precise in targeting the mechanism of disease. Additionally, a lot of patients who get diagnosed with ICI myocarditis, they also get their ICI stopped, which is not great from a tumor control standpoint. So these are the challenges in the field right now. Abatacept was the first hope towards targeted therapy. Dr. Mozneb and James Allison's teams were able to show that in the CTLA-4 het PD-1 knockout mice, that abatacept, which is a CTLA-4 modulator, was able to improve survival in these mice, as well as reduce immune cell infiltration into the heart. However, we're just beginning to scratch the surface of understanding the mechanism of ICI myocarditis. So our group took an unbiased approach to this problem by performing single-cell RNA-seq, TCR sequencing, site-seq, and site-off in paired mice who had a PD-1 knockout and a slightly autoimmune MRL background, as well as patients who had been treated with ICI with and without myocarditis. And what we saw was that there was an expansion of circulating effector memory T cells in both the mice and the patients with ICI myocarditis. And we also saw innate cells in the hearts of the mice. Interestingly, cells were clonally expanded and also seemed to persist in many cases despite a steroid treatment. And we felt that this served as a potential biomarker and therapeutic target in the disease. So since that time, there's been a couple of landmark studies in the field. So Dr. Axelrod, we just heard from, was able to show that some of these pathogenic antigen-specific CD8 T cells were recognized in albumin's IPSC. And Wang and Shikokoma et al. Were also able to show this in a different mouse model. And then Panama and Chun and Jesus Jimenez from Corey Levine's group showed that some of these effector CD8 T cells were secreting interferon gamma, and then they were actually recruiting potentially T cells in the heart. And if you actually did interferon gamma blockade, you could improve survival of these mice. So really intriguing data. Our group has been able to take this approach where we start with a population of patients and also pair it with the mouse model, and then collect samples from these patients and perform this unbiased single-cell multi-omics to look for disease targets, with the goal of eventually mechanistic validation and bringing it back to the patient in terms of therapeutic translation. And so with that, I'd like to share a really exciting story from my lab. This was a work done by Vivian Huang, who's now an MD student at Cornell, and Yin Sun, who's a current postdoc. And so really to start, we try to get a global picture at the origin of ICI myocarditis by taking our PD-1 knockout mice and going back and doing serial time point analysis single-cell multi-omics at each time point with paired heart and blood samples. And what we saw was that at 2 weeks, prior to any emergence of clinical onset of myocarditis, we saw these CXCR3 expressing T cells, CD8 T cells, come into the heart. And these T cells were expressing high levels of cytotoxicity markers, transamin and ferrin, as well as being clonally expanded. And interestingly, we also saw at the same time emergence of CCR2, CXCL9, and 10 positive macrophages come into the heart as well. And these macrophages were also expressing high levels of interferon gamma response elements. And so to really try and understand what was the chicken and what was the egg, we performed a macrophage depletion experiment where we used liposomal clonidine to deplete phagocytic cells in our genetic knockout mice. And what we found was that with the macrophage depletion, we actually saw a significant improvement of survival in these mice, as well as decreased immune cell infiltrates and increased cardiomyocyte death in the hearts of the mice treated with that macrophage depletion compared to those that were treated with controlled liposome. And also the CXCR3 expressing effector CD8 T cells were also reduced in the heart. And this is then to some extent also similar to data that was found by Dr. Levine's group using a different method of macrophage depletion. So at this point, I want to take a step back. So what are the druggable targets in ICI myocarditis? So we think that there's some sort of primordial event of T cell priming of cardiac antigens that are presented to T cells by antigen-presenting cells, possibly in secondary lymphoid organs. And of course, some of these cells could have this H kinase selection, as Dr. Axelrod highlighted. Upon administration of the immune checkpoint inhibitors, peripheral tolerance is released and these T cells are activated. In the form of activation, they are expressing also pro-inflammatory cytokines, including interferon gamma, that then signals to interferon gamma responsive macrophages, including CCR2 positive monocytes in the circulation. And we saw this too in our study in 2022, where we did paired heart and blood single cell multiomics. And these macrophages are then recruited to the heart. Upon reaching the heart, they are then secreting chemokines CXCL9 and 10 that then signal to CXCR3 positive CD8 T cells that then get recruited to the heart. We know these CXCR3 positive T cells are clonally expanded and very cytotoxic. And last thing, I think there's a chronic inflammation phase where CCL5 plays a role. So in terms of thinking about possible drug targets in the disease, there's an opportunity at the antigen level in checking antigen-specific therapies. There's an opportunity at the co-stimulation, co-inhibitory blockade level with abatacept and other similar drugs. And of course, there's the interferon gamma element and the JAK-STAT inhibitors, which have actually shown that there's been some exciting clinical data coming out of the June-Wha Salem's group looking at ruxolitinib and treatment of ICI myocarditis. Where we decided to focus on is the CXCL9 and 10 interaction with CXCR3. And just to double check that CXCR3 is a translationally relevant target, we went ahead and looked in the hearts of our patients with ICI myocarditis, and we found that they were indeed expressing high levels of CXCR3 positive CD8 T cells, as well as CXCL9 and 10 positive macrophages. And so to study this, we used what we felt was a more translational relevant mouse model by treating MRL mice with dual immunotherapy with anti-PD-1, anti-CTLA-4. And so we actually induced myocarditis first, and then we were able to show that treatment with anti-CXCL9 and 10 compared to control was actually able to reduce troponin levels in these mice, reduce immune infiltration, cardiomyocyte death, which is not shown here, and then improve survival. And so we found that this is an exciting drug target for ICI myocarditis. And to try to understand how the CXCR3 blockade reduces myocarditis, we turned to a transwall migration assay system by putting CXCR3 positive T cells in the top well with the porous membrane at the bottom, and then CCR2 positive, CXCL9 and positive macrophages in the bottom well, with the idea that if these macrophages are indeed recruiting T cells, that they can see increased migration across the membrane of these T cells. And indeed, what we saw was that in the presence of macrophages, we see increased migration of these T cells down to the bottom well, which is inhibited by blocking CXCR3, as well as blocking CXCL9 and 10, both individually and in combination. And last but not least, if you remove macrophages like the bottom well and just a pure chemokine, CXCL9 and 10, you also see increased migration of these T cells. And of course, the elephant in the room is the effect of blocking CXCR3 on tumor control. And so this is an exciting project that's spearheaded by me. And so what we did was we injected mice with B16F1 melanoma tumor cells, induced tumors in these mice, and then showed that with ICI, of course, you see reduction in the tumor size, but also with concurrent treatment with anti-CXCR3 and anti-CXCL9 and 10, that you don't have a decreased control on the tumor size. So this is exciting preliminary data that we're excited to get into. And I just want to give a nod to this paper that is coming out, hopefully any day now, from Tom Nealon and Kerry Reynolds and Thori Milani's group, where they show paired heart, tumor, and blood single-cell multi-omics from patients with ICI myocarditis. And interestingly, and I'd have to get a little bit more into the types of tumor. I don't know if Imova was in there, but they actually didn't see any shared antigen-specific T cells between the heart and the tumor, suggesting at least in some tumors, there are differences in the T cell regulators that invade tumors versus those that invade the heart. And so in conclusion, we're excited to explore chemokine receptor blockers as a new therapy kind of in the armamentarium of therapies for ICI myocarditis. And the one nice advantage of these therapies is that they are fast on and fast off, and they have a potential to make tumor killing medley. So with our ultimate vision that we can get away from glucocorticoids and focus more on targeted therapies, and also that our targeted therapies will have a negative effect on patients who are trying to get their tumors controlled by immunotherapy. And lastly, just to touch on biomarkers in ICI, our group did pioneer the use of routine troponin screening in patients who are getting ICIs. And Sarah Waliani, who's now an onc fellow at Dana-Farber, really spearheaded the first 214 patients where we were able to find subclinical cases of ICI myocarditis, with troponin elevation up to 10,000 nanograms per liter with this screening protocol. And now Evelyn Chun, who's a cardiology fellow in my lab on her F32, and Maya Wanabe are spearheading the next analysis on this cohort of 428 patients, alongside a similar cohort of unmonitored patients to look at patient outcomes. And the hope with biomarkers in ICI myocarditis is that troponin and perhaps some novel immune biomarkers can help us distinguish between patients who are high risk who will go on to develop fulminant myocarditis compared to patients who are low risk and who hopefully will have the opportunity to continue their immunotherapy uninterrupted. And so with that, I'd like to thank everybody in my lab who's done the actual work, and as well as my mentor at Stanford. And my husband and my two sons, Rhee is on the left here, is trying out the stethoscope, completely got prompted I was sitting and enjoyed it. And then T, who's 4 months old. With that, I'd like to thank everybody and take questions.
Thank you. We have 5 more questions coming. It's a great talk with everybody. Really enjoyed it. Thank you so much. It's possible that, in a couple of decades, maybe a couple of years from now, ICI therapy, even CAR-T therapy is being obsolete. And the future of cancer therapy is probably going to be cancer vaccine, where every patient gets their cancer sequence and the neoantigens were identified. And then there is those neoantigens are injected into the patients. That's when people think that there is the future of therapy for these patients. And there are very promising data that are coming out. Is it anybody actually looking at those neoantigens for those that have already been published to see any of that can cause cardiotoxicity? And that's probably going to be the future of cardio oncology, to identify the neoantigens that can also cross-react with the heart.
It's a very interesting question, and I think I'll stick on an answer because I think our panelists are not as working actively in cancer vaccine.
Right.
I think it's a really intriguing idea, and it also, like I showed with the study of the microbes, there's some really interesting possibilities for mimicry across topics that might mix those immune responses that are now fully sort of anticipated in advance. Definitely one of the struggles of targeting cancers at all is that they're derived from us. So there's always the possibility of some of those off-target effects, and I think that's obviously why we see some of those off-target effects in immune checkpoint inhibitors. So I don't think this field will go away with targeted vaccines. I think there's truly a lot, especially if, like you said, every patient gets their own vaccine. There's third patient has another possibility for unique side effects that we never been seen before. So that will only increase to me.
If somebody gets
cancer vaccine, I don't see how any additional medication would help them. Those should actually be very effective. That's why there is so much excitement with cancer vaccines. So,
It's got in the middle.
so about next talk by both speakers. So the question I have is about the immune IRAs basically happen simultaneously. Cardiovascular being reversed and several members actually certain iPSCs coexist. So and you do sort of detailed sequencing cytostic SCR and SC. Would you check other organs to see if there's a cross which helps you identify the problem from sort of relatively simpler labs and getting tissue by heart?
Yeah, that's a great question, and one we thought about as well. So when we did the single cell multi-omic study with the myositis patients, we also did look at what we call B patients, so patients with other itises, including thyroiditis, the skin itises, etc., hepatitis, and essentially, we mostly focused on the blood signatures, but even there, we did see clonal T cell expansion as well, and some of these other itises. The one distinguishing factor that was pretty remarkable about the ICI myocarditis patients was just the degree of clonal expansion and the degree of expansion of these T cells. And we didn't see, for what it's worth, any common TCRs shared between the itises with the thyroiditis versus the heart, but that was in our patient population. And we also did not look in the organs themselves, which I think is a key critical part of this. So to answer your question, I think that work is only important and it still remains to be done.
And in animal second, you could not to do that in animals because it would be similar all across the board.
Yeah, absolutely. We just haven't looked, but yeah, it's also important to take a look.
Yeah, you bring up a really good point too about IRAs basically being a cardiovascular risk factor, GCOS, right? Because we don't know if the presence of any other organ involvement could also lead to downstream or represent what are called pro-atherogenic effects anywhere in the body. And in our group also, we have a big data pool of about 6,000 patients, patients who see your check. Like, this is very early work. We're hoping to explore some hypothesis that toxicity in other organs could lead to further, or might be associated with further downstream cardiovascular outbreaks. I think that is an opportunity for any major cancer center to also leverage their data to look at that. And I have a question about the myeloma data. I was intrigued why you selected that particular group of patients. So what I was really understanding, how do you know that it truly was that particular phenotype, or what's called Kung syndrome? Kung syndrome is an immune deficiency that occurs with myeloma, probably be in T cell. So how do you distinguish Kung syndrome from ICI myocarditis?
Thank you for the question. I'm not sure if they particularly looked for a good syndrome, but those patients, for the most part, in their cohort had a biopsy for being myocarditis, and level clinically significant.
Yeah, it's interesting because in our pathogenations, before ICI we diagnosed myocarditis, specifically in patients with myeloma. So it can be very difficult to distinguish the two. Question, very interesting, doctors, so as it works. I had a question about preclinical models of human disease and these strengths and limitations thereof. The variant of hardcore has almost complete exclusive expression of the alkalized heavy chain, whereas even when our tests reverse that dominated by the large parts of the amylase heavy chain, a very small amount of alpha. So the more that you see to the challenges that these kind of mirrored monodomes dominated by alpha-amylase heavy chain, perhaps over on a stagnant deck, if you will, I'm sure that's potential energy.
Yeah, there's always limitations, in terms of using our mouse model. We were intrigued though about how striking the similarity that its use was between mice and humans, and in terms of we observed these T-cell ventricle in both mice and humans, as well as the TDRs you recognize in the human disease, where it overlapped with how mice and expanded T-cell was from the ventricle. in human disease as well. So we did one criticism about it is specifically low use, even there was actually some outcasts in Ardehali by Kamdar, that, I overlooked or at a low level is possibly sufficient to drive this disease. I think that there's likely to be other agents involved as well, especially in human disease. It's more complicated than just autophysin, but it's possible that this might be an initiating factor given sort of autoimmune firmness of some of these T cells. But it's a really good question, and I'm really finding out. Always keep in your mind the limitations where we right now.
One potential going more honest. One may not be perfect, but because that one is highly flat moment sensitive, you can argue the immune system hypothyroid and completely switch it to the heart. Without human heart proportionality, then that would give you a clear purpose of fiction.
Last question, very great, quick answer.
And I'm actually a cardiologist. Questions for Dr. Yang and Dr. Zhu. But since we know the broad heterogenic signal of IPSCs, do you think getting a CTA value or a CAD at the time of presentation in addition to criteria should be part of the algorithm, because also to monitor the downstream heterogenic effects? And my question to the leader.
So, sorry to clarify. So you were saying at the time of suspected myocarditis? Yeah, I mean, I think, and I'm sure there's going to be a whole, series on this discussion where the meeting, but certainly, at the time of the clinical presentations, essentially a total, invasive angiography or corneas CT, and they've also been added to around the clinical presentation to patient. I tend to lean forward more towards non-invasive because it does allow us more, extensive and arguably accurate imaging of degree of atherosclerosis. And it would be ideal to do both. But I think that
Didn't give you the biopsy.
yeah, and the biopsy again, that is obviously depending on your clinical suspicion, but also knowing that a lot of centers don't realistically have access to that. But I think, a particularly challenging clinical conundrum is when you have both. You have both acute kidney syndrome and epidermis myocarditis. And we have unfortunately encountered cases where there was kidney bias in ACS and people weren't really focused on the myocarditis, i.e. they sent to the patient and moved on. The patient started having blurry vision, muscle weakness, and no else being attached to the mat. So I definitely think there's these overlapping syndromes, which maybe fairly potentially worse prognosis, and your ideal whatever image you have, and also making sure that myocarditis is in the background, either to make sure that you should get. And as doctors alluded to, steroids are not ideal, but probably until we not more targeted and understanding mechanistic ways to use the therapy. That's also another important tool to get on early to reduce morbidity and mortality decision.
And really quick for Dr. Zhu, without any transaction medicine in one of your models, where's the mode of screening for undiagnosed autoimmune diseases?
Yeah, that's a good question. So, when we first started the troponin monitoring algorithm, we really were hoping to catch cases of early asymptomatic myocarditis and intervene before there's an opportunity for it to really take off with the positive immune feedback loop and become uncontrollable, at least by steroids alone. With other autoimmune diseases, I think it's a really fascinating question because currently there's not really any guidance to screen for these other autoimmune diseases, but there also is an opportunity to kind of smolder potentially. I think it's been a challenging area that's starting clinically just because, the evidence to say that there is an increased incidence of other autoimmune diseases in concurrence with IPSC myocarditis just hasn't been that clear. I guess concisely, we haven't screened for other autoimmune diseases, and we kind of started with the most fulminant thing, but there could be a role there. I just don't know. Yeah.
Please join me in thanking the speakers for the team. Next chair, next session.
Hello again. I am Rhee and from the Lincoln Royal University in Knoxville, Tennessee. It's my pleasure to facilitate this next session about the lives of non-woody RNA vascular stress response. And our first talk is about cardio oncology and pharmacogenomics with Dr. Rhee.
All right, so changing gears back to Dr. Hossein and iPS cells and some of our vascular phenotypes as well. So my lab works on using human iPS cells to model chemotherapy-induced cardiotoxicity. So I know everyone had a little bit about iPS cells this morning, and I just want to reiterate the power of this model, but also the complexity. So we see this model as essentially the only ideal way we can question questions to do with how the genome interacts with cardiotoxicity in a cell culture model. So iPS cells used to be a very boutique and complex technology. It was very hard to work with and very time consuming. And one of the things we tried to work on in my lab in the last decade is how to make this technology more amenable. So we're in a situation now where instead of doing smaller projects with just few, a handful of iPS cell lines, we can do projects up to 100 iPS cell lines at a time. And this is mainly getting about from us really being focused on simplifying of these culture conditions. So now we can take cells one bit of blood, go through this non-integrating and non-DNA modifying prototype technology, and make iPS cells that are essentially commoditizing whole technology. So this has involved generating a new media. So recently we published on the actual nutritional requirements of iPS cells, and now we have a cell culture media we call PMEM that actually pushes the cells more into the primary state. And in that primary state, that very locked in primary state, the cells grow faster, and the differentiation of various lineages is more reproducible. So the main lineage of interest for us is cardiomyocytes. We're working on differentiating cardiomyocytes. It's now our 20th year of working on this. And as you can see, we can make pure populations of cardiomyocytes. More recently, we've worked on whether we can make these cardiomyocytes have a phenotype that is closer to an adult cardiomyocyte. I don't like describing those fetal cardiomyocytes, but certainly they have some immature aspects. So we pulled apart the media for how this is done. We now have a new maturation media that really pushes these other kids ourselves a much more mature phenotype. So we worked on doxorubicin for a long time. I think, as many of you are aware, the dose of doxorubicin is limited by its causing cardiotoxicity. So for us, that tells us suggests an interesting idea that if we could essentially figure out why the cardiotoxicity happens, not only can make it a safer drug to use, potentially you can improve its efficacy as well. So we've had a few different numbers today of what percentage of patients experience toxicity, and that really depends on the sensitivity of the detection mechanism. It's safe to say doxorubicin is one of the most widely used immunotherapy drugs in the world. So that's a very important question. How do we prevent this cardiotoxicity? Just as importantly, how do we figure out which patients specifically are going to experience this toxicity? So nearly a decade ago now, we essentially established that the human iPS cell model, if you generate iPS cells from patients who have cardiotoxicity, their iPS cell, their cardiomyocytes are more sensitive to doxorubicin in vitro. So this is a really important finding for us, because not only does
it suggests that we have a model that we know works. It also essentially confirms that doxorubicin-induced cardiotoxicity is genomic in nature. For an iPSC, the only thing it's receiving from the original patient is the genome. The cells are fully reprogrammed, they're demethylated as part of that process. These are essentially new cardiomyocytes, so they're not knitted with environmental factors or other variables such as BMI that might be influencing the response to your drug. So it really tells us that we have a model in which we can essentially figure out the mechanism or this predisposing of these patients to toxicity. Now, somewhat frustratingly, when we looked at the mechanism involved, here's a simpler version of the image you saw this morning. We see that actually all of the major pathways that are related to doxorubicin cardiotoxicity are actually upregulated in these patients who were sensitive to doxorubicin themselves. So we had exogenous species DNA damage, the sarcomeric dysrhythmia, the type of mitochondrial dysfunction. So when we did this work, we were very excited that we had an in vitro model that seems to recapitulate the patient's sensitivity, but also very disappointed that it doesn't seem to be recapitulating on specific aspects of it. It seems to recapitulate a lot of different aspects. So now that we know that this disease is genomic in nature, when you can go back to the candidate gene and genome-wide association studies that have identified variants associated with doxorubicin-induced cardiotoxicity, and essentially query whether or not they're actually having an effect within our in vitro model. So we did a bit of work where we developed an algorithmic ranking of variants and genes associated with cardiotoxicity. So you've probably heard today of a lot of different drugs that are causing some sort of cardiotoxicity, whether it be cell death or myocarditis. Doxorubicin is essentially the only drug, the only chemotherapy drug that has any real-world data for us to be able to look at it in this way. So through this ranking, we've identified as 105 genes that could be associated with doxorubicin cardiotoxicity, over 400 individual variants, and we essentially worked through these, looking at the best associated, which is RNRG, the retinal receptor gamma, and then working down to the only variants that have ever been replicated within this in SRC28A3, which I believe you'll hear a bit more about later. So it was interesting because I didn't expect wanted to go back to these papers. What I was really interested in, now that we've ranked the variants, can we essentially validate their role in doxorubicin-induced cardiotoxicity in some sort of high throughput manner? So we added to the algorithmic ranking by adding expression data, either from adult fetal cardiomyocytes as well as like the cardiomyocytes, and then use CRISPR-Cas9 to knock down each of these genes individually. So we ended up knocking out 38 genes in total. It's the largest knockout project that have been done in iPS cells. And we developed this piece of data here. So it's a lot of data to take in one go, but previously we suggested that SLC transporters, they're the transporters that bring drugs into the cells. If you knock out an inward transporter, the cardiomyocyte will effectively get less doxorubicin and therefore be protected against toxicity. University ADC transporters are typically FOX transporters, and they push doxorubicin out. So you can knock one of those out, the cell is essentially seeing a higher dose. And somewhat satisfyingly, this completely unbiased data in all of these genes, purely based on algorithmic ranking. On the right there, you can see 3 different SLC genes are protected against toxicity, and then all the way to the left, you can see ADC genes are risk. So not only does this tell us out of all the data that's ever been generated on doxorubicin-induced cardiotoxicity, which genes have the largest effects, it also indicates which type of design of study is best powered. So
quite intriguingly, right in the middle there are RAG and cell4, which come from two of the GWAS studies. So actually, this is telling us that GWAS is very good at detecting subtle effects, but the candidate G studies will also seem to have ruled out the genes that had a larger effect on the toxicity. So we're in an interesting place now. There's within this list that be some very interesting genes. NY87 is among the most among the genes that when you knock out increases the risk the most. We don't really know why that is, but we did do some level of functional validation on top of purely kill curves. So essentially, we categorized each of the genes into 1 of 5 different families, whether they affect genetic function, species, or DNA damage, or drug transport, as we mentioned earlier. So this was published earlier this year. If you want to look at that more in depth, it is a lot of data to take in one go. But essentially, now we are aiming to rank variants to the stomach by essentially any means using this methodology. So moving on, we've also had a long-term interest in vascular disease, so chemotherapy-induced vascular disease, and much like we use doxorubicin as our prototype for cardiac disease, we've used nilotinib as the kind of hopefully clearest phenotype artery disease. So nilotinib is an interesting drug to us. It's a second generation of CML. The graph on the left there shows you that it is clearly more effective. That is 3 setter, and the imatinib that is. And on the right there, so this is from the 10-year follow-up. So from basic research labs, I can't work on the type of toxicity that was discovered 6 months ago. We need a huge amount of data because we're going to invest a lot of work. We have to write grants on this, and so we really need to have really good solid data. So it's been quite satisfying that we want to get that 10-year update, and it's at the point now where it's pretty conclusive that the toxicity from nilotinib is very much not only vascular, but specifically artery disease. So you're seeing ischemic heart disease, peripheral arterial occlusion, and then ischemic cerebrovascular disease in the right. So, and the numbers are pretty high. I can't work on a disease that is 1% of the population. We work on doxorubicin, which is 10%, and it's still very hard for us to recruit these patients. So we now know working a lot of different artery disease that the numbers should be pretty high as well. Not like saying recruitment of these patients is easy either. So our first study on this was really to do that work that we did at the cardiac studies when we made a very clean, reproducible model. It is easy to do these types of experiments poorly. We wanted to make sure that the endothelial differentiation is as high as quality as our cardiac differentiation. So we made a study set as we previously done with the cardiac genes, and essentially developed a differentiation protocol to pay 100% pure endothelial cells that can go through at least 9 passages whilst maintaining 100% purity for you. That kind of snaps notes. At the bottom right, there is an awesome value comparison between our iPS-derived endothelial cells and primary endothelial cells, and they're essentially perfectly aligned. So we're very confident the type of endothelial cells will make it our high quality. So, but then by having nilotinib versus imatinib, so 2 drugs are treated to patients who have the same disease, it means that we can do quite a good comparison. So that these could be age, etc. So we spent a lot of time comparing nilotinib to imatinib, and as you can see from this data, nilotinib impairs endothelial cell migration, proliferation, and then it increases nitrate oxide levels. Conversely, we know that nilotinib does not differentiate smooth muscle cells in the same assays, and so that's a big step forward for us. In a human model, we've proven that the nilotinib-induced vascular effect is specifically happening in the endothelial cells. So going back to the doxorubicin, the test that had to be proved that cardiomyocytes had a body endothelial cells or fibroid loss. So nilotinib is
and a BCR article targeting tyrosine kinase. So the real question that is exactly what is that drug doing? So we specifically knocked out ABL1 to see what this is an on-target effect of this drug. And from this work, we can see that actually it is not the specific effect of melatonin, making why it is targeting specifically has this vascular effect is off target. So currently we're just working on finishing up, look at exactly what the off-target effect is within these cells that is really kind of pushing on to the patient's effects going forwards. So I will stop there. This is my man for actually doing all the hard work. I appreciate the time, all the questions.
Our next talk is about molecular omics with Dr. Maedeh.
Today I'm going to follow on what Paul has already been showing. So I'm going to hopefully show you a good example of using PURT use for the stem cell. So basically, first of all, understand the role of SNERS in doxorubicin use cardiotoxicity, and more importantly, using this cellular model to discover new cardioprotective agents. So what you can see here is that basically on the x-axis, the timeline, and on the y-axis, the number of drugs, the $1 billion US dollars invested in research and development. And as you can see, the drug discovery process has been not consistently providing, and it is not as efficient as we would hope for. And the main reason why that is basically the lacking of efficacy of new novel drugs in the drug discovery pipeline. And if you can see those bar charts, you will find out that 52% of drugs fail in the drug discovery pipeline because of the efficacy. So and this percentage actually increases from I think 48% in clinical trial phase 2 up to 55 or 58 in clinical trial phase 3, where more patients are recruited. And actually what this study already emphasizes is that we need more human relevant cellular model to complement the models that we are using right now, because basically the translation of the HTC in the big learning model as well considering what in humans. And secondly, it also emphasizes that we need another source of information to guide us which gene to target to treat certain diseases. And here comes the role for pharmacogenomics. So over the last 20 or 30 years, we have experienced a huge empowering in all aspects of pharmacogenomic research over the research. All of this success stories have resulted in a significant increase in the cost of one single human genome sequence of $100 million. And this was only 20 years ago, to less than $1,000 nowadays. And this is associated with the number and increasing number of genome association studies that have been published since 2015, as you can see. And actually, this study here shows that drug discovery projects that started with a human relevant genetic information showed significant high potential success rate in drug discovery volume. So on the x-axis, you can see the GCOS rate, drug similarity, drug indication similarity, and on the y-axis you can see the success rate in the approval rate in the drug discovery project. That you can see from this red line here is that the drug discovery project started with GCOS rate, showed almost twofold higher success rate compared to the drug discovery project that did not start with any human genetic data. And actually, this alteration increases from 2-fold up to 4-fold if the GY has normal deleterious effect. And it even increases up to 8-fold in case of mammalian traits, which makes sense because those diseases, they have maybe one gene or a few genes that can be disposed to these inhibitors. So how can we use iPSCs to, how can we integrate iPSCs into this process? So normally what happens in genetic research is that you have a patient with the disease and not gene control, you get the DNA, send your blood sample, you get the DNA, and then you genotype millions of steps, or you go or genome sequence, and then you do the genotype phenotype analysis, and eventually you identify a SNP that is associated with your disease of interest. However, this is very important information, of course, because it gives us some information on the genes involved with in the disease that we're tested in. But this is just statistical affiliation, so it doesn't tell us basically what is the cause or how does this affect ability in the disease that was tested in. So here comes the iPSCs technology. So from the same blood sample, we can isolate PDGFR point those PDGFRs into CD cerebral monitor cells, and then reprogram those to have iPSCs that is prior to the patient with that SNP. And using the CRISPR-Cas9 and other gene technology, you can either edit this SNP back to the reference allele and then differentiate those two cell lines into whatever cell type that we're interested in, and then couple these cells with multiple high throughput functional validation assay. You can actually identify the causality of this SNP, at the beginning of action, this cell inhibition to the studied phenotype. So a good example of such process is those sorts of induced cardiotoxicity, and you have heard a lot of bookshelves in cardiotoxicity. Today, I just want to point out to this Australian market forecast research, and it shows that the usage of something is increasing. So currently, we are, around $1.2 billion, the market value looks promising, and it is expected to increase up to 1.8 in 2027. And considering that the cost of this chemotherapy, there are thousands of patients treated with this chemotherapy every day. However, it is very important because of the severe cardiotoxicity, which basically we need to identify novel cardioprotections that can, mitigate this adverse event. This graph here again, so this graph comes lines today. It shows genes that can help with in the mechanism of induced cardiotoxicity. A lot of genes. It's clearly a polygenic phenotype with many genes involved in such toxicity. So, those two forest plots summarizes SNPs associated with induced cardiotoxicity, SNPs related in doxorubicin pharmacodynamic and pharmacodynamic pathway, and those are ranked by their script size and the p-value and significance. And as you can tell from those two table of two forest plots, there are two SNPs that are well replicated and most significant in the context of doxorubicin cardiotoxicity, as there is an receptor the acid receptor GABA, and the other SNP is in the solid carrier transporter called SRT28A3. Today I'm going to explain this project that basically focuses on this solid carrier transporter. And one of the studies that pinpointed this association was published more than 10 years ago, where they identified a SNP in the solid carrier transporter, and this SNP actually is associated with a lower cardiotoxicity in the pediatric cancer population. So patients with this SNP were basically protected against doxorubicin-induced cardiotoxicity. So what we have done is that we recruited a bunch of patients from this study. So we recruited 3 patients with the reference allele, basically, and another 3 patients with the variant allele, that did not show any toxicity, and one healthy individual that actually did not have any steps associated with toxin toxicity. So the issue what I apply is that we first observed the genotype is spent in the series patients. We observed that this transporter is expressed in our cardiomyocytes, and then we assessed basically, we wanted to see whether or not our model can recapture the anti-mutated percent. So we did a cell viability curve, basically a curve treating those cells with increasing growth of the thrombosin, and we have found that basically the SLC variant cells are kind of more resistant to the thrombosin compared to the SLC reference cells. We confirmed this by quantifying that this has this B7 activity, and since this is a solid transporter, so we thought to look into the so basically, yes, sir. So we looked into the cardiovascular accumulation of toxin in those cardiomyocytes derived from patients without cardiovascular, and we found that patients with this have significantly lower intracellular accumulation of toxin compared to control cells. And also lastly, we did protein quantification, and we found that the protein expression on the soil transporter is decreased in the patients with ourselves from the patient with this study. So then we wanted to confirm the identification of this gene in cardiomyopathy toxicity. So we also did CRISPR-Cas9, and we knocked out this transporter and overexpressed the transporter into an isogenic cell line derived from NC individual. And we did the assays. We found that the cells that overexpress this transporter are more sensitive to toxin compared to the knockout and the overexpressive line. And also they had more apoptosis compared to the knockout and the isogenic cell line. And also the overexpressive cell line showed a higher toxin accumulation compared to the knockout and the isogenic cell line, basically. So then we moved to another question, which is basically the GCOS identified kit is according to Carol's negative getting age from 14, so it does not alter the amino acids. So we had this question why basically what is the latest basically says it does not alter the amino acid. And having considering the fact that these steps are inherited in haplotype stranges, they are not inherited as a single clone. So we hypothesized that this step might be inherited with other steps, including the other ones. And to test this hypothesis, what we have done is that we basically developed a most effective sequencing, targeted sequencing platform in which we basically divided this the gene locus that encodes for this gene into 9 different pieces, and then we PCR and combiners overlapping 9 epitopes. We coded all of those epitopes for each sample, and then we sequence those samples using the number sequencing. We combine those sequencing, and then we have a single peak that overlaps with the SLC2883, and then we look into steps identified in our patients. So we focused mainly on steps that are co-inherited in the 3 patients that actually were protected from cardiotoxicity, but not in the 3 patients that actually did have cardiotoxicity. And it turned out to be that there are 24 steps constituting the haplotype that basically can inherit in one block. And we have confirmed this using by doing genetic equilibrium analysis of the 100 individual with original sequence. And then to basically rank the likelihood of these steps to be because we have used or we have done in simple functional validation, looking into EQGL database and basically chromatin regulatory database, and also regulatory region and cardiac tissue. And based on this analysis, we have identified a set that is most likely has the most likely to be ozone. And interestingly, this threat is located here in the first exam. In the slide side, the first exam of overlapping RNA that overlaps with our target gene. And so basically, what we have done next is that we basically edited this step in two of the cell lines back to the wild type, and then we looked into the effect of editing these edited cells versus unedited cells. And we have found that by editing this variant back to the reference allele, those cardiomyocytes became more sensitive to those chromosomes compared to the non-edited cells, and also they had more intracellular accumulation of those chromosomes compared to the non-edited cells. So we still follow that this alone is potentially implicated in starting rotation against toxoxide-induced cardiotoxicity. So since this study is related in the strong non-coding RNA, so we hypothesized that the cardiothoracic effect might be related by the strong non-coding RNA. And to this is hypothesis, we have overexpressed the strong non-coding RNA as it relates to line. And then basically we looked into the expression of Blaes in cardiomyocyte, and we have seen that by overexpressing non-coding RNA, basically is actually in the expression of this transported in our cells. And then we ran the curve, which basically confirmed that overexpressing this strong non-coding RNA renders the cells less sensitive to doxorubicin, and also it decreased the intracellular accumulation of doxorubicin in heart cells. And then at this point, we have provided a proof of concept for the application of cellular transporter in doxorubicin cardiothoracic. So we wanted basically to follow up with that to see if we can inhibit doxorubicin uptake in the blood cells, and in such way we can protect cardiomyocytes from doxorubicin cardiothoracic. So what we have done is that we have selected 17 million SLC substrate, and we basically tested the whether or not this substrate can increase the intracellular accumulation of doxorubicin. And screening those 17 molecules, we have found that CRISPRi was the only one that actually decreased intracellular accumulation of doxorubicin both 1 hour and 3 hour post treatment. And moving from this cherry-pick screening to more kind of unbiased screening, I mean, this therapy is truncated for whatever reason, but what we have done here is that we tested the cardioprotective effect of 1,200 drugs. So in our cardiomyocytes, and then this drug screening resulted in 34 cardioprotective molecules. And we looked into the top 10 cardioprotective molecules, and then basically out of which we identified 6 FDA approved. And since cardiomyopathy is more fat, we quicker as compared to the drug discovery, we focused only on FDA approved drugs. And interestingly, we can support that Disipramine is one of those top 6 drugs. We observed the result that we have found in the smart screening by basically calculating the efficacy of all of these drugs. And again, Disipramine worked out to be one of the most protected out. And here you can see the kill curve of cardiomyocytes treated with Disipramine, anti-doxorubicin cells treated with only doxorubicin, showing the hypothetical Disipramine. So and then we moved to just the cardioprotective effect of Disipramine in vivo. So we have treated mouse and mice with doxorubicin alone and doxorubicin plus Disipramine. And what we have seen here is that mice treated with Disipramine showed significantly lower decline in cardiac function quantified by observed children interaction after 3 weeks of treatment. And costly to, to rule out any possibility that this drug might affect the side of the anticipating might affect the side of the system because obviously that tested that in a lot of the contract providing and it did not see any other cytotoxicity of those things and pointing that other stuff with this is going to plus botulinum. So to sum up, we have shown that the idea with the stem cell is basically a very good model to currently suggest that there is a something induced by toxicity. We have this underlearners, this transporter, and we have discovered an organ tumors that is highly protected, and what we can, translationally speaking, what we can do that we can screen for this before during the drought, and then basically start the ovulation based on the likelihood to get towards growth setting, cardiovascular, and from those stations that are most likely to be a cardiac protector, we can actually increase the dose effect or increasing the time of treatment so that they have more anti-cancer basically based on that. And basically that was it. And now let me send my number and also my forward that number this work has done any was that. So thank you all. And thank you for your attendance. Yes, we definitely give more.
I guess I would like to take my question. Or not. Oh, there it is.
Bob, actually, the guy looking at FCs. So on these people there to feel like you can get out to to give up on parts that in private those the most yes you guys that think that no art I mean the motivation is down. So I'm not go there anymore. Yeah, if you get out, but they should advise them on that. Sorry, so what's the question? You have to letting it now to keep doing what you want to do is sleep, right? So, if they can go directly looking at the pick out expression level genes. So, how do you think like that? Can that be like, more faster to identify genes and burn? There's two different angles to that. One is when a gene was discovers that he's not necessary and the definition for the story is a state project and control at the level biology discover the area to defend usually it's just an all seconds that needs to do it. And then when it comes to the changes in gene expression, the reason we're so interested in you say in a base that's been generated for toxicity GCOS is it means it discovers a very gives what previous tradition to the drug response, but it's not going to be it's otherwise essential. It's good. So we're very careful not to look at just any achieving, talking to title based by mitochondrial that would be the best thing to knock out. We can't get that something bad. So by using this GCOS data, I think that's what gives us the most viable targets. We are moving on to do much more the transcriptional side, that involves projects to be hundreds of years down the line. It is very, very exciting. The enormous work and people to find our brand exactly how one can that is the day.
Just add one more thing. So when you looking at it actually gives us, some information regarding the gene, like the expression of the differential expression of gene, right? But it doesn't help out in terms of identifying posers. Because if you would look into the EQPR with these 24 cells that are co-inherited all together, all of them will give you an EQPR signal, which that is speaking. You have the same, right?
Therefore, I get it.
Please join me to say if this is development. I think what's the problem or the
So I'm back here on the first session for the last session. This is our training award quotation. The committee selected from 4 abstract-based presentations. Now you will have everyone present themselves with notes. Starting off with John Lee.
It's just you have to associate that you have a problem with that. Did you ever in the first use of what are really what the edit by that and Indra, out of the people that are kind of one office. The sense between putting that much digest as edits over the website. But that you can imagine one one with different causes. Like infection, autoimmune disease, vaccine, and also the medication that was done for immunotherapeutics inhibitor in US hepatitis in cancer therapy. And you see that Dr. June-Wha started a center to address the critical gap between the patient care and the science. The most problematic one, which is the extra permanent vascular. And this coordinated and it is characterized with sudden and severe cardiac intervention, leading to the clinical shock and failure arrhythmia. Currently, there's no approved therapy for this active pulmonary myocarditis. We've been doing other research for many years on immune myocarditis. And what we've learned is JAK stimulating pathway is very important in ICM myocarditis, as just Dr. Khan who had been mentioned. The crosstalk between the macrophage and the T cells of ICM myocarditis mediated by the interference with the interferon gamma and the cytokinase with the IL-2, IL-6, they are related, they are not, and through the JAK stimulating pathway. And the ruxolitinib, which is a non-selective JAK inhibitor, has been showing promise in other inflammatory conditions, for example, myelofibrosis and GVHD including. We have a lot of comparing evidence to show that JAK inhibitor improves the ability of outcome of ICI myocarditis. In preclinical models, we use the genetic knockout in mouse model to mimic the ICI myocarditis. The first line of line therapy like steroid and the immunotherapy failed to rescue the mortality. Or the immune checkpoint inhibitor, the JAK inhibition inhibitor also include ruxolitinib. So you can improve the survival of ICI myocarditis compared with vehicle control. And also the multi-center patient experience shows that for the ICI myocarditis with the respiratory heart muscle failure patient, if they treat with the protocol with ruxolitinib, it is dramatically reduced the mortality by 60% to only 3%. Based on these findings, we hypothesized that ruxolitinib can also improve the clinical outcome in active pulmonary myocarditis, and we believe that a OMICs study can offer valuable mechanistic insight in the molecular network. So this is the first active pulmonary myocarditis treated with ruxolitinib. She's a 20 years old young female presented with chest pain and fever, showing severely reduced heart function LVEF only 10% with cardiac shock and acute elevated the biomarkers, cardiac tests. She was transferred to New Delhi and placed on mechanical support with F1 and the heart biopsy showed that a coronary myocarditis could actually spring. She started with the high dose steroid and the resistance, but her condition is still critical and she was on the heart condition standard 1. due to the continued generation overrides with the added of the zeros and the privately, and we observed subsequent improvement of order cost structure and also time of motor improvement, and over time heart function improved from improves from 10% to 80% by day 8, and achieves the with the mapping the support of Ibdala and Apple. She was discharged day 19, and she's doing very well. 2 months follow up. To study the molecular mechanism of the repeated causal, we expected the column tissue pre-treatment at also the blood samples at 4 times different time points pre-drugs, but after steroid, day 5, day 8, and 2 months. So at baseline, we observed the elevated jets that different pathway, particularly in the both peripheral blood and also column tissue. On the left side, there's the heat map shows elevated jet expression in the PDGFR, as well as the downstream status and also related gene signaling. The ruxolitinib or chosen treatment targeting JAK1, JAK2, and also TYK2 are not JAK3, which is a perfect fit for all patients because all patients JAK3 is not significant but regulated. iPSC study shows that iPSC staining of the cardiac tissue shows the presence of CD3 positive T cells and also CD68 positive for macrophage, as well as the elevated expression of HLA-DR. HLA-DR is interferon inducible inhibitor, which indicates the activated inflammatory process mediated by JAK cells in the participate cardiac tissue. This providing the treatment rationale for the using the ruxolitinib for this active pulmonary myocarditis. So our research plan, we including multi-single-cell omics to look at the rapid recovery of drugs treatment, including the FACC auto-antibody protein and 10 single-cell RNA sequencing for the transcriptomic and TTR PCR sequencing for the TTR PCR laboratory, and also cell surface protein section for the proteomics. Longitudinal single-cell proteomics by CRISPRi and also transcriptomic by single-cell RNA sequencing shows consistently the dynamic change in the immune cell population. As you see, the red color monocytes and also the gold color NK cells significantly increased after 80 days treatment. More details with the RNA sequencing on the genomic shows that the target reduction of pathogenic NK cells and population of monocyte or intimate monocyte. This monocyte is the high structure of CDR2 and also HLA-DR, which is involved in antigen presenting and gene presenting and cell collaboration and collaboration and the stimulation of the T cell. This is also the cell type that infiltrates the heart, causes the myocarditis. Ruxolitinib also leads to the significant difference of the downstream JAK signaling, step 1 and step 3 in the virus cell type, which is consistent with the mechanism of action of the rux. If we focus on only myeloid cells, we observed there's a near complete operation of the specific myeloid cell types. And these cell types usually in red color only shows in pre-treatment. And this class where offered by the outside members on interferon stimulations. And these genes are associated with the immunoactivation. There is a wide variety of hypoproliferation, which is completely inhibits often cross treatment. If we focus on lymphocytes, and we also observed on the left side of the graph shows ruxolitinib show reduce the pathogenic lymphocytes of the cells and CD8 T cells. And these T cells are also a driver of ICI mark values, and as well as the donor T cells until significant decrease. On the right side, there's the warning. On the right side, the warning plot shows that the critical immune toxic genes like coenzyme B in the nuclease suppressed after treatment. Ruxolitinib also reduce the T cell lymphocytes. On the left side, there's the bar graph shows the hemoglobin increase after the treatment, and the hypo expanded like more than 100 colonel had decrease after the treatment. On the right side, the channel diversity also shows that BCR, TTR, colonel diversity increase. This indicates a normalization of the immunotherapy and with a more diverse and balanced population of T-cell B cells. So, in conclusion, our key findings is the closer that's for treatment of active pulmonary myocarditis with proximity, and which leads to the rapid clinical recovery. And we observe the checks as the pathway hyperactivation in both blood and the cardiovascular, and which provides the rationale of the treatment. And they got immediate after treatment, which supports the mechanism of action. One OMIC study demonstrated that Blaes treatment reduced the pathogenic immune cells and suppressed the cell health genes and restored the T cell and B cell diversity. This study indicates that targeting the JAK-STAT pathway shows promise for treating the active pulmonary myocarditis. And the efficacy we observed that therapeutic proximity by non-steroid. And these results support analysis scale clinical responses. Most importantly, there are 4 myocarditis patients who have been successfully treated without oxygenated adequate that. Thank to all the event extension of Cardio Oncology, Immunology, and our collaborators and patient, and especially thanks to German that she already said evidence and support. Thank you all for your attention. Thank you.
Thank you. That's the context of that too. My name is Rhee Chen, and I'm the director of the Rhee Institute. It is now my pleasure to introduce the next contestant in this wonderful training awards competition. Dr. Rhee Hossein will present a talk. Thank you very much.
Thank you for your introduction and thank you for giving me the opportunity to present my work today. So the title of my work is New Onset Cancer Treatment Dysfunction is uncommon in patients with HER2-positive breast cancer after functional cancer therapy. Implication for imaging surveillance. Breast cancer is the most common cancer in female. We do not know why women with breast cancer have a HER2-positive disease. Currently, adagrasib are still being used as a treatment for HER2-positive breast cancer. And cardiovascular is the recurring side effect of all treatment. These steps of asymptomatic LV dysfunction is actually in the study during treatment, and the consistent rate is about 2 to 4% during treatment. In the previous single study P31 trial, comparing the cardiotoxicity between anthracycline, cyclophosphamide, paclitaxel without trastuzumab. The cardiac event, which was defined as NYHA class 3 or 4 heart failure or cardiac death, was higher in patients who were treated with trastuzumab, with incidence of 4.1% compared to the patient who did not receive trastuzumab, with incidence of 0.8% during a 3 years follow-up. The current ESC guidelines suggest routine cardiac imaging surveillance during targeted therapy, with repeated cardiac imaging with every 3 months during cancer treatment. However, after the completion of cancer therapy, the frequency of the cardiac surveillance remains unstable. And in the absence of the clear evidence, the ESC guidelines suggest that echocardiogram should be performed at least once at 12 months after undergoing cardiac therapy. In the long-term surveillance, the echocardiogram is recommended according to the patient's risk, and most of the patients with breast cancer is classified as moderate risk due to the cumulative dose of doxorubicin received and the radiation dose that they receive. So echocardiogram is recommended every 5 years for patients with moderate risk with a class 2 recommendation. For patients with high and very high risk, such as those who receive doxorubicin more than 250 milligrams per deciliter, echocardiogram is recommended at year 1, year 3, year 5, and then every 5 years. The longer-term surveillance have a great level of evidence suggesting a limited data. So data on the timing of cardiotoxicity during and after therapy can be valuable to provide guidance for long-term follow-up. Therefore, the objective of our study was to determine the time and incidence of cardiotoxicity, both systemic and diastolic dysfunction, in patients with HER2 positive breast cancer after completion of cancer therapy and compared to the incidence during therapy. Based on clinical experience, we hypothesized that most events will occur during therapy, with fewer events during long-term surveillance. The iPSC trial was a prospective cohort study that enrolled adult women with stage 1 to 3 HER2 positive breast cancer, scheduled to receive anthracycline and trastuzumab with or without adjuvant radiotherapy. And we exclude patients who have contraindication to cardiac MRI and to the administration of gadolinium-based contrast agent, as the primary aim of the breast trial was to assess myocardial tissue characteristics during cancer therapy. And we also exclude patients with life expectancy less than 12 months, patients who have prior exposure to anthracycline, and patients with history of cardiac disease. The recruitment occurred between 2013 and 2019, and echocardiogram was conducted at baseline and every 3 months during therapy at the end of trastuzumab, and then at year 1, year 2, year 3, and then year 5. Clinical visit was also conducted at baseline at 6 months at the end of trastuzumab, and at year 1, year 2, year 3, and then year 5. The comprehensive echocardiogram was performed by dedicated and experienced sonographer. The LV ejection fraction was analyzed by 3D data using the 4D ortho amplitude, and that function was classified into a diastolic grade based on the 2016 American Society of Echocardiography guideline. And as we began the enrollment in 2013, we used the CRISPRi cardiotoxicity criteria to define CRC. And the CRC was defined as a reduction in PDGFR by 10% or greater from baseline to less than 55%. And that dysfunction was defined as any diastolic grade more than 1 or higher, or worsening from baseline by 1 grade or higher. And this is the baseline characteristic of the patient in our study. That 136 female HER2 positive breast cancer with the mean age of 51 years old, and only a small proportion of patients have traditional cardiovascular risk factor. And the most common immunotherapy recommended use in this study was the FACC regimen that contained Inhibition and Translational. The mean dorsal visit equivalent dose was 304 milligrams per deciliter, and 88% of patients received radiation therapy. And when we consider the baseline severe risk according to the HFA high-cost criterion, most of the patients in this study were at moderate risk. Moving on to the main results, which is the incidence of the CRISPRi. So during cancer treatment, 27 out of 100 patients, or 19.9%, developed CRISPRi, and all patients were asymptomatic. The most common timing of the CRISPRi was at 9 months during treatment. And in the long-term data, due to the COVID-19 pandemic, the follow-up was available at 128 patients at year 1, 170 patients at 2 years, 111 patients at 3 years, and 98 patients at 5 years, but the follow-up is still ongoing. And after completion of cancer therapy, there was no new patient who had CRISPRi, and about 18% of them still have persistent LV dysfunction in the long-term follow-up. For gastric dysfunction, 19% of the patients developed gastric dysfunction during the treatment, and most of them were classified as grade 1 gastric dysfunction, and the timing of the gastric function occurred mostly at 6 months during the treatment. After completion of cancer therapy, only 6 patients had new gastric dysfunction, and about 30% of patients had persistent gastric dysfunction in the long term. And since we did not have any new CRISPRi after completion of cancer therapy, we want to perform exploratory analysis as to factors that may have contributed to the development of new gastric dysfunction after completion of cancer therapy. We realized that given a small number of patients who developed new gastric dysfunction, this was the only exploratory analysis. And we look at age, baseline cardiovascular risk factor, dorsal visit equivalent dose, radiation dose, CRISPRi during treatment, and the baseline severe risk. And none of these risk factors were associated with new gastric dysfunction. And we also want to highlight that the wide confidence interval reflects the small number of patients with new gastric dysfunction. Our patients will also follow clinically for the development of any cardiovascular risk factor and were treated appropriately as per the standard of care. 72% of the patients now have clinical data follow-up up to 5 years, and we have seen about 9% who have new hypertension, 5% had new diabetes, 10% were started on an ACE inhibitor, ARB, or little longer because they developed new or worsening hypertension, and 4% were started on SGLT2 inhibitor into diabetes, and 13% were started on statin. In summary, we observed that 1 over 5 patients developed asymptomatic CRISPRi during treatment, and in the long-term follow-up, there was no new CRISPRi. 1 over 5 patients who developed CRISPRi during cancer therapy have persistent cardiac dysfunction in the long term. For gastric dysfunction, 1 over 5 patients developed gastric dysfunction during treatment, but in the long-term follow-up, only 6 patients had new gastric dysfunction, and 1 over 3 patients who developed gastric dysfunction during cancer therapy have persistent gastric dysfunction in the long-term follow-up. Most of the patients in our study are in the moderate risk group, and our data support the current ESC guideline recommendation that the moderate risk patients do not require ultrasound more than 20 and 25 years post-therapy. But we don't have sufficient evidence to support the recommendation for the high-risk group, as only 2% of the patients were classified as high-risk in this study. Our study has some limitation. So our standard study include only one basic patient. However, the strength is that we have a meticulous imaging with over 1,000 picogram done. We do not have complete follow-up in all 136 patients, but the remaining 38 patients will complete follow-up by next year. And the baseline unfortunately is similar to the population that already has a follow-up. And we don't have follow-up data beyond 5 years, but we still following them ongoing. So in conclusion, our findings suggest that the level of cardiac rotation cardiac imaging after conventional therapy in patients who did not develop CRISPRi during treatment is low. Patients who develop CRISPRi during treatment still need a long-term imaging surveillance. In our program, patients will follow Ardehali and implement of cardiovascular risk factor as per the standard of care. The clinical follow-up and risk factor management may have contributed to the lower extent of CRISPRi and thus its dysfunction in the long-term follow-up. So we suggest that clinical follow-up and risk factor management are significantly in this population. Thank you very much for your attention, and I would like to thank my Cardio Oncology teams for joining me for supporting me.
Thank you, Dr. Olser, and that very excellent presentation. I'd like to ask the next interviewer, Dr. Keung, to give his talk. Thank you very much. From South Africa, the most South Korea. It is an honor to present our work in the Global Cardio Oncology 2024. The topic I'm going to talk is about the adherence to non-diabetic medication and cardiovascular outcomes among patients with cancer on nationwide population-based core study. The question is about the major cause of death, and it is widely recognized as a cardiovascular syndrome. However, in recent years, there has been an increase in their cancer patients that are at risk due to early detection and advanced treatment. In the United States, the number of cancers survivors was estimated at 18 million in 2022, and is expected to reach 6 million by 2032. Similarly, in the UK, the cancer survival rates have doubled over the last 4 decades, noting that 50% of cancer patients now live for at least a decade after their diagnosis. These improving survival rates have been a limitation to long-term scale of cancer patients. Cardiovascular disease is the second most common cancer cause of death among cancer patients, which is only caused by cancer cells. And cardiovascular disease and cancer share several risk factors such as smoking, alcohol consumption, and physical inactivity. And cancer biology characterized by low-grade inflammation. Therefore, proper measurement of cardiovascular disease and cardiovascular risk factors are important to achieve a better survival in cancer patients. The diabetes, treatment risk factors for cardiovascular disease is a prevalent commonality in cancer patients, which is present in 25% of individuals with cancer. Comorbid diabetes in cancer patients significantly increase the risk of complication of cardiovascular disease or infection. Simultaneously, the presence of cardiovascular disease or infection in cancer patients can directly lead to adverse outcomes, including death or hospitalization. Furthermore, these events may have indirect consequences by disrupting cancer treatment, thereby worsening outcomes. Therefore, the identification and the measurement of diabetes is critical for comprehensive cancer patient care and for safer management. And the integral component of diabetic care is adherence to diabetic drugs, with this perspective in mind, our current study aims to investigate the relationship between anti-diabetic drug adherence and clinical outcomes in cancer patients. For the study, we used data from Korean NHL, National Health Insurance Service, which can have insurance system covering the entire population of South Korea. The Spanish Chinese National Study Cohort is known for one of the representative national population-based cohort, giving researchers and policymakers information about abstinence drugs usage and services among citizens. The cohort recruited 1 million individuals, 10% of all population in Australia, from 2002 until 2006 and tracked them until 2013, which is a total of 11 years of follow-up. These database includes demographic characteristics, diagnostic costs, restrictions, and inpatient and outpatient details. Focusing on the patient diagnosed with cancer, we found 99,000 cancer patients in the database. From here, we only included patients with 10 most frequent cancers and excluded those who had previous cancer at baseline and with other exposure criteria such as very young age and old age, very low income, that either had cardiovascular effects at very early of the time period. And to keep only 2 diabetic patients, we used 2 medication. Ultimately, the study comprised 7,900 individuals. Then we analyzed the adherence to the antidiabetic medication in the study population. Here we used the medication production ratio, NPR. This ratio is calculated as the days of supply prescription divided by the refill period. Patients were categorized based on their NPR scores into 3 groups: good NPR over 0.8, moderate 0.5 to 0.8, and poor NPR below 0.5. And adherence was defined as an NPR below 0.8 in line with other published studies. With all this data given, we used Kaplan-Meier analysis, post-nutritional information model, and SQL expenditure examination. And to analyze a relationship between all-cause and cardiovascular mortality, which is a primary study outcome, and other various cardiovascular events. In 7,900 cancer patients with diabetes, we found that 14% had NPR values over 0.8, which is a good adherence, and 22% as a moderate, and 37% as a poor adherence, leading to the result that nearly 60% of cancer patients with diabetes are not inhibited. And between groups, they had similar preferences of gender, age, income level, and other variables. And the initial therapy usage was a little higher in the moderate adherence group. However, the mood and trauma adherence group shared similar initial therapy usage. To see the association of antidiabetic medication adherence and overall survival, in cancer patients with diabetes, the Kaplan-Meier curves were plotted, and it revealed that the good adherence group showed the highest survival rates followed by the moderate and poor adherence group. As subnormalities by age and gender show generally similar trends to the Maedeh index. In the first group, we have a variation level, which adjusted for gender, age, residential area, income level, and previous history of cardiovascular disease. In comparison to the good adherence group, both the moderate and poor adherence group exhibited 1.70-fold and 2.11-fold increase in the risk of all-cause mortality. Especially for regarding the cardiovascular mortality, the moderate and poor adherence group demonstrated 1.51-fold and 0.10-fold heightened risk. Also, both the moderate and poor adherence group exhibited 1.32-fold and 1.24-fold increase for the incidence of new onset of any cardiovascular disease. Consistent findings supports the risk of associated with non-adherence to antidiabetic medications were observed. For each new onset of specific subtype of cardiovascular disease, such as ischemic heart disease, peripheral artery disease, cardiovascular accident, and heart failure, risks are increasing in the moderate and poor adherence group. Not only the risks are elevated in regarding the non-adherence, and that actually analysis on healthcare expenditure based on antidiabetic medication adherence, when the total healthcare cost of sum of all healthcare insurance reimbursed costs from the initial diagnosis of diabetes for each individual until the end of the study period were defined by the number of individuals. The healthcare costs per individuals were significantly more higher in the non-adherence group
which is the moderate and poor adherence, and which translates to good adherence to have significantly lower medication experiments compared to the others. As a summary, in cancer patients, about 60% are not adherent to antidiabetic medications. Risk of all cause and cardiovascular mortality increase in the higher levels of non-adherence, and non-adherence was associated with the development of non-new-onset cardiovascular disease. And healthcare costs were increased in the non-adherent group, non-adherent patients, giving the importance of clinical attention to ensure adherence to antidiabetic medications among cases with cancer patients with diabetes to prevent adverse cardiovascular events. On this study had several limitations. First, given its retrospective nature, examining the catch-all difference between the medication and adherence and clinical outcomes is challenging due to potential adverse or other component factors. A specific numerical values for blood glucose control levels such as HDL-C are lacking, limiting DTM adherence to glycemic control. Analysis did not differentiate between classes of antidiabetic drugs, concerning that medical adherence among cancer patients due to a small sample size within each drug class. And despite utilizing a large nationwide database, the study had only related to a small number of eligible participants. Well, numerous studies have highlighted the significant role of adherence to antidiabetic medication in glycemic control and reducing bipolar and macrovitamin conversations. Our study highlights more importance of antidiabetic adherence in cancer patients. Previous meta-analysis study analyzing 318,000 individuals showed that non-adherence was only 30% in the general population, and in the study in the Korean population, the non-adherence was 42 and 63% in another general population. Compared to the current study in cancer patients with diabetes, the non-adherence was 60, resulting that the non-adherence of antidiabetic is still more higher, not lower in the cancer patient compared to the general population. And for the risk of all-cause mortality and the incidence of cardiovascular disease and ischemic heart disease, the previous literature of general South Korean population, similarly using the National Healthcare Insurance Database, showed that even though the individuals at the lowest adherence, the proportion of days covered, similar to IPR in their study, is below 1.2, and the hazard ratio for all-cause mortality is 1.45 in the general population. However, in our study, the all-cause mortality was over 2, and the incidence of cardiovascular accident and ischemic disease is more higher than the general population. As a conclusion, this is the final slide, and maintaining quality control in diabetes, particularly through ensuring good adherence to antidiabetic medication, is crucial for preventing adverse clinical outcomes, including overall cardiovascular mortality and reducing healthcare loss, especially in cancer patients. And second, addressing this alarming trend of adherence in diabetes on a cancer patient is crucial for improving outcomes. And checking medical adherence to other chronic disease in cancer patients are also needed. Thank you. That's all for today, and for this sheet.
Thank you very much, Kathryn. And now we will have the final contestant for this afternoon, and we will welcome Dr. Rajat Satya.
Thank you for the panel, for the audience, for the organizers, for the kind invitation. My name is Dr. Rajat Satya. I'm a cardio oncologist and advanced heart clinical transplant fellow at WashU. And today I'm pleased to present our title, Cardiovascular Events in Older Patients Receiving CAR-T, and analysis of the nationwide medical beneficiaries. So I have no relative disclosures to the talk. Just a brief cartoon of how CAR-T work. Basically, the patient's not is collected. He's had no isolated, not that genetically modified to express chimeric antigen receptor that are targeted against certain specific molecules of the CAD cells. And then these T cells are replicated in vitro to become millions, and then they are reinstilled back into the patient with the hope of targeting cancer population without affecting the normal tissue. So it's one part of the non-dilutive therapy on the market. Over the last 7 to 8 years, the CAR-Ts have been rapidly developing. Starting in 2017, we started with having CAR-Ts targeted against the Hodgkin lymphoma, and then over the years, we have now one targeted against mantle cell follicular lymphoma. In the last 2 years, Rotelizumab Plus targeting multiple myeloma patients. So the initial trials and what we know about the status of the cardiovascular system. So in the early trials, there was very minimal cardiovascular toxicity of these CAR-T agents. But then once we started using that in the general population, there are several small scientific reports, mostly single institution or registries, that linked the significant cardiovascular adverse event when they used this CAR-T. And the third was that maybe patients who were enrolled in the clinical trials were carefully selected and did not have high learners and they might have cardiovascular risk factors. So what these studies also find that these patients had a significant incidence of major adverse cardiovascular events, and the range was very good depending on how these studies define MAS, but mostly were in the range of 5 to 10, 20%, as varied by the population and by the center. Most of these studies also showed that the incidence of MAS was associated with increased short-term mortality. These studies also linked the occurrence of MAS with high-grade cytokine release syndrome, and they suggested that the early use of the immunosuppression interleukin-6 receptor blocker tocilizumab to be associated with potential benefit in reducing the risk of MAS. While these studies have been made to shape our understanding of this potential risk, we identified several gaps in knowledge that we are trying to address with our work. So these studies have mostly been a small single sector or a small registry. And as you know, this limits the generalizability, it limits the power to detect uncommon events, and it also limits the statistical power to adjust for potential findings. Also, these studies included younger patients, and there's no big studies that looked at older adults that could have a cardiovascular events, especially with their underlying cardiovascular morbidity. There is also very little data on what happened to these patients after they're in the hospital, both with the occurrence of new post-discharge MAS and also with the effect of MAS on short and long-term mortality out of various remission. There's also not much data about the two new multiple myeloma CAR-Ts. Most of these studies have been conducted with the older CAR-Ts. So with these gaps in mind, we actually studied incidence and predictors of MAS with CAR-T in a large national real-world data using a contemporary cohort period with the more recent CAR-Ts, and we also focus on older adults over age 65. We want to study the association between MAS and cytokine release syndrome, but also with the recently recognized immune effector cell-associated neurotoxicity, or ICANS for short. And we also want to study the association between MAS and mortality and against not only the CAR-T admission, but also following discharge. So our study design is we used 100% of all the Medicare population, while the Medicare's virtual research data set to include the 100% fee-for-service beneficiary over the age of 65 who received CAR-Ts. We required the patient to have uninterrupted enrollment in Part A and Part B for at least 12 months before they received CAR-T, because we used that time frame to calculate their baseline characteristics. We established our cohort by identifying patients who received inpatient CAR-Ts using ICD-10 procedure approach. Our timeframe was much made in the beginning of 2018 to about the end of 2023, or a temporary period. And we, as I said, identified baseline characteristics, cancer time, primary cancer therapies captured from the Medicare cohort files and supplemented by ICD within the 12 months preceding the CAR-T administration. So we defined the MAS as a composite of the following. So acute heart failure, cardiogenic shock, acute myocardial infarction, cardiac tamponade, ventricular arrhythmias leading to cardiac arrest, complete heart block, and stroke. We excluded patients who had the same MAS level within 3 months preceding the CAR-T admission to try and be very specific and avoid having chanting a diagnostic protein classification. We also captured CRS and ICANS using codes that were available in the study period between 2021 and 2023. So we used standard statistical methods to use and construct our prediction depending on the association models with multivariate international exposure to another. Few caveats to our models that we did not include Bayesian arrhythmias in this initial phase. And we did that potentially for two reasons. The first thing is that we used it as a predictor variable, so we did not want to use the same variable as a predictor outcome. We also did not reliably think that the codes could reliably distinguish products in vivo Bayesian arrhythmias. We also did not put the cancer therapy in the models to avoid polylinearity with the cancer therapy. And we had CRS and IPSCs for the year starting 2021 to 2022. So we used these in the models only in those particular years. So a total of 3,292 patients received inpatient CAR-T. And you can see the trend over the years, how the CAR-Ts have been increasing rapidly. Initially, diffuse large B-cell lymphoma patients in that view were the most common indication for CAR-Ts. But then starting in 2022 onward, much of the myeloma patients have exceeded those. For these patients, we observed that there is a change in 5% of our patients. And when you look at the events that these patients have, heart failure was the most common event, followed by stroke, followed by academic and after that, MRIs, cardiac shock. This is the table point to comparing diagonal analysis of the patients who have MICs did not have Bayes. And the differences are the ones highlighted in kind. So patients with MICs were more likely to be older, were more likely to have a diffuse large B-cell lymphoma compared to other types of cancer, and were more likely to have more burden of underlying cardiovascular risk factors in previous analysis, with more hypertension, diabetes, atrial arrhythmias, CMD, heart failure, and cerebrovascular disease. When we constructed a multivariate model to predict any associations, we found that baseline cardiomyopathy or heart failure, baseline antiarrhythmia, and baseline cerebrovascular disease or stroke were strongly associated with occurrence in hospital days. And we also found that multiple myeloma patients had a protective demonstration who were prepared to patients who received CAR-Ts for their diffuse large B-cell lymphoma with the protective demonstration of 0.64. When we looked at the association between MACE and index mortality and MICs, we found a significantly higher patient mortality for those who developed MACE at 27.9 versus 2.2% with a very significant p-value. And we also found that people who had MACE had a longer positive stay compared to those with not also with a significant p-value. About 3.6% of our patients died when we were starting to process via extension. Where we adjusted for all the potential confounders of different baseline comorbidities and cancer type, MACE was still a strong independent predictor of patient mortality with an adjusted blood ratio of 85%. Into with the confidence that that's significant. Now looking at the association between MACE and cytokine release syndrome and ICANS, cytokine release syndrome occurred in 67% of our patients, being consistent with previous literature. What's very interesting is that unlike other studies that had a strong association between CRS and MACE, we did not observe such an association at the occurrence of MACE was not that patients who had or did not have cytokine release syndrome. We however observed a much higher occurrence in MACE with people who had IPSCs at 11.4 versus 4%, whereas we did not have IPSCs and significant p-value. Now moving after the hospital discharge, when we looked at 3,174 patients who made it to discharge, people who had MACE have much higher 1-year mortality compared to those who did not have MACE. The line is those who had MACE, the line you are using to not have MACE. At the 46% versus 25% for the 1 year or it was mortality. And this difference was still significantly positive when we adjusted for several different founders and baseline comorbidities and cancer type, just patent ratio appropriate that. We identified hypertension, atrial arrhythmias, and diabetes as independent predictors of 1 year mortality in cases of variation.
And we also similarly found that diffuse large B-cell compared to other cancers have higher 1-year mortality. So what about patients who did not have MEIS during their index admission? So we had 3,016 patients who survived index admission and did not have a MEIS clearance. 9.2% of them had a MEIS developed within the first 12 months follow-up. And from these, 47% died first year, and the median time to MEIS was 34 days. And heart failure was still the most common MEIS during follow-up, followed by strokes and repeated MIs. So certain limitations and considerations this is the retrospective administrative data. So there's always a potential risk of inherited founders and possibly diagnostic with misclassification. However, we used codes that have been very well validated in previous studies, and we also were very specific to not include patients who have a MEIS within the same group with 3 months preceding the CAR-T to try to reduce the risk of diagnostic code misclassification. We also don't have data regarding the cause of death, whether it's cardiovascular or cancer-related. This is some of the potential usual work that the NIH to do by linking these patients with death certificates and trying to get the cause of death. We also don't have data about the tumor burden and staging of that affects the other type. And this is also some of the future work we're doing by Medicare data that we have for this year in registry to try to stage and assess the tumor burden in these patients. We also need longer follow-up, especially for patients in silicon or other CAR-Ts, once large tumor from it. So in conclusion, in the largest Canadian national sample of over 3,000 older adults who were hospitalized with CAR-T, we found that MEIS clearance was 5% during the index admission and 9% during the first year of follow-up. We also found that patients with pre-treatment cardiomyopathy, late-stage arrhythmias, cerebrovascular disease, CAPS, and diffuse large B-cell were at highest risk of developing MEIS independent of other baseline characteristics. And unlike other studies, they said we did not observe a link between cytokine release syndrome and the appearance of MEIS in this population. When patients had MEIS, that was associated with longer hospitalization, increased in hospital mortality, and also increased 1 year or close mortality. So with this, I would like to thank you all and thank my unrestricted Cardio Oncology team in March, especially Dr. Muredach, my editor, and our extended collaborator. I'd be happy to take any questions that you can have.
Thank you for figures for the presentation. There's room for about questions. Anybody want to ask analysts?
How's that? Okay. Well, here's a question for Dr. Chun. I really enjoyed your presentation. And I think the conclusion is that being compliant with diabetes medications and having good diabetes control is particularly important in patients with cancer. We do know that in the general population and many other patient populations that people with chronic kidney disease. So is there any reason to think why this conclusion may not apply to people with cancer?
That we why these conclusions are not followed in declaration.
What did you address that question in people with cancer? I mean, wouldn't you expect that the findings would be the same, that being compliant with diabetes, even if you have cancer or maybe regardless of cancer, is actually important?
No, my conclusion is that taking care of diabetes, I think it is more important in cancer patients. And we understand that the cardiovascular risk is regulating in cancer patients, as our results said. And for the non-adherent thing, I just in the discussion section, I mentioned and non-adherence is more higher in the cancer patient. And with my very little clinical experience, I found that the cancer patients have a lot of pills to eat, and they are not taking care of any diabetic care medications. And then it's like this kind, people have a diabetes one thing and they can only focus on diabetic care, but if you have cancer and diabetes,
And the impact of diabetes is getting less and getting more or less attention about treating the glycemic control. And I think one of the cardiology analysts to do is that giving attention to this low impact of glycemic control should be given to cardiology to patients. And about why the cardiovascular risk is increased in cancer conditions, I think because the demographics are very different from the general population compared to cancer conditions. First thing, they might be over age, or they might have smoked before, and they might have more physical activity. And for example, like basic day, like T cells are more elevated, more activated, and more frequent in the cancer condition, and they might affect the cardiovascular risk and other things. And I think those are the differences between cardiovascular. Thank you very much.
Quick question here. And congratulations on the finalists. All presentations were excellent. And a question for Dr. is about the impact of proximity. Now we know that myocarditis and heterogeneous conditions can have different types of triggers. So do we know that if the impact of proximity changes where we're looking at checkpoint inhibitors of myocarditis compared to other causes of carditis?
Thank you so much for your question. And there are a lot of similarity between SCID myocarditis and other type of myocarditis for our patient, which we initially think this is giant cell myocarditis, but we didn't see large cell in our patient cardiomyopathy, and it only shows the lymphocytes myocarditis in the heart, and we treat that one. Very similar to myocarditis. So this is idiopathic myocarditis, and we don't know it's very significant.
Thank you.
So you might have your mentioned this. I think you're to do this that all day precisely and trastuzumab. But I'm curious, how much of that is an anthracycline effect versus a trastuzumab effect, and whether you guys are also looking at patients treated with non-anthracycline-based trastuzumab regimens.
Yeah, thank you for your question. So, all the population in our study were treated with anthracycline and trastuzumab. And so we were not able to stay with the cardiovascular is because of anthracycline or trastuzumab poly, the combination of the two. But the thing is that now, as I understand, as the breast cancer treatment has evolved, and now we are moving toward like a non-anthracycline regimen, right? So if we use non-anthracycline regimen, probably the incidence of cardiovascular will be even lower compared to the incidence in our study that we are using combination therapy. So I would suggest that our clinical implication that the routine echoprogram can be reduced is still relevant, because if you're not using both of them, the incidence of cardiovascular would be even lower, and we can slightly reduce routine surveillance.
Thank you. Then I'll just comment, I know we're kind of out of time, but Dr. Zach, it'll be really interesting to look at your data and look at actually mortality, how much is cancer related, how much of it is cardiovascular related. It sounds like you guys are on Travis is doing that work.
Yeah, thank you. I think this is a very good question that we are also hoping to answer, because it's very good to know what these patients, whether the mortality was cardiovascular related or relapsed or they got something else. It's kind of hard to do it from the database that we use, because there's the death is such a disparate as a net. What we're trying to hope to do is maybe get those deaths, link them to that certificate, and try to get the cumulative analysis of whether this was a cardiovascular related death, a cancer death, or some other cause of death. I think this is very important taking to the future.
Thank you.
Yeah, thanks.
Just very quick question, because
So, just to talk about that, thanks. Did you guys see a trend about counting in tolerance? Like what we're used to doing counting in terms of mates. Certainly our experience in that is Jesus time goes on better experience opinion with ours.
Yeah, I think this is a great question. I think if I'm understanding correctly, you're saying we observe the difference in trend in maze over time. Yeah, so we didn't do that specific analysis, but one of my thoughts sharing some of your thoughts is the association with CRS and how that have changed over time. I think the earlier studies have linked the occurrence of AS with CRS, and we didn't observe this trend. One of the possibilities that I was thinking about was that maybe we are now understanding that link better, so that people on HDL score were just using that earlier, people are recognizing that occurrence earlier, so these patients are receiving therapy earlier. So this could have negated that effect in our cohort over time. Because the same studies that showed that association also showed an association with reduced threat if you use the tocilizumab early on in grades 2, 3, or more in the CRS. The other thing that changed over time is how people define MACE, because that I think also changes your link with CRS. So we did not include HLA, we did not include venous thromboembolies. Previous studies mostly included these two conditions. And as the audience knows, there's a strong association with inflammation and arrhythmias, especially when people have over together with the leukine cytokines that continue actually arrhythmias, and they said CM14 is from the influence. So that could also explain a lot of association between CRS and our cohort. But that's a very good question. Could you just break down the gains occurrence by years? Most of our data have been used 2020 onwards. If you see the initial curves like 2018, 2019 had fewer patients. So that just trapped up over the last 2-3 years. But if we continue to follow these patients and include longer years, maybe 24 included, we might be able to do such an interesting analysis. Thank you.
So thank you very much, and please join us at the post-reception, the reception, where we will announce the winner. And then we'll just more people can announce the winner.
All right. So now is the best part of the afternoon. We really want to congratulate all 4 contestants for truly excellent presentations. They were selected among many abstracts, and the judges unanimously decided that these 4 will have the honor of presenting their data to us this evening. So before we announce the winner, we would like to hand these certificates to the finalists of today's presentation in this order. And the first presenter was Dr. June-Wha. And then the second contestant, Dr. Osman. And the third, Dr. Rhee. And last but not least, Dr. Mozneb. All right, so now we have the winner, and on behalf of Dr. Andreas Beyer, June Roach, and Joseph Atherall, we had a very difficult time deciding on who the winner would be, because maybe everyone is a winner this afternoon. In the end, like all competitions, we have to decide on someone who will be the gold medalist. And today, we have the pleasure of announcing that the person who's going to be getting the check later, is that right? Is Dr. Richard Osman. I think that comes to the end of the session, and we would invite everyone upstairs to level 6, where there will be a wonderful poster session for waiting.